Welcome to our first and not first brain metastasis meeting. My name is Matthias Guckenberger. I'm the Director of Outreach and Education for the Comprehensive Cancer Center. And I'm very proud that we're not just having yet another brain metastasis meeting, but that we're doing it this year in collaboration with the EORTC Brain Tumor Group. We're having a 2-day meeting because the developments you all know in brain metastasis are super rapid in all aspects, preclinical, translational, clinic. So many thanks, of course, to Matthias and to Emilie for co-organizing. Many thanks for the support. And without stealing too much of the time, we should have all the time in the world for research and discussions. Thanks for being with us. One short remark at the very beginning. You see that we have a number of sponsors and support. So, of course, as always, many thanks to our industry partners for supporting us in making this fantastic event possible. I'm looking forward to exciting talks, in particular to an exchange between neurology, surgery, radiation oncology, basic research, and lots of interaction. And with that, I want to hand over to Matthias Preusser, the chair of the Brain Tumor Group at the EORTC and co-organizer of the meeting. Thanks, Matthias.
Thanks. The next Matthias taking over. Welcome also from my side. Matthias Preusser is my name. I'm a medical oncologist based in Vienna. I'm the chair of the EORTC Brain Tumor Group, and it's my great privilege to co-organize this meeting today. We have a strong EORTC representation today with all the officers, Emilie, Giuseppe, our past chair, Michael, some committee chairs also in the room, including the CNS Metastasis committee chair. We were very proud to contribute to this program and looking forward to a great 2 days of basic and translational and clinical science in brain metastasis. Thank you.
Thank you to both Matthias, and so also welcome from my side to Zurich. I think you have seen the program. I hope you will have a lot of exchange during these 2 days, and I think we can already start because we have a busy day. So, Anna, please.
Thank you so much for giving us some extra time for the first session. That's very well appreciated, and we'll ensure that we have enough time for discussion. So we're starting with the first session, preclinical research, and it's my pleasure to chair with Tobias Weiss here from Zurich. And with no further ado, I will introduce our first speaker, Frank Winkler from Heidelberg, who will talk about the neural and vascular influences on brain metastasis. Thank you, Frank, for coming, and we're eager to hear.
Yeah, thank you so much for the invitation to this really wonderful conference. So brain metastasis meetings are always highly interdisciplinary, always fun. We met a long time in Marseille. This was great. Now the Zurich, I think, is not worse than Marseille. So really looking forward to these 2 days, and always great to come to Zurich, of course. And it's great that actually we won so much time. We gained so much time. So don't worry. I hope it won't be too boring, actually. So this is actually, and I didn't know actually that you introduced Anna, but I mean, this is clearly, if I would have known it, I would have chosen the very first slide as I did. So our group has dealt for quite a long time now, making use of this intravital photon microscopy, which allows us to study the very steps that are necessary to successfully colonize the brain for a circulating cancer cell, shown here, which is arrest by size restriction, then an active process of extravasation, and active, interestingly, not so much from the cancer cell side, but active from the host side, where the endothelial cells remodel, and then throw the cell out of the vasculature as published here 2 years ago. And then the cells need to maintain the strict perivascular position, so they need to rest or survive in a typical stem cell niche in the brain, which is the perivascular niche, and then they either co-opt existing vessels or induce early angiogenesis. And quite a lot of these cell lines, particularly lung adenocarcinoma, some breast cancer lines that we tested, not melanoma lines. Melanoma lines always grew cooperatively and not via angiogenesis. They are able then to outgrow to a clinically relevant macrometastasis in the brain. And we have kind of looked into many aspects, and a lot of these early step processes, stem-like features, some work Anna has done when she did some time of her postdoc in Heidelberg, but then also clot formation here that helps to these cells to arrest, MMP-driven extravasation, activation of the PA3-KmTOR pathway during the process of extravasation, which helps the cells then to survive in the brain, and VEGF-A-driven angiogenesis. And indeed, when looking into, and just for the shortness of time, I just can't go through all these angiogenesis data, but when looking into many mouse models, many different anti-VEGF-A, anti-angiopoietin 2 therapeutics, but also retrospectively into bevacizumab approval trials, it was clear that there is a strong potential of these anti-VEGF treatments to prevent outgrowth of these extravasated micrometastases, because they are arrested in a micrometastatic state. And bringing this to patients together again with Anna and others in Heidelberg, and this was a Vienna-Heidelberg collaboration. We retrospectively looked into patients, kind of really patients with advanced stage brain metastasis, no meaningful treatment option left decisions. Then we started with bevacizumab and could really see, I would say, in contrast to glioma, true responses, metastasis shrinking with bevacizumab, only showing that at least some of these, this was colorectal, this was breast, this was lung. So almost all patients got a clinical benefit from this treatment, and we saw, I would say, true responses of shrinking of these metastases, even in bevacizumab monotherapy, something you would never do outside the brain. So there seems to be a very brain-specific importance of VEGF-A for outgrowth, and also a role of bevacizumab for treatment, at least for heavily pretreated brain metastases, probably not first line, but at least as an option for advanced brain metastasis. So we know that we will learn a lot of this in the 2 days, that we have all now all kind of different molecular therapies against brain metastasis. So we can choose from a lot of those. And the big question of us was, well, is there maybe even a special twist to brain metastasis? The brain is, of course, full of neural cells, full of neurons. So are neurons themselves, are neural cells influencing this entire process? So brain metastases are by nature a multifocal disease. They diffusely colonize the brain, a bit like glioma. It's also difficult to treat them because the brain is a precious and a sensitive organ. And one question we asked in glioma, but also brain metastasis, is the brain itself involved in the malignancy of these diseases? Does it foster tumor growth? Does it communicate with the tumor? Do tumor cells use neural mechanisms? There are unwanted feedback loops and can be exploited therapeutically, which is more or less the field of cancer neuroscience. Indeed, in the last 15, 20 years, we have learned that in brain tumors, but also in almost I would say all extracranial cancers, the nervous system is crucial to drive initiation of cancer, growth, metastasis, and then resistance of these cancers. And in turn, and this is almost a little bit frightening, cancer can reprogram the nervous system. Cancer can track nerves. Cancer can reprogram the PNS to sustain its own growth. But even more frighteningly, the cancer can reprogram the brain and can really make circuit-specific changes to brain neurons, which then, via driving more anxiety, more depression, etc. in these mice so far. This is all data from mice so far, so it needs to be confirmed in humans. Then the cancer is driven again. So there is a harmful feedback loop apparently between brain plasticity changed by cancer, etc. So these are the mechanisms of nervous system cancer interaction. This is the last white paper. The new white paper is coming out now in 2026, soon in mid-April. Many of you might have seen that Doug Hanahan's mid-April paper is already out, so the new hallmarks of cancer. And hooray, cancer neuroscience is not a full hallmark of cancer, but almost a full hallmark of cancer there. So it's great for the field that Doug has appreciated that. And cancer neuroscience, so there's a long paragraph on cancer neuroscience where some of us were also kind of helping to write that. So in the end, what we discovered in gliomas was that they connect via via, in this case, membrane tubes to interconnect to a network, a communicating network that is constantly active in terms of calcium communication, where tumor cells are embedded in a communicating syncytium, and this syncytium is particularly resistant to therapy. And when tumor cells are embedded in this syncytium, radio chemotherapy does not kill them because of the higher higher ability to buffer problems like high calcium, etc. So the homeostasis of cancers in these networks is much improved. We, however, do know that many cancer cells can interconnect directly or via thin membrane tubes to each other, also separated with connexin-43 gap junctions, which are also necessary then to convey these calcium waves from cell to cell, because calcium and IP3, etc., is driving through them. And this is true for these longer membrane tubes, which we have discovered in brain tumors. Others have not discovered them in pancreatic cancers, and it's now increasing. They are very long-lived. They can live for hundreds of days between these cancer cells. The shorter-lived and thinner TMTs are also very important. They can be found in all cancer types. So there seems to be some neural behavior of cancer cells throughout entities. At least when you apply stress on them, and when you apply cytotoxic therapy, for example, then cells tend to do the thing that makes sense, right? To interconnect them to a resistant network. And this is not just, this is, I think, data again from Anna, I must say. So just to, from the Great Vienna Brain Metastasis data bank. So in certain cancer types here, we do see this very long membrane tube extensions in the brain here of a deceased patient. And we do see that also here's a breast cancer cell that also does the same here in the brain. And then dies, and then if two cells meet each other and interconnect with each other, they do survive. So again, kind of community is king. Community makes resilient, something we know very well from ourselves, our patients, etc. It's also true for cancer cells, unfortunately. So David Hausmann, a very gifted MD student in the lab, discovered that these networks are driven by pacemaker-like cancer cells, now again glioblastoma, which are residing in the hubs of these scale-free small world cancer cell networks and drive, so generate a beat of about 10 to 50 millihertz, which drives MAP kinase pathways in NF-kappa B in a frequency-specific coded manner. And this is providing a lot of resilience, because when you just do random damage to these networks, cells just die. And when you target periodic activity, you can really do a targeted, meaningful therapy against glioblastoma. So we are bringing this now to a clinical trial targeting this pacemaker cell with an inhibitor of this channel KCA3.1. This shows striking similarities to neurodevelopment. Again, there are pacemaker cells, also pacemaker neurons that drive neuronal network development. And this is certainly new opportunity. So our question was now, is that also true for brain metastases? Are these principles, features found in brain metastases? So the first discovery was yes, they do show vivid calcium activity when they have extravasated into the brain. Also co-activity, you see many co-activities here, oscillations, if you like, of cancer cells. Also converging to a 10 to 50 millihertz rhythm, where we know this activates MAP kinase, for example, in this frequency-specific coding. However, we didn't see pacemakers. So we didn't see this pacemaker mechanism. It was more a collective generation of these rhythms of these cancer cells. So this activity was clearly associated with growth. So the more calcium activity, the faster these tumors were growing. We then used calcium integrated technology to identify these pacemaker cells, to sequence them, and to understand their biology. And indeed, interestingly, this pacemaker-like cells had a clear enrichment of neuronal genes. So again, making this behavior, sorry, not pacemaker, please forget that. It's not pacemaker, but particularly calcium-active cells, not the pacemaker. Particularly calcium-active cells showed this typical neuronal behavior. So showing that high calcium activity in these cells is associated with a neuronal, at least expression phenotype. Immediate early genes were upregulated in these high calcium-active cells, which is interesting because Bendenine in Houston is showing this at conferences, not published yet. It's also showing now that neuronal input to cancer cells, so neuron to cancer synapses, are also upregulating immediate early genes. So it seems to be a converging biological mechanism of neural influences on cancer cells, that these EAGs are upregulated. There's a transcriptomic signature that we can derive from that, and when applying this transcriptomic signature, we can see that metastatic cells have a much higher calcium signature. Brain metastases have a higher calcium signature compared to extracranial metastases in patients, and metastatic cells have a higher calcium activity than the parental cells. As said here, you can see the parental line and here the brain metastatic line on the right. These cells, like glioma cells, also form a functional gap junction coupled network, which is interesting. Again, these coactivities, you see these oscillations here in these networks, and it is because it's kind of the connectivity decreases with neighborship, so to say. It's clear that this also depends on proximity in these networks, and it shows this functional coupling of the cancer cells. And here with the dye, you can see how this dye distributes in this network through passing through the connexin gap junctions. So when you target gap junction communication, then you can more or less silence communication in these networks, and you can then decrease cell viability and arrest cells in a G2M state. And this leads in the end to a therapeutic effect when you treat mice in a brain metastatic model where cells are injected in the heart, and then we follow them up in the brain. This is now melanoma. The same is true for breast cancer, also lung cancer. We can quite significantly decrease outgrowth. So a completely new idea how to tackle brain metastasis via drugs that are actually available. MFA is a drug that is already given to patients based on our discoveries in glioblastoma. A German trial has started, MACMEF, where patients with recurrent glioblastoma are treated with MFA together with temozolomide to make temozolomide more effective. And so this is already in the clinic, so it can be nicely used also for brain metastasis trials. There are probably much better gap junction inhibitors than MFA that really enrich the brain. We are testing them at the moment, and when this is successful, I think we have an excellent, very well tolerated situation for moving forward for a clinical trial. This is just the graphic summary. So in glioma, we have this more, well, I always tend this hierarchical communication. I always call it Kremlin style, but I think now we can also call it White House style, where we have these kind of pacemakers who dictate what's going around them in the entire organism cancer, and in other brain metastases, melanoma, but also in lung cancer, breast cancer, etc. We have this collective generation, this more democratic European Union style generation of rhythms and activity, and this is upregulating or associated at least with neuronal expression phenotype and also these immediate early genes. So we have talked about all these cancer intrinsic neural features, but we also discovered that there are neuron glioma synapses that stimulate glioma growth and invasion, where indeed remote and particularly also increased neuronal activity, like in hyperexcitability, which is typical for brain tumors in the environment. These tumor cells are constantly activated by neuron to cancer synapses, excitatory glutamatergic AMPA receptor on the cancer cell side. It's a specific calcium permeable AMPA receptor that also then generates this calcium activity. So it's a second big generator of this calcium activity in the cancer cell networks, next to this cancer cell intrinsic generation of which I've shown you before. Glioma brain metastasis, and this drives proliferation. Again, is also invasion in these cancers. It's very present in all samples that we investigated. So it's kind of, there's not one patient sample where we don't find these neuron to cancer synapses. Here in the patient samples where we identified tumor cells with 5-ALA, we could nicely see here excitatory postsynaptic currents, which shows a functional electrical coupling, synaptic coupling. And you can see that most of these synapses are located on these long membrane tubes in glioma, which makes these membrane tubes, which gives them a certain dendritic function for the cancer cell. So because we then moved on and saw that even on rapidly invading glioma cells, we could find these neuron to cancer synapses here, typical synaptic cleft, presynaptic vesicles, etc., postsynaptic density, and again EPSCs, and neuronal activity. So increase of neuronal activity by an optogenetic stimulation here, general-rhodopsin, increased also glioma invasion. So again, neuronal activity, well, thinking, whatever the brain does, is always stimulating cancer cell growth, but also cancer cell invasion. Our question was now, is that also operational in brain metastasis? So Doug Hanahan, mentioned before, Doug published back to back with our publication about the glioma neuron to cancer synapses, which we did back to back with Michel Mongi's lab in Stanford, and we didn't know we know very well of each other's work, so we have kind of developed this story quite back to back together for years. But we didn't know of Doug's work, and he showed that in breast cancer brain metastasis, macrometastasis, the cancer cell sits in a perisynaptic position and is not forming a direct synapse with neurons, but is sitting in a perisynaptic position and listens, so to say, to spilled over glutamate from the synaptic cleft. And here, NMDA receptor, so the other glutamate receptors type, is constantly activated. This drives brain macrometastasis growth in breast cancer. So our question was, are these direct synapses also found? This is kind of a wild guess, right? So why should a neuron form a synapse with a completely non-neural cell? You can still argue glioma cells have a neural origin. That's true. So is that possible at all? And indeed, we did see first that directly after extravasation, this activity starts. This was not a proof, because I've told you that there is also cancer cell intrinsic calcium activity generation. However, when we then compared activity in an awake state of the brain and in a deeply anesthetized state of the brain, so with very downregulated neuronal activity, we could see that there was sleeping or deep anesthesia is decreasing this activity, which kind of gave us certain first hint that there might be a direct neuron to cancer interaction. And again, calcium activity is correlated with brain net growth, and indeed then using correlative light and intravital photon microscopy together with 3D electron microscopy, we were able to locate, even on this just extravasated cancer cell sitting in this is red is blood vessel, green is cancer cell, large nucleus here, you can see plenty of presynaptic boutons from normal neurons sitting on these cancer cells. Again, cancer cell postsynaptic like in glioma, and this is true also for melanoma and lung adenocarcinoma. So a feature that is really present in all of these cells that are surviving in the brain. Again, EPSCs. So, and again, when you inhibit AMPA receptor by signaling by CNQX, you can really silence these synaptic interactions. AMPA receptor is highly expressed in brain versus in vitro conditions, and in our brain metastasis cohort in this German Prevent BM consortium, we could also find strong and robust AMPA receptor expression in these data sets. And interestingly, when you look at this cancer cell here in the brain, you can see that they really extend very long membrane, or not quite long membrane tubes into the tissue. Again, a little bit like these tunneling nanotubes or tumor microtubes. And these synapses are sitting mostly on these membrane tubes again. So similar to glioma, by extending these membrane tubes, cancer cell can receive much more synaptic input, which is of course the reason why neurons extend dendrites to also receive meaningful synaptic input from the neurons that they want to receive input from. Here you can see it again. And when we now either genetically interfere with AMPA receptor signaling, or when we give perampanel, which is a anti-epileptic drug that is quite active. I like to give it to many of my patients with brain tumors who are not responding well to other anti-epileptic drugs, because it's the only postsynaptic active anti-epileptic drug that is particularly active, because often when you have a glutamate overloading, which is frequently the case in brain tumor situation, and tumor cells produce glutamate, so this will then explains, I think, why you have often a very strong effect of perampanel on tumor-associated epilepsy. However, we were not treating this here. We were kind of trying to inhibit the synapses, and indeed we could decrease brain metastatic load and here in these animals. So opening up another avenue of how we can maybe better treat brain metastasis in the future, early steps and later steps. So in the end, what I've showed you is that there are not just this indirect perisynaptic pseudo-tripartite contacts between neurons and cancer cells shown by Duck's group, but there are also direct synaptic neuron to cancer interactions, AMPA receptor, not NMDA, and this starts very early after extravasation, and it's not just important for the growth of the macrometastasis. So this is kind of a little bit, quite wild that neurons apparently are so promiscuous to form synapses to all kind of cells, but it is what it is. And it seems that the brain metastatic cascade is governed by a sequence of neuron to cancer synaptic interactions, which is interesting. I mean, synapses of different kinds are, as you know, involved in the full function of the brain, and it seems that cancer can also be driven by different forms of neuron cancer interactions. There's potentially also a role for GABA synapses, potentially also a role for serotonergic, adrenergic, dopaminergic synapses. This is something that we are following up on. And then the question is, what about other cancer types, other organs? The quest is open now, and yes, there are first data suggesting that in pancreatic cancer, in several other cancer entities, gastric cancer, there seem to be neuron to cancer synapses. They need to be, however, fully characterized to be sure. You cannot give a talk today about anything in cancer without having a little bit about immunity. So I added this here because I think it's interesting. We have found that indeed, in order to reach melanoma brain metastasis, which is here in red, T cells shown in green here need to do one thing. They need to extravasate at a very specific spot, not in the tumor, not in the superior sagittal sinus region, but in a very distinct anatomical structure, which we call peritumoral venous vessels. These are pre-existing vessels of the brain where T cells roll, extravasate, and then they use these PBBs as highways to the cancers. It's an ICAM1 LFA1 dependent mechanism. And when you kind of downregulate that, they are not doing this anymore. And it's interesting that this is really the sole highway to cancer cells for T cells in the brain. And in order for checkpoint inhibitors to work, and they work pretty nicely in this model here, this needs to be functional. So now, from a cancer neuroscience perspective, our question is, of course, is the brain itself modulating T cell activity, T cell behavior, anti-tumor T cell effects? This is relevant question. It's interesting that nobody has really looked into that. You would think that the MS crowd and many other neurological scientists have looked into the question, how is neuronal activity itself regulating the function of immune cells, which is a very low-hanging fruit in a way, because there is a strong overlap of immunomodulatory or immunostimulatory or immunosuppressive signaling in immune cells and neurotransmitters, neuropeptides, etc. So T cells have a lot of receptors for neurotransmitters, for example, etc. So it's kind of a little bit weird that this has not been looked into. So we decided to use this model to look into that, and this are the first data, and we are now kind of following up on that because it's interesting that, again, when you have an awake state versus a deeply neuronal downregulated state in anesthesia, you find striking differences of T cell behavior here, migration velocity in these perivascular vessel spaces. So we need to understand what that exactly means, but apparently there are effects that are playing a role in extracranial cancers. People have found by and large, that innervation of the peripheral nervous system is down-regulating anti-tumor immunity. So when you denervate or when you inhibit beta-adrenergic signaling or sensory signaling, CGRP inhibitors, etc., you can make anti-tumor immunotherapies way more effective. We have shown this in collaboration with Andreas Trump team last year for pancreatic cancer. So no effect of checkpoint inhibitors in pancreatic cancer whatsoever in the models, but when you denervate the pancreas, suddenly you get quite a striking effect of checkpoint inhibitors against pancreatic cancer. And the question is that also true for brain tumors? So it's very interesting. We don't know yet. There's also one hormone that catched our attention, and so neurotransmitters, neuropeptides, hormones seem to play quite a significant role also in the brain. So this is ongoing, and we are doing this, others are doing this at the moment. So let's see, maybe we can at some point apply pharmacological or even other neuromodulation to influence anti-cancer immunity in the brain. That would be, of course, a nice outcome. So I showed you that the early steps of brain metastasis are driven by cancer cell vascular interactions. So we have new targets for brain metastasis prevention, anticoagulants, mTOR inhibitors, etc. The importance of angiogenesis for late and early brain metastatic outgrowth. So anti-VEGF-A therapies, bevacizumab, certainly also anti-Ang2, not so much developed, unfortunately, anymore, are brain-specific agents with preventive and therapeutic activity. I showed you these neural-like features, which include these collective calcium communications, which drive brain metastasis, which is the cancer cell intrinsic neural mechanism, and calcium inhibitors, which are, of course, as you know, widespread in the clinic, but particularly gap junction inhibitors are certainly interesting new anti-metastatic drugs. And our very first results with these gap junction inhibitors show that they apparently can also dramatically decrease extracranial metastasis. So it's something that is apparently not just operational in intracranial metastasis. Then I showed you that now we discovered these direct excitatory synapses between neurons and brain metastatic cancer cells from the very first steps. Certainly perimpanel as an interesting candidate for brain metastasis prevention, potentially therapy also. And then these perivascular veins or venules that are anatomical structures that allow T-cell homing and brain metastatic lesions in an ICAM1 dependent manner. So also implying certain ideas how you can translate this or how you can use this for improved anti-brain activity. So this is the article in Press which I mentioned. So the mechanisms of neurocancer interactions are growing. Now we also see I think brain-body communication or brain-body cancer communication is a super interesting area of research at the moment, circadian rhythm and cancer. So please don't post this so far. This is not non-postable as like the others non-published work. But then of course, how is metastasis itself influenced? I told you that. So there are really kind of interesting new ideas. And then what is the downstream effects in cancer cells? Which is also, I gave you some insights into that. So this is what I touched today, and but I think the entire field is moving forward very quickly. It's now on us to avoid some mistakes of the past. Of course, we will also make mistakes when we kind of try to translate this further. In glioma, we have several clinical trials already based on our discoveries. In brain metastasis, not so far. So I hope we can launch at some point also a brain metastasis trial, which disconnects these functional networks. A drug that disconnects the networks and then could be used. So we see surprisingly little side effects of these interactions, potentially because we are attacking neural features that are more relevant for early stage nervous system development and not so much for the adult brain. And then this requires certainly good biomarker development, also potentially for selection of patients, for monitoring of treatment effects. We need to make sure that we're hitting the target in our trials, and we need to certainly, this is of course something that we are all doing now in neuro-oncology, that we are collecting a lot of tissue, try to understand what we're doing, and then try to develop this further. A lot of potential actions and potential sites where you can inhibit these crucial neural cancer interactions or neural influences on cancer to improve control of these tumors, potentially in combination with other therapies, but also to make life better for patients, to decrease unwanted neurological side effects of cancer. In the beginning, I just touched on that. So a big thank you to all who did the work, the group, and all the collaboration partners. And I just want to close with one teaser. So we are now in the final phase of the new S2K guidelines for brain metastasis. So when Michael Weller did this, I don't know, 15 years ago, 20 years ago, this was, I would say, a smooth linear pleasure drive. Now it's not that anymore. So now you have 50, 60 specialties that are demanding to contribute to the guideline, and we are at 160 pages. I think if I would not be so strict, we would have been at 400 pages now. But it's still digestible, I would say. So we try to avoid to get too long. We have last fights with some specialties that also want to join, so end of the month we will now hopefully agree then on the last version that we I think have now a quite comprehensive overview of how brain metastasis should be treated, and I'm not sure that I will do this again, but it was a great experience, and I think so. We are aiming to publish the essence of this work this is all German, of course, but we will publish this in English, hopefully then in an article later this year, and so that we can make the most of it, of this endeavor for the entire field. And that's the end now. Thank you.
Thank you, Frank. What a start. And lots of unpublished data. Even though we're good in time, we're already advanced, but we have time for one burning question from the audience. Yeah, Matthias.
So great stuff as always. Have you already thought about clinical trial designs to translate this into clinical settings?
Yeah, that's a great question. And of course, trial designs in brain metastasis seems to be even more challenging than glioma. So it's, as you know, dealt, and together we have dealt quite for a long time to try to convince companies to go into brain met prevention trial design, because I think many of these activities are strongest in early stages and can have the most effect. However, this is challenging. It requires a lot of new thinking in the company. So I would probably not go this valley of death path again, and rather move to something where we believe we will see clear effects on brain macrometastasis, and then do the more traditional way and see whether we can achieve single agents effects maybe in advanced brain metastasis. That should be possible, particularly since our data shows that the more advanced these cancers get and the more treatment they receive, the more the connectivity increases as an adaptive mechanism of resistance, at least in glioma. We still need to show this in brain metastasis, but first data goes into the same direction. So that would even make it plausible to go more to the later stages, which is not a good idea maybe for immunotherapy trials and for other trials, but maybe for this, it's a kind of a probably plausible way.
Thank you so much. One last question, because we will have a pro and con in animal models. And so far, what we see from you here, this is all animal model, as far as I remember from my early days. Do you see some of the activities also in spheroid or cell culture models so that you could spare the animal model?
Yeah, I mean, of course, the big 3 R's, etc. So we always try to. So we have difficulties to grow our brain met cell lines in our brain organoids. They're not growing well there. And then, of course, as you know, the entire metastatic cascade, extravasation, selection of cells which can extravasate, and then selection which can survive, and selection, blah blah blah. All this is bypassed, of course, if you don't have the full metastatic cascade. So in glioma, we are super happy with our organoids, and we go. So I need to push the team to go back to animals because they have certain advances too. But in brain metastasis, I would still go to the. I mean, for the angiogenesis, for example, everything we found in the animals models was very much in line with what we saw in the clinical data, which I told. So this kind of analysis of the approval trials, what I was talking about. So I'm not. I'm of course animal models are animal models, and nothing is perfect. But I would be quite optimistic that there is a good prediction. For the clinic, if we do it right. One thing I think we need to what you need to think about, and this is my last sentence in the field, is we should really try to move away from these long serum cultured boring U87-like cell lines, which we have done this in glioma research. We also need to do this now in metastasis research. So we need to work really more and more with primary cell lines, cultured under stem-like conditions, which reflect much better the tumor heterogeneity. The problem is that not so many people are doing it, and there are not so many robust models. So we really need to do this as a field, I would say. I think that would be my 5 cents.
Thank you so much, Frank. And that's a good cliffhanger for our pro and con session. And I give over to Tobias Weiss to introduce our next speaker.
Exactly. With this, we move on. It's a great honor to introduce the next speaker. So Michael Levesque, he's a professor of dermato-oncology here in Zurich, very well known for pioneering profiling work in melanoma. And he will speak on biological monitoring of melanoma and response prediction. Mitch, great to have you here, and we are looking very much forward to your talk.
Great. Thank you for the invitation to be here, and I'm really happy to talk to you about our work today. So I'm normally a melanoma guy, work on melanoma biology and biomarker development, so this is not my normal audience, but what I want to do today is tell you a little bit about how we want to develop biomarkers to better predict response to different therapies in melanoma to, in the end, prevent brain metastasis. So these are my disclosures, and the most relevant one is Oncobit, with whom we've worked to develop some digital PCR assays that we have one slide on later in my talk. So why melanoma? So I think most of you know that melanoma is highly prevalent in the brain. So 30 to 40% of melanoma patients develop brain metastasis along the disease course. About 75% of melanoma patients present with a brain metastasis at the time of death and brain autopsies. And after the diagnosis of melanoma brain metastasis, the median OS is about between 3 and 13 months. And interesting to me is that there's a particular tropism, and this audience, I think, understands this better than I do. But melanoma has a preference for the frontal lobe, and in particular the perioatlantic area of the brain. And this is something that'd be interesting to study, and something that we'd like to go into in the future. So I think everyone knows that the success of immunotherapy in melanoma has been dramatic over the last 15 years. In fact, it's probably the best case of precision medicine over the last decade. And I think this graph shows this nicely from last year, that the 10-year overall survival rate for melanoma patients is about 52% with the combination checkpoint blockade, and stage 4 melanoma, stage 3 melanoma. A recent study from Christian Blank and Georgina Long showed that in a neoadjuvant setting that they can dramatically improve over the adjuvant setting in preventing further metastasis in stage 3 melanoma patients. But I think the bad news from this graph is that about half the patients are not benefiting, especially in the stage 4 setting. So can we develop better biomarkers that are more sensitive and also accurate, that allow us to predict the response at an early stage to change treatment for these patients and then ultimately prevent metastasis to the brain. So what I'm going to tell you about today is a couple of different biomarker studies that we've initiated and published, and then some work on the tumor microenvironment that's not published that I think we're all very excited about in the coming months. So just to catch everyone up on what we've been doing in the field, when we first started working on this topic, we have access to a lot of melanoma patients at the clinic in dermatology here. And so what we simply did is use the CyTOF approach, so single cell proteomics using 3 different panels, so 40 proteins per panel. You hear more about this today. And we've analyzed the immune compartment of patients before treating with the immune checkpoint and then also after 3 months of treatment. And we simply asked, is there some component of the immune microenvironment that's predictive of response to ICI or changes in response to ICI and the responders versus non-responders? And fortunately, we did find a signal. In fact, actually, we were expecting to see some dramatic changes in the T cell compartment at baseline, and we didn't observe this. In fact, what we did see is in the myeloid cells themselves that there was an enrichment for at least 16 markers of classical monocytes that were already enriched in responders at baseline before ICI treatment, and then were further enriched upon the 3 months of treatment to ICI. We could also confirm this by flow cytometry in a double-blinded experiment and show that the classical monocytes are highly enriched in the baseline samples of patients before treatment to ICI. And we could determine a cutoff based on the patient cohort that we generated here. That was that classical monocytes that are present above 20% in baseline patients, these patients have a very bad prognosis compared to those with a low classical monocyte count in the blood. So this was gratifying that we could actually get a signal from the CyTOF analysis. But we want to see that there was a way to confirm this in another type of orthogonal assay. And so what we did next is that we did single cell sequencing. This was now in the last couple of years that the technology has really expanded dramatically. And so we did the same type of study, except in this case, what we did is just CD45 sorting. So looked at just the immune cells in the patient blood and in the tumors, because the reviewers from the previous paper said, oh, that's great that you can predict in the blood, but what's happening in the tumor? And in the end, we wanted something that was minimally invasive, so we want to focus on liquid biopsies, but at the same time, the action's happening in the tumor. So the question is, what are these monocytes doing that are already present in the responder patients at baseline once they get to the tumor? So first point is that we could actually confirm that the classical monocytes based on single cell RNA sequencing were highly enriched in the responder, and this also in the tumor-free patients. So this is stage 4 or stage 3 melanoma patients. And these are patients receiving either neoadjuvant or adjuvant immunotherapy. And so this was gratifying that we could validate our initial results and that responders really had a high proportion of classical monocytes in the blood at baseline. But the next thing we did, which wasn't possible in the previous study, was to really look in the tumor microenvironment itself, because for all of these patients, we had matched PBMCs and CD45 sorted cells from the tumor. And we asked simply what is happening to the myeloid cells once they go from a classical monocyte in the blood to the tumor microenvironment. This is now just a static picture. So what we use is something called a velocity analysis. So it produces kind of vectors of differentiation of cell types within a microenvironment. And so what we did is looked within the PBMCs of the patients at the myeloid compartment, and then once those cells were in the tumor, how did their differentiation status change in responder versus non-responder patients? And what you can see on the top are the responders and the tumor-free patients, and the bottom, the non-responder patients. And there's actually quite a dramatic difference between the myeloid cells in those two different microenvironments. Whereas the responder patients, they both have a translational monocyte population, but in the responders, these start to express CXCL9 and then polarize to M1 like phenotype. Whereas in the non-responder patients, they have these transitional monocytes, but then there's an enrichment for this more M2-like macrophage. And this confirms also other published data from different groups showing that an M2 enrichment or M2-like macrophages in the tumor microenvironment are immunosuppressive and then prevent immunotherapy response. So we can start to see these transitional events, and then also the molecular features that drive those events at an early stage. And so the next thing we did is ask, well, these are now two separate studies in different patients. Can we use this tumor profiling consortium that we developed in Zurich and in Basel to where we developed a lot of molecular single cell functional studies on a small cohort of 126 melanoma patients. And we asked, can we use all of these multi-omics data to define better biomarkers to predict response? And some of this work has been published. And so I'll just quickly tell you the two different strategies I'd like to discuss this morning would be additional blood monitoring biomarkers. Can we use something simpler than a CyTOF type of approach where it's powerful but it's expensive and a bit difficult to implement in a clinical setting? And then the second is the spatial analysis of the tumors, the microenvironments, to do a much more high resolution analysis of potential predictors. So the first result of this was that we developed a melanoma NGS assay. So these are 16 melanoma genes that we then put onto a targeted panel for NGS assessment. And then we measured the ctDNA, so the circulating tumor DNA fraction in the patients at different time points. And now this cohort is 126 sort of non-selected patients, either stage 4, stage 3. Some of them are baseline immunotherapy, but some also are second, third, fourth, fifth line in some cases. The idea is to not have a clean, highly selected cohort, but a more real-world setting. And what you can see from the circulating tumor DNA measurements already that the fraction of circulating tumor DNA as assessed by this NGS assay is significantly different between stage 4 and stage 3 patients. And so already that gives us some hope that we can identify a signal of progression in these patients. And interestingly, when we look at the different histological subtypes, the cutaneous melanoma patients have a high fraction of ctDNA, as you would expect from mostly stage 4 patients. But the ocular melanoma also had very high shedding. This was not observed before, and something that we followed up on in the next study. What we can also see is that regardless of treatment line and the stage of the patient, that we could see already the ctDNA fraction, it was predictive of response to the next line of therapy. So be it first line, second, third line, regardless of the therapy line, it was predictive of the 3-month PFS within just that one ctDNA measurement. It was not predictive at 6-month PFS, and this and some other data I'll show you in a minute suggests that frequent monitoring is really necessary to assess ctDNA fractions, because these change very rapidly over time. And if we now, those were static measurements that could predict the next line therapy regardless of therapy status or treatment line. But the next thing we did is just ask the deltas. So can we identify a difference in those ctDNA fractions that are going up versus decreasing or not even detectable? And at 3 months, you can see this is significant. So this is now, once again, at any stage of treatment. So if there's no detectable ctDNA at either time point, then this is enriched for CR, so complete responders to any therapy line, and also partial responders. If there's a decrease of ctDNA in this time period, then there are also complete responders and partial responders, but then a larger proportion of progressive disease patients. If ctDNA is increasing at these 2 time points, then there are very few complete responders, and the majority of patients have progressive disease within 3 months. So the idea here is to try to make a simple test that would allow us to predict response regardless of treatment line. Unfortunately, it doesn't work at 6 months or 9 months. So this baseline and first follow-up ctDNA assessment doesn't really predict 6-month response, but it may in a more pure treatment setting, so first line or second line. But in this case, in an unselected patient cohort, there was no prediction ability. And so what the next thing we did is that we worked with the company Oncobit to develop a much more sensitive assay, because our hypothesis here was that, well, NGS is great, but it's expensive and it's not extremely sensitive. And so we developed a digital PCR assay that just target the hotspot mutations in the patients, and it's very easy to implement and very fast turnaround time. And so we can see in a cohort of metastatic uveal melanoma patients, 75 patients, we measured over 655 samples. This was published just last month, and I'll just show you this one slide to show that we can also at first line predict PFS. and the median PFS in patients with high ctDNA fraction at this baseline measurement was 2.7 months versus a low fraction of ctDNA, a PFS of 13 months. So we see a very clear separation between potential responders, non-responders in a cohort of patients with uveal melanoma that are effectively not treatable, so very, very low efficacy. OS is also quite strongly predictive, 19-month OS prediction with high ctDNA OS versus median not being reached in the low ctDNA population. So with that, I'd like to summarize the liquid biopsy part of the talk, which is to say that we think that very sensitive assays to measure ctDNA at multiple time points are prognostic for recurrence, survival, and treatment response to ICI and actually targeted therapy or any other therapy that we could measure. But for ctDNA, at least that you need longitudinal measurements because the fraction changes rapidly over time. You need to have multiple measurements to be able to assess not only treatment response but also pseudoprogression, which is a big problem in melanoma, at least in the first 3 months. And so with that, I'd like to switch now the last few minutes of my talk to tell you a little bit about some unpublished work that we're doing to investigate the melanoma tumor microenvironment, because while liquid biopsy is very appealing because it's minimally invasive, it's easy to do longitudinal sampling, the actions happen in the tumor. And so the question is, can we use more sophisticated spatial transcriptomics tools to better understand the interactions in the tumor microenvironment that are predictive of response? And so that's what I focus on now. In particular, you'll hear more talks about TLS this morning. We're interested in the tertiary lymphoid structures because other papers recently have shown that they are predictive of response to ICI. And we thought, well, since we have some spatial transcriptomics data from this tumor profiler cohort, it's an opportunity to perform some high resolution analyses of this question, because all of the papers up until now have been based on bulk microarray or bulk RNA sequencing data, and we think that there's now really a lot more resolution that could be provided by single cell technologies. So just as a quick reminder, this is a lymph node. This is where antigen recognition and immune activation happens, and a TLS is a tertiary lymph node, so it's a lymph node like structure that occurs in tumors and other wounded material, and it's identified in several cancer types, and as I said, it's been predictive of immunotherapy response. But since all the published work up until now has been based on bulk data sets, and the bulk data sets essentially ignore the spatial interactions in the tumor microenvironment, we said, let's actually look more carefully at those cell-cell interactions and ask if we can develop better TLS signatures. And I think this illustrates this very nicely. This is a Swiss artist named Urs Verli. It's entitled Kunstaufräumen. And his whole profile is that he takes art pieces and then deconstructs them, cleans them up. And so this is what we've been doing up until now with single cell biology. We digest a tumor, then we perform single cell analysis, and then we make compartments of the different cells. We did this with CyTOF, we did it with other techniques. But when you make these parts lists, what you miss is the interactions between those parts. And especially with something like a TLS, the spatial arrangement of those parts is really essential to understanding the biology. And so we think that this illustrates that nicely, that having a parts list is only the first step to understand those interactions. And so what we did is took the tumor profiler cohort, and this is now 76 tumors, melanoma tumors, in healthy lymph nodes, and we profiled them by 10x Visium technology. This is spatial transcriptomics, and this is work that we did together with two groups in Zurich, so Gunnar Retsch, computational biologist, and Victor Kurzer, who also funded part of the study. And the idea was that let's now look very carefully at healthy lymph nodes, so lymph node follicles, as well as lymph node metastases, so melanomas that migrate, metastasize to the lymph node, and non-lymph node metastases. And this is something that hadn't been done before, because essentially they conflated the signal of the TLS with metastases that went to the lymph node. And we want to actually identify functional follicles this way. So we identified a signature this way with the 10x Visium workflow. And then we applied it to positive and negative TLS samples from the cohort, from the Visium data. And so this is now an example of what we did. We did an unbiased signature search. So this was to identify spatially coherent transcriptional domains. So there's no human involved in this first phase, just to find domains in the tumor that have some kind of transcriptional spatial coherence. Fortunately, these domains here overlapped with the manually curated domains from the histopathologist in yellow. And so these are now lymph node follicles that we then derived a signature from our two follicles to get a very specific germinal center activation signature that is not just a whole lymph node. The next thing we did is that we did something similar for the melanoma tumors. So we took all of these 12 melanoma tumors with TLSs. We identified spatially coherent signatures, and then we overlapped them with the lymph node germinal center signature. So now we want to, by doing this, really identify the most functional lymph node signature within the melanoma metastasis, and also to do that in a clean way with extra and intra lymph node metastases. This is now an example of the validation set. So we took this as one patient with no TLS in it, CD20, CD21 staining to mark the B cells at different stages, and then you can see that there's no TLS from these two genes, but in our signature, our 8-gene signature is also negative. In a patient that's positive for CD20 staining and CD21, we can see that a really nice TLS signature from our study, which was good confirmation that we can identify really specifically these TLS. And then when we look at the whole cohort, now this is Visium data from 37 tumors, we can see also a strong enrichment for those that were manually curated as being positive. Now, I have to just say that this is not really the perfect gold standard, because they're just curating them based on morphology and based on CD20 and CD21 staining, which we know are not really sufficient to identify at least immature TLS. So we're confident that at least we can identify the mature ones, and we think that we're also getting a signal from the immature ones. So what we did is then validated this by multiplex immunohistochemistry, and you can see that these are all of our markers, and they really nicely mark the germinal center, but importantly, not just the B cells, but also some of the T cells and other cells in that tumor microenvironment. So then we compared our signature to the published signatures that are the standard of the field. Now these are just some examples here. This is a non-nodal metastasis, the CD20, CD21 staining. This is our signature here. So you can see that it's a bit broader than these two stainings. And then the published literature signatures, you can see that they're very broad. Actually, they're not very specific, so that they don't mark in any specificity these domains. And if we look now at intratumoral lymph node mets, our signature is very strong and specific, whereas the published ones are not very strong in the intratumoral met. So it means that the published ones, as expected based on their method, don't distinguish between a lymph node and a metastasis in a lymph node and the TLS that are formed inside the metastasis in the lymph node. That's an important distinction. So then when we take our signature and then try to validate it in the public data sets, TCGA, for instance, all the signatures perform well, because this is the melanoma TCGA that they all tested on and validated on. So we're confident that ours is also very strong. But if we remove all the lymph nodes now, and I think this is the crucial step, we remove all of the metastases that were in a lymph node, what you can see is now the Cabrita signature no longer is significant, because we think that because it was trained on lymph node mets, that it's actually mixing up the other genes that are involved in lymph node development that aren't specific to the germinal center. Whereas the TLS signature that we generated was quite strong and significantly different between in terms of predicting overall survival in this cohort. And then if we broaden it, now this was all up to melanoma, then we just applied all the signatures across the entire TCGA, and this is our signature here. You can see that in orange, green, or blue, these are the cancers where our signature can predict survival of the patients, and there are 7 of those where our signature is predictive, and then the other published signatures are not predictive in most of the cancer types. And what's very interesting, and maybe also specifically for this audience, is that although our TLS signature is positively predictive of response to ICI, to targeted therapy, but also overall survival, in some cancer types it's anti-correlated, and that's the case for low-grade glioma. This is the case for uveal melanoma. You can see a very strong anti-correlation between TLS and survival in these cancer types, and also LAML. And one example from lung cancer, we can see when we apply these signatures, I think this illustrates the point that the difference between our approach and the published ones is if we look now in lung cancer at CD20 and CD21 staining, we can in our signature can distinguish quite nicely this localization of the B cells, whereas the other signatures really are not specific to the TLS whatsoever. So that's why we think we can get this increasing resolution because the approach allowed for a more high resolution understanding of the interactions in the tumor microenvironment. So with that, I'd like to close by saying that we still think that liquid monitoring is a very important way to predict response of patients, to monitor patients over time with the goal of improving their therapies. So if we can detect ctDNA or classical monocytes, for instance, at an early stage and monitor them, then we can give patients better therapies that prevent brain metastasis. Ultimately, that's the goal of this kind of approach. We now, and I think I don't have to convince this audience, that single cell analysis helps us to understand at a very high resolution level the interactions in the tumor microenvironment that was shown nicely in the previous talk. But without the spatial context, I think the intravital imaging is one really important component of this to look in real time at the interactions there. But the spatial analysis allows us to really, in an unbiased way, to interrogate those interactions in this case, at a whole transcriptome level, so completely unbiased. So it's clear that this has to be an essential component to our understanding of those interactions. So with that, I'd like to close by thanking my research group, especially Victor and Gunnar, for access to the Visium data that they produced on the tumor profiler cohort, the biobank, the tumor profiler consortium, and most importantly, my team that did the spatial analysis for the last study. So two senior researchers and then two very talented master's students who did all the work that I told you about the TLS, and I hope that we submit that soon. So with that, I'd like to thank you for your attention, and I'm happy to answer any questions.
Thank you very much, Mitch, for a nice talk and also great teaser for the Pro-Cons liquid biopsy session tomorrow. Are there questions from the audience? Yeah, Dieter.
I wonder, a nice talk. I wonder whether you already found specific genes, so not specifically associated with the immune system, but specific genes in your array, because you mentioned a melanoma array, a gene array, and I thought you didn't show it, but did you find already resistant genes?
Yeah, it's a good question. So in melanoma, there's many mechanisms of resistance, and many of them are non-genetic. And so we intentionally included on our NGS panel known resistance genes, but they are a very small fraction of patients. So there seems to be a long tail of different resistance genes. So being able to monitor, and it's a good question to think about, do you just look at the levels of ctDNA, or does the content of the ctDNA matter, right? And so if you could predict, for instance, that an NRAS mutation occurs in the patient, that they're going to become resistant to targeted therapy. But it turns out the melanoma cells are extremely plastic. And most of the mechanisms of resistance are through epigenetic and transcriptional modifications that aren't detectable ctDNA. So we think that in the end, that was something we tried to do, but in the end, just the levels of ctDNA, the mutant allele frequency, was enough to predict response. But it may be that in some special cases that we can, especially for targeted therapy, where there's more genetic mechanisms involved, that is the case. But we are including them in our panel, and they're being used in the clinic today here in Zurich, but they just don't come up very frequently, unfortunately.
Sorry, follow up to that. So that means you can just isolate ctDNA, quantify it, and that's it? No downstream analysis?
Yeah, exactly. That was the goal, is that we started with very, very complex technologies, but I'm a translational lab. We want to do translational research, which means we don't want to just understand the biology, but we want to develop tools that can be used in the clinic. And the reality is no one's going to do single cell sequencing or spatial transcriptomics in the clinic, just in terms of the cost, in terms of the turnaround time. So the digital PCR assay is incredibly simple. It's just a quantification of the hotspot mutation in those patients over time. And that seems to be pretty predictive in tracking response.
Great. Are there other questions? Anna has a question.
Allow me one more question. It's a little bit more conceptual. I think you nicely focused on the tumor part and how we can predict better which tumor is responding. But my question is, are we missing out on the patient's characteristics, and couldn't be the 50% that are not responding due to the fact that this immune system of this particular patient is just not prone to respond?
Yeah, thank you. I think that's a really important question because in science, we're extremely reductionistic. Like we think, okay, we can reduce it to the simplest parts and then we can understand everything. But I think now in the era of spatial biology, and that's one example of just understanding the parts doesn't tell you the whole picture. It's also, you can say that's an analogy for the patient itself, that maybe the patient's diet or the patient's something else with the patient that's not measurable or not measurable yet is influencing that response. And so I think that's definitely the future of what we would like to do, is to try to integrate other measurements that are proxies for the patient's general health. So we have an FMT study, for instance, where we're measuring the microbiome, and we know that at least that's partially responsible. So I think over time, over the next 5 to 10 years, we'll be able to build a much better model, a patient-level model of understanding the response to ICI. And I think that's the future. But at the moment, we're still at the reductionistic phase, unfortunately. But you're right.
Thank you so much. So we're moving on to the first pro and con, actually. So get ready. It will be exciting. The pro and con sessions, they have a very short pitch by 2 experts, 1 pro and 1 con. They're only 5 minutes. And then we have some further time for discussion. So take out your notes, your cell phone, so that you can ask your questions and challenge them. The more lively, the better. Don't be shy. So we will have our first talk by Amanda Fitzpatrick from London, UK, and she will talk about pro on animal models in brain metastasis research. Thank you for your pitch.
Thank you. Okay, thanks everyone, and thanks to the organisers for this fantastic meeting. Of course, I'm hoping this could be an easier sell, given the groundbreaking talk we just heard from Professor Winkler. And I'd like to open this argument for the use of animal models by a quote from my postdoc actually last week, who said, "There's no model complex enough," and I just thought that was a great way to open this short talk. So we all here know that the metastatic cascade is a complex, multi-step process. But what has even greater layer of complexity is the metastatic cascade leading to brain metastasis, due to the extra challenge of overcoming the blood-brain and blood-tumour barrier. And now we have so many wonderful in vitro models now, transwell assays, organoid models, organ on chip. However, these offer just a reductionist, deconstructed view of the biology and don't tell the full picture. And there are some key steps that are particularly difficult to model using in vitro only, which are here: survival in the circulation, shear stress, crossing the blood-brain barrier, as I've mentioned. Importantly, tumour cell dormancy in the metastatic niche is very hard to model in vitro. That colonization and outgrowth that we saw in Professor Winkler's talk through the intravital imaging, and then the interaction with the microenvironment, which is so unique in the brain. Equally, angiogenesis, and then, of course, an intact immune system to study the interaction with the immune cells. And then finally, the pharmacokinetics of drugs crossing the blood-brain barrier. So I believe these are all very difficult to use in vitro models to recapitulate. In terms of the in vivo models available for studying CNS metastasis, there are many options, and there are many types of assays: spontaneous metastasis, experimental, established metastatic models. We can use rats, mouse, zebrafish, immunocompromised. But of course, the question must be, what impact have these techniques actually added? We can do all these things in labs, but what impact are they adding? So I'm going to break that down over the next 2 slides into mechanistic and therapeutic. And this slide attempts to kind of summarize much of the work shown by Frank Winkler's talk. But essentially, over the last 15 years or so, there have been these really landmark discoveries about how metastatic cells engage with the blood vessels. It's an active process of colonizing in the perivascular space. And then these really fascinating interactions with the resident cells that are unique to the brain, astrocytes, neurons, and microglia. And these are all such key landmark discoveries, which of course we are all hoping will lead to novel ways of treating brain metastasis. And then this slide just summarizes some of the ways that in vivo models are key to shaping the therapeutic landscape. So, you know, the intracranial penetration of novel TKIs that have been developed for these malignancies with EGFR, ALK, and HER2 dependencies wouldn't have been possible without the ability to test them in vivo. And then, of course, the finding that in syngeneic mouse models that surprisingly brain metastases can respond to immune checkpoint inhibitors really paved the way for these clinical trials that show responses in patients, particularly melanoma. And then not limiting it to drugs, animal models have been key in pivotal studies looking at advanced radiotherapy regimens, novel imaging techniques, and importantly, strategies to permeate the blood-brain barrier through mechanisms such as focused ultrasound therapies or nanoparticles, which might have an increased ability to cross that very tight space. In vivo models really are the key translational bridge towards patient benefit. But of course, no technique is without its limitations. And in the in vivo model space, we all know that this is expensive, time-consuming, requires highly skilled procedures, and of course, there are ethical considerations. Equally, you know, in the human setting, we can actually capture the genomic evolution and therapeutic pressures that ultimately shape the way a cancer will respond to treatment. So the in vivo setting can't capture that, unfortunately. So I believe that in reality, we do need all three. We need our in vitro models for our target identification and to deconstruct the mechanism. We need our in vivo models to put it all together and give us the full picture of the biology as much as we can, and look at causality, which we can't do in human studies so easily, which look at the end result. And so this is the end of my argument, and I'll now open it to my, well, yeah.
That's great. Thanks a lot.
Yeah, thank you. Go over.
Exactly. And then the microphone. I'm not functioning anymore, but I speak up, so it's a great pleasure to introduce the next.
Yeah.
Okay.
Thank you.
Molecular oncology here in Zurich, and the director of the clinical trial program at the Comprehensive Cancer Center Zurich. So no better person to speak against animal models. Andreas, we are looking forward to your arguments.
Okay, Tobias, thanks a lot. So, you know, I will spend my first 30 seconds to make a confession. And the confession is I've done many different animal models in my life, okay? So I think the reason why I've been asked is because these last 8 to 10 years, we have focused in the Profiler Consortium to really work with patient material. And that's why I will make this point in my talk. So let me just guide you through the main arguments why animal models may be difficult, you know, to promote progress in medicine. One is obviously it's a very simple argument. You know, we do all this testing, we do all the preclinical work, we bring a substance in clinical testing, and then the attrition rate is still 90%. So despite doing all of this, which of course does also hold true for cell culture experiments. So you can use the same argument to tackle cell culture experiments. And this has not really changed. You know, when you go back 10 years, 15 years, 20 years, it's remained very much the same. Then the second one is a bit more about the brain model themselves. So probably, you know, even if we do have specific models which address the brain, be it for primary tumors or more of a topic in this meeting for metastases, it doesn't predict positive outcome in phase 2 or phase 3 trials. So there's no correlation between success in the preclinical model and the one in the clinical model. And then we have the models for brain metastases compared to human models, cell models, or models we generate directly in trials by getting tissue from patients, which obviously for brain mets is a bit more difficult than, for example, when you have got skin cancer or something like that. But we also see, you know, I mean, it's difficult to generate. So if you do not inject cells directly into the brain, for example, then peripheral models of tumors in mice, they tend to be very difficult for brain metastases because they often do not develop brain metastases, or because of the regulation which tells you when you have peripheral progress, you need to kill the mice, so you won't see the progress in the brain. So that's obviously something which makes using mouse models a bit difficult. Then we have this low spontaneous metastasis rate of more or less all the models. You know, when you look at RIP-TAG mice, PYMT mice, and there's so many different models, and almost all of them do not develop brain metastases during the natural course of the disease we observe in the mouse. Then something else which is also interesting, it seems there's no good model for edema generated by tumors. So, I mean, I just found the publication. It's not something I have tried in my lab, but basically, you know, be it the primary tumors or be it metastasis, they usually produce edema. And one of the first steps for our patients when you have a new mass in the brain is you give steroids because you want to reduce the edema. And this is something which is very poorly reflected in mice models. Then, well, that's one of the oldest arguments. Obviously, there's some ethical concern because you need to keep mice under special conditions, which are not the natural ones. And people tell you, well, this is ethically a difficult point. Then, and I found that's an interesting argument. I'm not sure I can share it, but you know, in this publication they said, well, there's so many different drugs. We are never able to produce so many mouse models and mice in facilities to be able to test all those drugs. Okay, so obviously, you know, when we look at models which look at mechanism, this doesn't hold true because you can use the mice to look at mechanism and you don't have to use them to test drugs, obviously. But it's still a limitation. And for the mechanisms, of course, you know, when we do a floxed mouse or something else, a transgenic mouse, then usually we tackle one gene or we tackle 2 genes. But the real tumors do have hundreds or thousands of mutations. So it's just a very kind of immature biology compared to human cancer. And then finally, you know, and again, this is not related to basically doing research to look into mechanism, but it's looking into research to produce drugs. And the FDA has removed end of 2022 the requirement of running mouse experiments in order to be able to give drug approval. This doesn't mean FDA is not asking for it anymore. They have still asked for that for every single drug which has been approved, but the regulation has changed, and in some time in the foreseeable future, perhaps it maybe we get drugs which have not undergone animal testing and will gain FDA approval. Okay, so, you know, yesterday evening I was thinking, okay, this is a new pastime. You can ask ChatGPT or you can ask Copilot to produce images. And I prompted Copilot yesterday, February 11th, please generate an image advocating against the use of animal models in the research of human brain metastasis. It took quite long, so I waited for kind of 5 minutes until this of an image, and I thought I wanted to share this with you. I'm not sure what the message is, but it's an interesting exercise, and I think it's something we're gonna see in the future, obviously, more and more. Thanks.
Thank you so much. So please, our pro and con speakers.
Yep.
Come here.
Yeah.
We're so good in time. So timekeeping is our talent, Tobias. So we have time for questions and discussions. But yes.
I think it's an important and interesting point to realize that we're not even sure whether what we do in animal models is reflective of what may happen in clinical trials then. So my question is, would it be informative to set up a kind of an animal clinical trial that mimics an ongoing human clinical trial and see whether we come up with comparable results in the end?
Well, I think that's a very good question. For brain mets, I can't answer it because I've not seen it happen so far. But for example, for colorectal cancer, people have started to run co-trials in mice. So they treat the mice for different lines, and then they find out, whether it predicts response. Alberto Bardelli has been doing this, for example, in northern Italy. It certainly generates some interesting results. It started 10 years ago. It hasn't made it into the clinic yet, which shows in a way it's not robust enough for everyday use, but as a concept, I think it's an interesting concept.
Yeah, I think it's a great concept as well, but I think that there would be a challenge to obtaining the really relevant tissue, which is the brain met tissue, because increasingly patients don't undergo resections, so you're not getting that biological material to run a sort of PDX trial for brain mets. I think there are some efforts to do that in various groups, but whether you could have sufficient numbers, and I think that the cases that are operated on, there's a bias there because they're generally larger, and they may be younger, fitter patients as well. So you're always going to have a sort of biased group. Of course, they're the poorest prognosis, the hardest to treat tumours that have to be resected. So that would be relevant there.
There are other comments?
I was wondering, what does the EMA say about the need for preclinical experiments because of our European region? And does the FDA ask for alternatives if they don't find it necessary to do preclinical experiments?
So for the first part, I have to tell you, I haven't looked into any news from EMEA. I believe they have not changed anything so far, but please tell me if I'm wrong. For the FDA, yes, they are asking for alternatives, and FDA has been running a program on organs on a chip since almost 10 years now. So basically the thinking is, this is mostly also about toxicity testing. So again, not mechanistic testing, but the idea, FDA's idea is this could be perhaps replaced at some point by this organ on a chip model. And there's interesting data on liver toxicity, for example, which can be tested in quite a reliable way using organ on a chip.
Andreas, do you know the position in Switzerland of Swissmedic?
Oh, I can tell you, yes, there's no news on that end, and Swissmedic still does it the same way.
More questions up there. Until you're there, Tobias, may I allow you, Amanda, a question?
Yes,
because you were the pro speaker.
Yeah.
Brain metastases are an extremely complex process. Do you think that we have really a model mimicking that process from there is a primary tumor and actually there is this one cell going from that primary tumor producing the brain met, outgrowing to a macrometastasis that then also has symptoms?
Yes, of course, and I think Andreas had a slide about the lack of the low rate of spontaneous metastasis models, and we know a lot of the work we've seen is intracardiac, isn't it? But I think patient-derived models can help to recapitulate that, and some of my work has used leptomeningeal models we've created from CSF, and then we put them back and see some do spontaneously metastasize. So I think the increasing use of these PDX models will help.
Thanks. Up there.
Hello. So I think this also should be seen as immune-related studies and non-immune-related studies, right? As for example, the PDX models are extremely difficult for studying immune system unless you have a completely humanized, mouse. Like, I think genetic mouse models are way to do immune stuff. So any thoughts on how reliable, from both sides of the aisle, of how reproducible and how robust these models on both fronts are for immune-related studies? Very broad question. Sorry.
Yeah, it's such an important point about the immune context and PDX models, because you just, my understanding is humanized models are not really that successful, but I'm not up to date on that work. But that's where the patient sampling really sings, is when it comes to the immune context. But of course, you have those barriers to sampling in the brain that just make it really hard to also understand the early parts. I think that we all want to intervene earlier. And that's what, their talks we've seen this morning talk about, intervening early when at the first steps of colonization, or maybe even before the tumors even reached the brain. So I think that's really hard to model, to assess in a patient. So yeah, any thoughts on that?
I think that's a very good answer. Let me just add one point, and that is, the classical immunotherapy in humans is checkpoint inhibitors. Although there are many other things you can do, but in the end, it's basically 95% is checkpoint inhibitors. And checkpoint inhibitors, there's people claiming they would never have been developed if we had believed in mouse models, because PD-1 inhibitors perform pretty poorly in mouse models. So the one problem is the biology of the tumor itself. The other problem is that we have difficulties in mimicking the effect of the therapy in mouse models, and that's also true for immunotherapy.
So, is it allowed to bring a little bit aggressiveness in the discussion? A little bit, at least. Yeah. Okay. So according to Amanda, I'm quite sure that we will not get rid of animal models in brain met research within the next couple of years, at least. Yeah. And a question or a comment to you, Andreas. We have cited a paper, I think, Journal of Neuro-Oncology, that animal models do not reflect brain edema. So there are other papers which show the opposite. Yeah. So also, I personally worked a long time ago in a rat model, MRI based, showing that the brain edema, and we could clearly show that the VEGF knockouts had much less edema. So it's possible, and it was possible 25 years ago with the old-fashioned MRI techniques. So I would be careful with saying not possible or so.
Yeah. That's fully okay, and I'm not claiming you can't show any edema. I mean, this was a review citing many different kind of approaches, and in many of those approaches we did not see edema. So I think what can be clearly stated is this link between human brain metastasis, which very often does have edema, is not the same in mouse models. I think that's safe to say.
Yeah, thank you for this discussion. I think it's an important discussion. I would actually claim the opposite. So in humans, sometimes you have patients where you have no edema at all in brain metastasis situation. You have a situation where you have some edema, you have huge edema. The same is true for animal models. So you have animal models where you have a lot of edema, which are androgenic, and then you have more the cooperative growing, which have no edema. So I think it's kind of, depending on what you want to model, I think you need to pick your model wisely. And so that allows, so even now with the not perfect animal models, we have in brain metastasis research the opportunity to model, I think, these growth patterns quite well in humans. And I think it's clear there's nothing better at the moment, I think, than animal models, particularly for the metastatic cascade, right? Because that's important. If you just implant your brain tumor cells somewhere into an organoid, that is not modeling anything when it comes to a metastatic cascade, and that I think is a highly artificial model that will not tell us anything. So we can, of course, test everything directly in patients or go to China doing that. I'm not sure that it's ethically better than using mice, so I would question that severely. So what's the alternative? I don't see an alternative. I think what is important is that when you find something in animal models, it's super important to go to the human situation, go to human data sets, go to tissue, try to confirm that what you are studying is really there in the human disease. And I think this is frequently lacking, and this is probably one of the questions, one of the problems that we need to face.
And particularly for new discoveries they have to, right? I'm not sure if the field of cancer neuroscience would have born if we would have not had any mouse models.
Not
so
at all.
exactly. So
Would have been started.
yeah.
Thank you so much. Do you want to comment?
Just to reply on the rat context. So I think that we underuse the sort of larger animals as well, and that's one of the limitations as well of the in vivo setting, is that, you know, probably a mouse is not the best, but it's a substitute for the biology that we can't recreate in labs. But of course, using rats would possibly mimic the setting even better.
Thank you so much. So with this, we close the first session. You can continue the discussion during the coffee break. We have 20 minutes of coffee break. It will be served just right outside. And please reconvene here at 11.
So, welcome back after the coffee break. We're going to continue with the next session, which is going to focus on novel diagnostic approaches. My name is Nathalie Albert. I'm a professor of nuclear medicine at the LMU Munich, and I'm going to co-chair this session with Professor Roland Goldbrunner from the Department of Neurosurgery, Professor of Neurosurgery in Cologne. And I don't know if you are aware of that, but I think it's a great pleasure to have him here today, because in Cologne, it's the start of Carnival today. So it's really special that he's favoring this session. I would like to now announce our first speaker. No need to introduce him, Professor Andreas von Deimling, Professor of Neuropathology at the University of Heidelberg, and he's going to talk about his favorite topic, methylation profiling, but today not for gliomas, but for brain metastases. No, you have yours.
Thank you for this very kind introduction. Yes, it definitely is a favorite topic, but regarding brain metastasis, it is indeed slow moving. So I would like to start with the essentials, my disclosures, and then move on to 3 slides which are known to many of you. I apologize for it, but I think they're still important. Why is methylation of interest in typing tumors? And the principle is very simple. Throughout development from a single cell to a complex organism, every cell runs through a constant programming and reprogramming, which is to a major extent, let's say half, governed by methylation sites, predominantly in promoter regions. The other axis, like histone modifications, are not touched with this, but the methylation status of CpG sites in promoters are extremely important differentiation. And in each stage of differentiation, a certain cell type has a rather distinct methylation profile. If a tumor occurs, something important has to happen. This normal development of methylation towards the final differentiation is disturbed, is interrupted. And with a grain of salt and very simplifying, very strong, it's kind of frozen. So a methylation profile is frozen in that developmental process, allowing to track back to the cradle of the tumor by looking at the methylation profile. Of course, as this arrow indicates, the methylation profile is not completely locked. I mean, there are definitely 1,000 genes which are in tumor progression, reprogrammed, and where the methylation profile of the according CpG sites in the promoters are altered. But remember, there are still about 20,000 other genes which are not touched by this process. And they built a very stable backbone for this concept of typing, of classifying tumors by the methylation profile. And very important is, it's it's only classification which is possible. It's much harder to address things like progression, malignancy, because that is obviously in the tumor very dynamic throughout the tumor development to end stages. This is not addressed, so methylation is basically very useful for classification and not for grading. This has been used to quite success by employing actually different approaches. So this general concept is very platform independent. You can do it by direct sequencing of bisulfite converted DNA. You can use nanopore recognizing methylated CPG sites itself. You can probably do it, it's not known yet, but by the upcoming 5 or 6 base sequencing offered by different employers. What we have done, most of our data rely actually on chip analysis, which suffices for this attempt and has the advantage of being extremely simple and technically foolproofed. And also a very big advantage, it works with formalin fixed tissue. So you can work with FFPE material, meaning we can transport material, we can assess material from the past, which is especially important for looking at the profiles of tumors which are exceedingly rare. We don't have enough fresh tissue of rare tumors to generate sufficient amount of profiles to identify a common pattern. In brain tumors, this one worked out very nicely based on the first set in 2018. You see 2,800 reference cases were used nowadays. Our colleagues work with the second version, which is based on roughly 8,000 reference samples, and we are now working on the next version, which probably will rely on something like 30,000 reference samples. You see, we are running against a ceiling regarding probably detecting novel tumor types and subgroups, and we need more and more large sample size in order to come to advances. The specificity of this approach is very good. We can improve basically on the sensitivity, meaning recognizing more tumors with a high confidence score. This has been also done with sarcomas, where we have initial approach from 2021, which has now been replaced just at the time being with a new version, which has just been pre-published at medRxiv. And these are the two actually models which work well and have common use. Now let's move towards the epithelial tumors. If we address metastasis, we are of course in the middle of epithelial tumors. Let's put the melanoma metastasis aside. So, how would you approach, for example, cancer of unknown primary, which is maybe one of the biggest challenges for this approach? I personally think, this is my opinion is, you probably cannot approach this by just doing a classifier which is for metastasis, pooling metastasis and trying to match new cases to this wouldn't work. This wouldn't work because we have several evidence. We've heard this morning from Frank Winkler, for example, that tumor cells, epithelial cells in the brain, directly interact with the brain tissue. And of course, this has a profound effect also on their expression profile, methylation expression pattern. And tumors from other organ sites will do so too. But the same tumors which interact with the brain, if they interact, for example, if metastasis to the lung, it will be different cells they're interacting with, yielding very different responses. So I think a very good approach probably is generating organ-specific epithelial classifiers, which also must include the typical metastasis occurring in this organ. And this is also why the reason why the next brain tumor classifier has 30,000 samples. It will include the typical tumors metastasizing to the brain. And One of the null hypotheses is that tumor metastasizing to the brain will behave or look different than another metastasis from the same tumor to another organ system. This is actually the first epithelial classifier which is ready to use. This is currently in beta testing. This is a skin tumor classifier, and although skin tumors are a very special thing, it helps, for example, very nicely distinguishing nevoid lesions from melanoma or from tumors from the Smits tumor group, which is definitely an important clinical question. When looking in the literature, just looking at the cup, I just listed, and the numbers are very idealized. They're simplified. These are the typical tumors which are underlying cups. 90% of the cups have an origin in one of these organs. On the brain, if we restrict ourselves to brain, most of them are from lung, from mama, kidney, and then a little bit from others. Yeah. Melanoma set aside, but usually melanoma are not that big of a cup problem because they have such a specific immunoprofile. I think usually they're recognized by pathologists. In the literature, very few papers have addressed metastasis for diagnostic purpose and using methylation profiles. One study I found, would like to briefly introduce, is that from Kalina et al., which have specifically looked at tumors from the pancreas. And they have looked at pancreatic ductal adenocarcinoma, at intrahepatic cholangiocarcinoma, at normal bile duct, and metastases of these tumors, of the first two. And they have a very restricted panel of tumors. They are separating these apart. Their controls on the right side are about 10 tumor types: stomach, colon, breast, esophageal carcinoma, lung, only adenocarcinoma, liver, hepatocellular carcinoma, adenocarcinoma from the rectum, and uterine tumors, actually only the endometrial carcinomas, prostate adenocarcinoma. Using this restricted set, they were pretty good in predicting also from metastasis where these tumors come from. But I'll show you a couple of images, and they should alert to the problems. Here, this is a very nice kidney, and what you see here is, I would like to point your attention to this group. Here, the PAADs, and this is the same tumors. And what you see here is these don't match because they are just done on different platforms. One on the old 450K array, the other one on the EPIC1 array. Now we have EPIC2 arrays. So using archival data performed on different platforms will require a lot of probably also cleaning, and in order to make them matchable. Another interesting aspect is, sorry about this, is this group up here, the intrahepatic cholangiocarcinoma with two groups. A little bit weird. Well, upon questioning the authors, turned out, well, one of this group probably is a group of tumors from Far East, and they are parasite induced hepatic cholangiocarcinomas. Sorry. So that means we have quite a different presentation of tumors which should look alike. The same here. This is actually an argument which connects to what Frank Winkler talked about this morning. This only shows pancreatic ductal adenocarcinoma, and the brown ones are the green ones are the primary tumors. Brown ones are metastases to the liver. The light brown ones are metastases from the peritoneum. You see, they cluster completely different, and this is an argument for that we probably have to address this by looking for each metastasis in its host organ. We will not be able to make a metastasis profile which accounts for metastasis in lung, liver, brain, and elsewhere. What is very nice about this project, this paper, they created a page, a homepage, and this allows accessing this classifier, and I tested it out, it's still in use, which is quite unusual because mostly these classifiers are built, appear on a homepage if they do, and are taken off the homepage usually for liability reasons or else after a short time. So it's always a big problem to generate these classifiers and keep them alive for a long time, which requires a couple of things, of course, updating all the time, but also really having a safe environment where you can house them without a university and institution being afraid of liability problems. I'll show you now the current attempts to generate these epithelial, this organ-specific classifiers. We just talked about pancreas. This is a pre form of a pancreas classifier. It shows different pancreas tumors, and although also already a couple of other tumors are included, which frequently occur in the pancreas, such here the inflammatory myoplastic tumors, or the desmoid fibromatosis is seen quite sometimes in the. So we'll run through a couple of these images. This is liver gall. You see also there's already a rather large reference set, and the attempt again is to include also tumors which are not primarily originating in liver gall, but which are metastasizing to this site. Quite similar. Lung and pleura classifier. You see here a very big group up here, which is, for example, the Ewing sarcoma, very frequently metastasizing to lung. Blue in this case, and gray is the ones to the pleura. Gray is those ones to the lung. So there's not a significant difference between these groups. And what you also see from this image, we have not that many different primary lung tumors. So in this case, already the majority of tumors in this pre-classifier are metastasis to this organ. And this is how I would envisage over a long time to solve that cancer of the unknown primary, that we have all these sets which are then mended toward classifiers, and you just run an methylation profile from an unknown tumor through all these classifiers and see where it sticks. You may get a couple of hits, and then again it's up onto the clinician or to the pathologist that, oh, this is more likely, or maybe to add on with specific tests. We also have quite some progress on ovary and on the uterus, showing nice reference sets already, although in these we have not included a lot of metastatic tumors, which is to follow. And it also exists for the bladder. Good. Just something to demonstrate that we don't have a monopoly on nice images with little points. This has been invented much earlier, and probably to a greater mastery. I'd like to come back to cancer of the unknown primary. So how do we address this problem? As I've just told you, my personally preferred way of addressing this will take a couple of, or probably 2 years or so at least, to really come to a point where we can effectively use it to address the CUPS. How can we approach it? Several approaches have been taken. One, for example, by Matthias Preusser and his team in the United States. What they did is they looked for CpG sites, the genome, which exclusively are related to organ-specific development. By doing so, they came up with a nicely and published classifier, which gives a prediction for organ origin, not for a tumor, but for an organ. This would work. Unfortunately, this classifier is not publicly available. But this would constitute an approach. Another approach, alternative to that huge holistic approach of generating the classifiers for each organ, is taking just the most frequent tumors which we encounter in brain and build a most frequent metastasis of the brain and build a classifier upon this, and then validate it with to cancer of unknown primaries. And this image shows you, just I compiled this over the last weeks. You see here typical tumors which metastasis to the brain. So adrenocortical, kidney carcinomas are a small group probably here. Then colon carcinoma, adenocarcinoma, gastric carcinoma is here, and all the way down, mamma is included, ovary is included, pancreatic is included. So this covers a considerable amount of the tumors which we encounter as in the neuro scene as metastasis to the brain, and the reference cases for these groups are already available. That's all done. What we would need is really something to verify this. We need a validation set. And this is maybe here, if you are here, this may be something where you might be interested. If you find such an approach for the time being, for the next 2 years of interest, this can be finished within a short time. What it needs is a larger series of cancer of unknown primary, preferably which have been after a couple of months being solved. If these tumors could be analyzed and plotted against this and validated, then there would be a tool which could be used for the next couple of years to address these tumors. And such an early classifier would be easily hostable as a tool, as an experimental tool in a safe environment, and could be made available to everyone who wants to use it. So this is something I would like to present to you, and I hope I'll get some response, because I find this quite exciting. And with this, I close my talk and thank a couple of people for many people for contributing to these projects. Thank you very much.
Yes. Thank you, Andreas, very much for introducing us in the world of methylation clustering. So is there any question from the audience? Frank?
Yeah, thank you, Andreas. Great talk. Coming from the notion, and I think you have also kind of conveyed the notion, this is still true, I believe, that the methylation profile tells a lot about the cell of origin and where it is coming from. And this is why we can kind of have this very nice Disney plot with all the entities, etc. However, you also showed that the microenvironment can have influence on patterns of methylation. Probably, not so maybe clear, but more on a maybe slighter scale. So how do you weigh these two aspects? You have this, let's say, very robust and stable methylation pattern throughout tumor evolution and in different environments. And at the same time, you have slumps changes that are different when tumors grow in different environments. And can we use maybe this information even to understand in these cancers which pathways are activated, cancer neuroscience, right? Are they? Do we get a monoclonal phenotype of certain cancers or cancer cells? And can we even go down to single cell methylation, maybe to find certain subpopulations of cancer cells that have different traits?
Thank you, Frank. I mean, what we really are lacking are systematic studies of tumor samples, metastasis, localized to different organs from the same person, and this on a larger scale. You probably need something like having 50 sets of samples in order to address this question. Another problem, which is obviously not really mentioned because it was too trivial, I didn't mention that, but of course we also have a problem whenever we have a metastasis. It may not only be the tumor host interaction which has an effect of the methylation profile. Of course, we always have a certain amount maybe of contamination of the homing of the housing tissue, which also contributes to the shift, to a potential shift. This is also not clear how big the one or the other effect are against the other. I think this can be, but this can be probably in silico be sorted out. But what is really lacking is a large group, a large set of tumors, and this may be also something for this group to set up a systematic study of metastasis, multiple metastasis of a patient series.
And single cell, what do you think is that on the horizon? Or, I mean, people are touching it already.
Sure. Yep.
You believe that?
I think single cell is a wonderful tool, and I'm scientifically highly interested. I'm more a mechanic person, so I'm more trying to work towards tools which can be used which can be simplified and used on large scale. And this is something where I see single cell at the moment due to the prohibitive costs, not yet in the diagnostic field.
That's true. I mean, the nice thing is you would kind of have an easy calling cancer cells with methylation profile. So
absolutely.
That would be kind of really straightforward.
Okay, Matthias.
So I wonder, you showed these different clusters according to relatively broad categories, lung adenocarcinoma, lung small cell lung cancer, and so on. But in a clinical setting, our classifications are much more sophisticated at the moment. For example, HER2 positive, triple negative, HER2 low, EGFR mutant, not mutant, and so on in lung cancer. Is there this granularity in methylation profiling? The first question. And the other one is, do you think methylation profiling correlates with the likelihood of responding to a targeted treatment?
You're addressing a very important question. I personally think that methylation analysis is primarily targeting classification. I think it's a completely different axis than those analyses which are targeting how do I treat the patient best. There still is definitely sequencing right now, the workhorse at all, which cannot be replaced in any way by methylation analysis, and probably the future will also be proteomics. I think this will be one of the future things. So I don't think that methylation will be replaced by these others, because the stability and the lineage, the results you can obtain towards a lineage are best if you are using methylation. But then, if you address how far is the tumor is gone, what are the targets, how do I treat it, then I think methylation plays a very small role. Yeah.
Okay, directly correlated to that.
Actually directly correlated to that, because I was wondering, we always have this rather large cloud of tumors that cluster together. Can't we have there a closer look in there? What is, like, for example, the dot on the outer right and the downer left, and if there are differences, because it's still clouds, they're like not totally overlapping, and what is the biology difference between that? And could that maybe refer to what Matthias said to, for example, response or also prognosis?
I'm not sure whether I can give. Could you please let me know what this cloud consists of? So are you talking of a single tumor type or?
So if you look on your plots, what you see is then that there is certain an entity has a cloud, but they're like not totally overlapping, but there are also outer outliers of that cloud, and whether there is an information behind that.
There's first of all, you of course you always have this cloud because you have certain amounts of differences in the tumors. You have always certain amounts of different interactions, probably with the tissues, certain amounts of contaminations, certain difference in tumor purity, which is assessed. This all plays a major role in this. So the point is that if you succeed in identifying the core of such a cloud, you can build a classifier which also is able to predict the cases which are a little bit more in the periphery as belonging to this core.
You're fine with that? Okay, so we have to move on to stay in time. Thank you very much, Andreas. I have the pleasure to announce Professor Marion Smits from the Department of Radiology and Nuclear Medicine, Erasmus Medical Center from Rotterdam. Please, Marion.
Thank you very much for the introduction. I'm usually not used to the table being too high for me, but I'm actually going to lower. I think there's a button. Yes. See, technology. Thank you so much for the introduction and for inviting me here to this meeting. It's an honor to be here. These are my disclosures. Of interest is that I have worked with BRCA and contrast agents, but I won't discuss these studies here today. So, does this work? So what I'd like to talk to you about is several challenges that we have in medical imaging and basing our sort of new developments on those challenges and how we want to address these in the future. So the first thing, of course, is something that we all encounter, and you encounter our increasing demand and problems with the workforce by the waiting list for scans and for reports. There is a huge European shortage of radiologists and radiographers versus, of course, an increase in the demand not only for imaging of metastasis, but also, of course, an increase that we know of the incidence and prevalence of metastasis and the demands for imaging. The graph you see is an overview of the number of radiologists in Europe in 27 member states per 1 million inhabitants, and you can see mostly the red one is the median, the average. And you can see that there is a huge variation in the number of radiologists available corrected for the number of inhabitants. And so you can see that, of course, some countries will have more trouble with facing these demands than other. And on top of that, of course, as we all know, we have the aging population, the improved survival of cancer with an increase in brain metastases, novel treatments that are often costly and much more costly, of course, than a single or several MRI scans. And then we do more imaging and also more screening, which means that we find more lesions that were otherwise asymptomatic and potentially never of clinical relevance. I'm sure we'll come back to that later in the debate. So all of this means that we're dealing with a lot of challenges here and how we're going to deal with that. So one of the things to reduce the waiting list for the scans is to reduce the MR acquisition time so that we can increase our throughput. There are many ways already how we do that. These are techniques that are in development, but many of them are already common practice. And these are all hardware solutions. So this is ways of cleverly designing the scanner and the tool and the radio frequency pulses such that we can speed up scanning dramatically. More recent is, of course, we can't talk about new developments without talking about AI, deep learning image reconstruction. This means that we get much more noisy images, but we use deep learning to basically reconstruct this into a nice looking image. And I'll show as an example in a minute. And this is now available from all three MRI vendors for certain types of sequences, not all of them. So this is becoming more and more common in clinical practice. Another development has been around for a while is the so-called synthetic MRI, and the idea is that you do a single scanning sequence of 5 to 10 minutes. Is now getting closer to those 5 minutes or even much shorter, which is quantitative. So we get quantitative T1 values, T2 values, and from those we can reconstruct or calculate the weighted images that we're used to. And again, I'll show you a few examples of this. This is not yet widely used because it doesn't quite have the same spatial resolution as we are used to in our current imaging, but it's becoming much, much better. And I expect that this can really increase our throughput dramatically and make all of our imaging easier. So deep learning image reconstruction, and like I said, all vendors have this now. So this is a T1 weighted image. There's no lesion here without the deep learning reconstruction of the spine, and this is with the deep learning reconstruction. You can see the images are much sharper and smoother than without, and this is a T2 weighted image of the thoracic spine. Again, comparing these two, you can see the dramatic increase in the image quality. It looks much nicer. And you can use that not just to make the images look nicer, but like I said, you can then acquire much more noisy images, and that means you can reduce your scan time and then apply these reconstruction images algorithms to then have the same kind of image quality in the end. So you can have multiple ways of setting your filter, and if you do that or your reconstruction algorithm, you can reduce your scan time by about a third or even more, going from almost 3 minutes to 1 minute for just a single thoracic spine scan. And this is what we can do now. So imagine with all of the advances in AI that we're getting, how this could look in the future. So there's already talk about, having a full brain scan, in 1 to 2 minutes with everything that you need. A little bit of a future perspective, but I think that's not too far away. So this is the synthetic MR. So on the top, you can see the reconstructed images that come from this single scan that you do. And at the bottom for reference are the images acquired with a conventional sequence. And you can see specifically, we can compare very well the FLAIR images, and you can see how comparable they are at the level of the brain and the brain lesion. Again, we're not quite there yet. It's not available for all of the image contrasts. FLAIR was in fact very, very difficult to do. But this seems to be something that we can really look forward to in the future. So there is, in our world, a very big debate, and this is very petty, but been going on for ages. What is better to do, a 2D image or a 3D image after contrast of T1 weighted scanning? And the problem here is that you have to use different readout techniques. So spin echo versus gradient echo. Can't go into the details, but it means that you have on the one, it's a trade-off. You have on the one hand a much nicer, on the right spin echo, you have a much nicer sort of delineation of this lesion, but you don't have as high a resolution, especially because of the slice thickness, which of course is the advantage of the 3D imaging. And what this means basically, this very often, and what is also recommended, is that both sequences are performed. And if you just keep in mind that each sequence is about 3 to 5 minutes, this really, of course, drastically increases our scan time in the scanner, and that means no new patients in the scanner. So the big question, of course, is, and now it's meme time for those who are on the socials, ¿por qué no los dos? Why not both? Why can't we do spin echo 3D? So, well, again, that is something that has been recently developed. It is there now. It's also known as black blood sequences. They're not truly black blood. They can be made black blood. But what it means is that we now have the advantages of spin echo, and it's 3D, which has a superior sensitivity even for small lesions. It is particularly good at 3 Tesla, so higher field strengths, but does work at 1.5 Tesla as well. And the idea is that this can probably replace these combined protocols, so we can reduce, again, our scan time, even though this sequence does take a bit longer than a gradient echo sequence. So just some examples here. So this is a paper where they looked at patients that were scanned with both sequences in random order, and they had various groups of patients. So there's 4 groups basically, where in group 1, and that was a significant number of patients, the black blood sequence saw lesions that you could not barely see on the gradient echo scan. Then there are 2 groups, group 2 and 3, where they could be seen on both scans, but it was easier to see in one sequence than the other. And there were only 4 patients out of these just over 100 patients where the gradient echo scan saw the lesion and the black blood sequence did not. What is interesting is that these were all melanoma metastases, and the thinking is that these lesions were actually not enhancing, but these are the hyperintense T1 weighted lesions that you see with melanoma without potential enhancement, and that is why these were false negative. But overall, it seems that these new sequences are working very, very well and can help us save scan time. Of course, another way, so that's our patient throughput. Another thing that we're dealing with as radiologists is that we, of course, have to look at all of these scans so we can increase the throughput and reduce the scanning waiting times. But of course, if you're now waiting for 4 weeks for a report, that is not always going to help you. So, of course, this is where we're all hoping for AI to solve this problem, and that it will help us detect lesions automatically and also outline them for volumetric analysis and follow-up. So if you look at the meta-analysis, there's 42 studies already, so this is a very extensively studied field. In 2024, this was, large numbers of patients and lesions, almost all retrospective and lots of them single center. But we see that there is a fairly okay sensitivity of about 85%. But smaller lesions are particularly a problem, and of course, those are the ones that we miss as radiologists as well, and would like the algorithms to pick up for us. Since then, a new study has been published from Amsterdam, and they used a very nice sort of training and external test set and a very nice algorithm specifically focusing on these small lesions. And you can see that there is very high sensitivity of detecting lesions with very reasonable false positives, because we don't want all of these false positives either, which is very comparable to the human raters as well. So again, my hope is that we will get this software soon, and that this will help us, get reports done quickly. And I mean, to be fair, will probably do better than me at the end of a long working day. So lesion characterization, I think there is not much we can do in terms of predicting the primary tumor with MRI, but differentiation from other tumors and tumor-affected lesions. For this, advanced imaging techniques are generally quite useful, such as diffusion. I won't go into too much detail, but one of the things I'd like to show you is that in glioblastoma, to differentiate metastasis from glioblastoma, which of course is something that we encounter quite a lot, perfusion can be very helpful. Nothing is 100%, but some things can. Sometimes you see things that can really help you make that diagnosis. Is this a pointer? No, this is not a. Oh, it is a pointer. It's a pointer there. Yeah. Okay, let's try again. Can I go back? So where do I point? Oh, the top button. Yeah. Yeah, okay, here we go. Okay, so I'm giving it away, but what we can see here is a lesion that looks very, very similar on the post-contrast imaging. Both are solitary lesions, but what you can see here is that there's high perfusion in both of the lesions, but in the surrounding area, in the metastasis, we see very low perfusion. This is pure edema, no tumor whatsoever, whereas in the surrounding area of the glioblastoma, we see that there is high perfusion. So due to the, hypervascularity and already tumor infiltration in these lesions. So that you don't always see it, but if you do see it, that gives you high confidence that this is more likely glioblastoma than a metastasis. So like I said, prediction of primary tumor, we are not very good at that. Melanoma is maybe the exception, where of course we have the hyperintensity, but again, we can also see that in other lesions that have hemorrhage, so that doesn't make it a definite diagnosis. There is some research into AI. It's all very much still in the very early phases, needs external validation, and is basically not very good. So novel treatments. So this is where assessing treatment response is, of course, a major part of my work, and this is complicated by treatments. So we have by the new treatments that we have, as you all know, we have the pseudo-response, the pseudo-progression, and we'd also, of course, like to, especially with costly or invasive treatments and toxic treatments, to have a early assessment of treatment effect. So pseudo-response, we saw this morning already that anti-angiogenic treatment might have benefits, but of course what we're looking at with MRI doesn't really help us differentiate between a true response or a pseudo-response, and can also confuse our assessment along the treatment course. And I'll show an example in a minute. Of course, pseudo-progression we all know, and I'll talk about that in a second as well. So what we have here is a lesion who is a brain metastasis, a patient with a brain metastasis who was treatment lung cancer patient, who had a what looked like an apparent reduction, or at least there is a reduction of enhancement and also of edema, and it seemed to then, upon follow-up, to recur again. So get bigger. But if you then take into consideration that during this first follow-up, actually this patient was treated with bevacizumab, and then the bevacizumab was stopped, and then we see this, apparent increase. So to be honest, we have no idea, of course, was there indeed a treatment effect and did the tumor recur, or was this just the anti-angiogenic effect with reduced leakage and reduced edema due to the bevacizumab, and is the lesion in fact just unchanged? And again, there are some studies that claim that they can see, can assess this with advanced imaging techniques, but to be honest, I don't think they are good enough to ever base our opinions on. So this is really an issue, and I think will become more of an issue if we start treating more with anti-angiogenic treatments, and especially, of course, in trials, this will be very difficult. When are we going to call something progression, and when are we going to call something response in these instances? Another thing to keep in mind, so perfusion is used, of course, a lot to assess, to see whether we can distinguish between tumor progression and pseudoprogression or radiation-related effects. But what is important to remember is that lots of these findings are based, and accuracies are based on glioma research. And what we see in specifically in metastasis, so this is in metastasis, that we see that the sensitivity and specificity is actually a lot lower than in high-grade glioma. So we can't just translate this information from glioma to metastasis. It makes total sense, of course, if you think about it, but it's, the literature on the brain metastasis is much more scarce than it is on glioma. The other thing to keep in mind, or what we do is realize is that the most commonly used perfusion technique relies on T2 star weighted imaging. That means that's very sensitive to changes in the magnetic field, such as due to blood hemorrhage and the iron that deposition that you get after a hemorrhage. And after radiation therapy, what you generally see is a very nice ring of hemosiderin in what was the lesion. But of course, that makes this perfusion technique useless. So we have to use other techniques, such as arterial spin labeling, which doesn't rely on this T2 star weighted imaging. And in this case, there was no increased perfusion there, and this was a treatment effect. So tiny little sidestep, because this is my favorite sequence of all times, is arterial spin labeling. And the reason that I find it is my favorite is because I find it magical. We don't inject contrast, and we get this information about the perfusion in the brain. So what we do with this is that we image the brain first, just a normal, it's not entirely normal, but an image of the brain. And then what you do is you basically add a magnetic label to the blood flowing into the brain, and you image again the brain, and you subtract these images, and you do this multiple times, and then you get a perfusion weighted image. I find that magical. Another slightly, but this is my number 2, is chemical exchange saturation transfer imaging, CEST. It's also a bit magical because, again, you don't give contrast, but what you can measure is protein content and a proxy for cell turnover. So amide proton transfer is most commonly used. And basically what it does, what happens is that protons, as you know, proton MRI depends on the spins of the protons, and when they're bound to certain molecules, they have a different kind of spin and a different frequency than when they're in the free water pool in our body. And the free water pool is mostly what we measure, and there's an exchange. So the protons bound to molecules exchange with the free water pool and conversely. So what you can do is basically you sensitize these bound protons, and then you measure for a certain amount of time, which exchange to this free water pool, and then you measure that free water pool, so you have a sort of an enhancement of effects. That's a very, very simplistic, the way I understand it, explanation. It's not just sensitive to amide, it's also sensitive to amine, to pH, which is very interesting, I think, as well, in the context of cancer research and cancer imaging, and other ways as well, glucose, glutamate, etc., and other contrast agents. So it is a very powerful, I think, and non-invasive tool. So it has been applied. So again, it's mostly in the research domain, partly because it's not a very harmonized sequence yet. Most scanner vendors will have it, but you need higher field strength, so 3T or upwards. So we can't, apply it just anywhere and everywhere. But the initial sort of research is promising. So this is an example of early treatment response assessment where patients were scanned several times directly after stereotactic radiosurgery. And the idea was that if you see a low protein content blue, this lesion was going to respond, so after 1 week, whereas if there's higher protein sort of signal, the lesion was not responding. So the same has been applied to differentiating between pseudoprogression or radiation necrosis versus tumor progression. And again, we see sort of the same idea. So look at this image, the APT, the amide proton transfer. This lesion has a higher signal and this one has a lower signal. And this is plotted here as well. In the radiation necrosis, we have lower signal than in the tumor progression. So again, this could be hopefully a much better technique than perfusion imaging to make this distinction. So I want to spend the last few minutes on planetary and global health, because I think we all hear it, but it can't be said enough that we have to take into account what we do with us as a healthcare workers, but radiology, of course, is a massive contributor to carbon footprints as well, and other ways of polluting our planet. So there are many initiatives. Green radiology is a real thing, and I think there is finally some acceptance that we really have to do something. But one of the polluters is contrast agents. So do we need to give contrast? Well, I think we all agree that this tiny, tiny little lesion that we see here can't be seen on the T2 weighted imaging, and we can only see that on the post-contrast image. If we wait a month, the lesion is bigger, and we have also visibility on the T2 weighted imaging. But yes, for that small lesion detection, of course, we need the contrast. We also know that more contrast will probably help you detect more lesions. This is an exceptional example. I think it is exceptional in a patient where a patient scanned only a few days later with a double dose of contrast, we saw 10 times more lesions. Like I said, I think this is the exception, but it does show that we have much higher sensitivity if we give more contrast. That comes, of course, at the cost of false positives as well. So we do need to be very, very careful with that. So it's clear that we do need contrast agents. It's clear that it is a very important adjunct to our work. But as you also know, we all call it gadolinium. It's actually gadolinium-based contrast agent. Gadolinium is a rare heavy metal mined as part of rare earth element mining, which is very, very polluting. And gadolinium itself is very toxic. So it is, of course, packaged in a chelate that will make it safe for use. We also know that there is an accumulation of gadolinium, or at least GBCAs, in the brain and also in other parts of the body. We don't know what that means yet. We do know that there is a dose effect. So the more doses you get, the higher this accumulation is. And of course, that becomes an issue now that patients survive longer. You know, we have, maybe I should save that for the screening. I'll let that go. I'll save it for the screening debate. And one of the other things is that the contrast agent is treated through the body kidneys. So this enters, of course, our wastewater, can't really be taken out properly or filtered out. So this actually shows up in our tap water and in our soft drinks, and specifically soft drinks based on tap water. You might think, how bad is it? Well, it is bad. There's a study from Germany where 6 major cities, the gadolinium content was tested in soft drinks in McDonald's and Burger King, and they all showed lots of gadolinium. This is not true for the bottled stuff, so you're safe there. But it is in our tap water, and this one is even more scary. So this is, don't look at the colors. The colors are just the types of contrast agents, but this is checking the concentration of contrast agents in our tap water. And we see, especially in Europe, it's really high. This is still, it's still below sort of toxic levels, but it accumulates. It's very stable, and it just enters our tap water. Even where you think you're safe, our water comes from the Alps, it's there. So we have to be aware of gadolinium. So can we reduce it? Hopefully we can. There are contrast agents that are using less gadolinium. They have a higher relaxivity basically. And we can hopefully use AI for this, because the same tools we use to enhance our images, we can also use to use less gadolinium and still get the same results. So this is a study where they did that for various lesions, and there was a fairly high agreement between full dose and 10% dose. There are even, and again, we come back with the magic, but this is too much magic for me. Can we actually calculate where contrast enhancement would be, even if we don't give contrast, based on the non-contrast images? And this is the true contrast enhanced image. This is the one that's reconstructed only from these sequences. This is glioma. So I don't know how this works in tiny, tiny little micrometastases. Magic, maybe too much magic. But remember, of course, that contrast isn't everything. So these are two contrast enhancing lesions again. And with ASL, my favorite sequence, we can see that there's high perfusion in glioblastoma, low perfusion in metastases, which is really indicative of the differentiation as well. The last development, also in the sense of reducing our burden on the planet and equitable access to healthcare, is this. This is an MRI scanner, which you can just, as you can see, just wheel up to the patient. And these are the images. They are not the most amazing images, as you can see, but remember, this is 64 millitesla. And again, this can only be done with hardware, but also, of course, AI sort of development. So to conclude, we're working on getting you your scans and your reports sooner. I think we have a lot of challenges in assessing novel treatments. And remember, of course, that findings from glioma don't translate to brain metastasis. CESS is potential, but it does require higher field strength. Remember ASL, and of course we do need to use contrast agents still, but do consider this, that we have to be maybe careful with our use. And with that, I'd like to thank you for your attention.
Thanks a lot, Marion, for this really fantastic talk, as usual, and thanks for giving us some insights into the moving field of radiology. So you've shown very comprehensively that there are lots of innovations. I would like to start with one short question. I was amazed by the AI-based reconstruction and the really nice image quality that you see there. However, if I want to be provocative, if I look at photos of myself based on AI, they look also nicer, but they don't represent the truth. So how do you make sure that you're really seeing the truth?
That's, of course, a very, very valid question. And this is the worry we have as well. And so what the vendors tell us is that it's not generative AI. So it doesn't make the images for us. It just reconstructs out of fairly sparse data a better looking image. So it reduces the noise rather than, well, brushing away the lesions. Of course, the question is, where do you go? How low can you go, basically, in your sparsity, or in your, well, where does noise? Is it just noise? So I think that is an unanswered question. So obviously these are commercially available tools. We have asked this from the vendor, is this safe? They say we have tested this extensively. So, as long as they tell us the truth, we try to trust them. But it's something we really have to be very careful of. Yeah.
I would allow one more question. Nelike?
Thanks, Marion, for that fantastic talk. Very inspiring. I have a lot of questions, but I'll be very brief. On the metabolic MRI, which I find equally fascinating, I was wondering, do they also have as much trouble with blood products in the metastasis like the other sequences? And is there a lower limit of the size of a metastasis that you can do the metabolic MRI on?
Yeah, very good question. So the CEST is sensitive to blood. So I'm worried that, the papers don't report this. So they just give you the nice images and the nice values that they measure. We don't actually have that experience ourselves yet with CEST in brain metastases, but I do have the same feeling that we'll probably run into the same problems with treated metastases that have hemorrhaged, that we won't have perfect findings. And as far as lesion size, this is low resolution imaging, so we can't expect to have an answer on tiny lesions. So you'd have to think of lesions at least about 1 centimeter, I would say, actually the same as perfusion. So don't ever expect to get an answer on a lesion that's smaller than 1 centimeter.
Thanks a lot, Marion. We will now move on with our pro and contra session, which is going to discuss screening MRI, yes or no. The first speaker is going to be Nicolin Hainz, who unfortunately cannot be here today, but has recorded a video. So we are going to start with a video.
To look at the pros of screening MRI for brain metastasis. I'd like to start with the fact that the incidence of brain metastasis is increasing. This is multifactorial due to advancements in tumor treatment, increased survival in patient age, heightened awareness, and advancements in MRI. So the problem isn't going away. It's actually a growing concern. Let's take breast cancer, for example, and here brain imaging is only performed if neurologic symptoms develop. But what about the asymptomatic patients that are discovered through screening? We look at this recently published study, cohort 2,600 patients, and the asymptomatic patients identified through screening had a significantly lower number of brain metastases and a smaller diameter of the individual metastases themselves. Now, you might not think that 7 millimeters makes much of a difference, but if you consider the narrow treatment windows we have for eloquent brain areas, such as the brainstem, where one brain metastases over 1 centimeter cubed are predictive of a high-grade toxicity, then 7 millimeters might make all the difference. Now, you can counteract that using lower marginal SRS doses, but that might also lead to lower local control rates. Again, smaller brain metastases are associated with better outcomes after SRS, and the increased local failure rate is due to the increased tumor size, and with melanoma, for example, the hemorrhagic brain metastases, which are inherently larger and compound to the increased local failure rate. Eventually, you run out of treatment options as tumors over 3 centimeters are then considered for upfront surgical resection, and SRS is discontinued for anything over 6, or discouraged, sorry, anything over 6 centimeters. Smaller brain metastases are also associated with less radiation necrosis after SRS. So take a look at this study here. We have 160 patients with brain metastases from the 3 major primary sites within the body metastasizing to the brain. And these are the 2 slopes that we need to be looking at. So these are the metastases over 1.5 centimeters in diameter. And these here are the ones under 5 millimeters. And you can see that for every time point, the probability of radiation necrosis is much higher for the larger tumor size. At the same time, smaller brain metastases are associated with less treatment toxicity. So we see here the green arrow on the left, that is decreasing tumor size as we go from bottom to top. And on the right, the orange arrow, we have a decreasing percentage of patients with toxicity as we go from bottom to top, i.e. smaller tumor size. If that hasn't fully convinced you, then consider this. So screening with MRI for patients with metastatic melanoma. Here, 116 patients included in this study from 2021, and a third of all brain metastases were detected asymptomatically by 6-month screening, which has the potential to change management in affected patients. Now, I know what you're thinking. Screening isn't very cost effective, but it can be cost effective in certain scenarios, such as these patients here. These were patients with brain metastasis treated by upfront radiosurgery. They then compared those that were followed to those that were not followed, i.e. became symptomatic prior to imaging. And the symptomatic patients required more craniotomies, longer hospitalization rates, more likely to have post-treatment toxicity, and required more on average to manage on a per-patient basis. Gadolinium deposition is also a concern, but when we consider the next generation of gadolinium-based contrast agents, gadopiclinol here, which requires only 50% of the actual gadolinium metal itself on for a full doses when compared to the prior generation of contrast. Interestingly, images with gadopiclinol are preferred qualitatively and quantitatively due to higher relaxivities. So at the end of the day, next generation gadolinium contrast agents, less gadolinium, better lesion depiction. So to summarize the pros for screening MRI for brain metastases, asymptomatic brain metastases are associated with better treatment outcomes. We have reduced neurologic emergencies. Screening can be cost-effective in certain scenarios. MRI techniques are improving. MR contrast is improving. But at the end of the day, we do need prospective randomized trials to evaluate the benefit of screening MRI in terms of overall survival. Thank you. Okay, so in the next 5 minutes, we are going to look at the pros of screening MRI for brain metastasis.
Once is enough, then it's unfair.
So unfortunately, Dr. Hainz is not here. He has shown us his arguments pro MRI screening, but who is here is Volkmar Müller from Hamburg-Eppendorf University Hospital from Germany, and he will tell us his arguments contra routine MRI screening. Please.
So I must say I have a conflict of interest because I see the patients, and it's obviously that my previous speakers has some bias on using these machines, maybe, but I think we're going to find a nice compromise in the end. So of course I don't have to tell you that we have an increasing problem, and we are facing patients that will develop brain metastasis. So I think we all must consider this, and of course I have a contraposition, but I will of course be open for discussion in the end. So just to repeat the clinical question, should we perform routine MRIs on every asymptomatic patients in the sense of screening and of course staging? And I'm going to focus on breast cancer. As you might have seen, I'm a gynecologist and I'm involved in breast cancer treatment, but I think it's a good example for everything else we will discuss in my next one. So I looked into the guidelines, and you don't have to read them because none of the guidelines I found is really recommending routine screening in the context of breast cancer. And there's going to be a change in the ABC guidelines, for example, that's been under revision now, but it's not going to recommend screening for everyone, I can tell you already. So as this is an open question, of course, there are ongoing trials. Otherwise, if everything would be clear, there wouldn't be ongoing trials. And these were the trials I found, several trials, some of them just examining the incidence, and that's, I think, a challenge, and we would need to discuss that in the end. Some of them also combining this with an intervention, and that's what we need. And also the outcomes. I might say that overall survival is always demanded, but maybe it's not the good endpoint, although I should argue in this way because we're never going to see that maybe. So there's one trial of these I had shown you that was already reported last year. You might have seen that. And it's just the incidence. So it's describing, of course, not very surprisingly, that in high-risk breast cancer patients, we see higher incidence, and this is increasing if you perform repeated MRI over time. But still, this publication ends with the conclusion, we have to do additional trials. So it's not really a solution so far. And there's another publication I found, which is a cohort comparison between patients with symptomatic and asymptomatic patients. And you might have seen the slides that my previous speaker colleague showed a publication from our group, but this is retrospective data. And of course, you can see that the lesions are smaller, but here's the outcome in overall survival, and this is not altered. It's no difference between asymptomatic and symptomatic patients. So you might conclude, at least in breast cancer, older cohort, also room for discussion, but that in breast cancer, the survival is not very limited by brain metastasis for many patients. Still, I think you have to consider it. So, no demonstrated survival benefit, no randomized trials, and these retrospective cohorts that were also published that stated in survival benefit, of course, were always not able to rule out the lead time bias. So early detection doesn't always mean better outcome for patients. And of course, I don't have to tell you there's a harm, a potential harm. It's the stress for the patients. So you have to inform them about that you're doing a procedure that is not recommended. And there was the argument that lesions that are detected early can be easier, have radiation therapy. That's one point, but also you have to consider that you have to do more radiotherapy, even not at the same time point, but retreated radiotherapy. So then I was happy about the previous talk, also about MRI and the limitations of capacity. So who really should do all these screening MRIs and then, of course, discuss with the patients. So they are, Frank was mentioning the German guidelines, and we had this discussion also there. We need qualified centers for these patients, because if you have a finding that is not leading to symptoms, you don't have to treat immediately. And we will see systemic therapy options for these patients. So there's a practical, I think, hurdle for this implementing, and I think we should also consider this. And then, of course, what is done in the clinical practice. So you could say this is the guidelines, but all my colleagues do this. This is a survey that was initiated with colleagues from my group also. And we did an international survey about the practice on screening for brain metastasis and breast cancer. As you can see on the left, there's 13% of our colleagues who often or always do the screening. So this is not a majority. So I think I don't have to convince the majority of you not to do routine screening. That's my conclusion. No survival benefit, potential harms and high costs. We need prospective trials. I think this is I was not so happy to write it in because I think this is only half of the way. So we also need to consider the patient's wish. And this is something we might discuss. So we're looking forward for the discussion. And thanks a lot for the invitation and the great talk so far. Thank you.
So Marion Smits takes over the part of pro-screening. So
no surprise.
Thank you very much for that. Yeah.
I had about 2 seconds to change my position here. So
I think we are all a little bit in between. So I think it's easy to.
I'm really confident.
So this stage is open for discussion.
First, because Hélène Bulbeck, you're listening. So maybe you could start and give us your opinion of the patient's representative point of view of the screen MRI. What is there? You need a microphone.
Thank you. So I run a national brain cancer charity, which is very patient-facing, very supportive in the UK, and I think we're probably the only brain tumour charity that supports and grows a community of people living with brain metastases. And we meet every month, and every month the topic is MRI scans. There is no standard protocol, and I don't know what the solution is. I've spoken to the Royal Marsden, but they are constantly living in the shadow of scanning, either for their primary cancer or for their brain mets. And then, of course, when they've had the scan, they've got to wait for the results. And I've spoken to various centres and, they've said, well, there's the issue of gadolinium. You can't have more than one scan in one day. So there has to be a workaround, a solution. But at the moment, people living with brain mets are being completely defined by their brain metastases because they're always living in the shadow of MRI scans.
Yeah, I think that's fantastic that you give your perspective. And I can mention that for the survey, I was in the contra part. So I could have also shown for the pro part, the part we did that also with patients, and maybe you also contributed because I can tell you there were more patients answering this survey than physicians. So it was about 600 patients. And the question was considering you could had a MRI screening routinely to detect brain metastasis, although it is not known if it's an advantage for you to detect it early. Would you like to have a brain MRI screen? And there was a majority that really would like to have an MRI screen. And then there was another question: would you like information about the possibility to get an MRI screen to detect it, although this is not known? And then a huge majority of the patients that answered the survey, and that was not only Germany, that was Europe, even Canada and US, said, yes, I would like to have information. So what we are currently doing, because we did the survey last year, is we try to get some information on the ABC platform about the potential that an MRI scan with asymptomatic situation might be a benefit, but it could be also harmful. And we're doing this together with some patient advocates, and if you'd like to contribute, that would be very, very nice.
I don't think. Thank you. Every patient has their own appetite for risk, and they have their own context and they have their own values. And it's the same with primary brain tumours. People don't understand the risks of introducing a screening program. So it comes down to having that personal conversation with each patient and their family.
Yeah, I think
I have people with a glioblastoma who are living a really good quality of life, and they decide not to have a scan because, you know, it can just transform how they perceive their daily activities if they know there's progression. Whereas the caregivers tend to put pressure on the patients to have scans because they want to know what's going on.
That's a very good perspective. I try to avoid this and in the clinical practice, but yeah.
Yeah, I think this is an excellent comment and contribution, and I would say I agree. So I always tell patients, so you can turn every healthy person, just making a whole body MRI scan, you can turn every healthy person into a non-healthy person anymore. Yeah? A lesion, maybe something, maybe not, etc. Right? So this wonderful manager whole body MRI scans, I think, is one of the worst things that are done to the poor managers paid by SAP and others, right? So they have a lot of then people concerned with potential lesions, well, we just follow up on them. It's a great money maker system for the radiologists, I agree, but probably not so much for human beings. So I think on the other hand, when you have showed this nicely, it's clear that there is potentially even a prognostic factor, right, of having a clinically asymptomatic brain metastasis. And then, of course, in the time of tucatinib and antibody drug conjugates, etc., you can early on make more informed treatment decisions when you know this cancer has the tendency, the propensity to spread to the brain. So I think in the end, but I still would, because we don't know, we need these trials. We need really trials to compare this, very clearly prospective, randomized, and then see whether we get in the end a survival and quality of life. Then quality of life is super important, yeah, and probably not just the typical quality of life, but also questionnaires, etc. That really cover what you just mentioned, and then we can probably get an idea on that. As long as we don't have that, I'm not seeing these on the horizon. I think it's very fair to discuss this on patient basis. I would do the same with the patient to just understand the specific needs and. We know that some patients are much more concerned not knowing, and some are much more concerned knowing, and that's a very specific individual, I would say, trait.
And that's our challenge as humans and as clinicians, and I'm happy to hear this because this is the sense we try to get into the guideline, I think.
Yeah.
Can I just comment on the radiologists make money thing? So you're not jealous, yeah, okay. Yeah, well, you could have all become radiologists and sit in our dark room with me, so come on. No, I mean, it will be different in every country, but in the Netherlands and also in the UK, radiologists do not make money out of, these extra scans, and actually oppose the extra scans as well, because like I said, we can't manage this at all. And if there's one thing I hate most in reporting is futile scans. So the negative scans or the scans where I have this tiny little dot and then I know that we're going to discuss this in the MDT and say, do you know what this is? And I don't know, it's a new dot of enhancement, could be anything. We follow this up, is something that actually really, I find that really troublesome. And I think most of my colleagues do. We're not in it. I mean, some people may be in it for the money. The other thing I wanted to say is that things might change, of course, if technology are being helped and reports are available straight away. So you take at least that uncertainty or anxiety waiting for your results part away. And that might, I don't know, that might change perspective to say something pro, because that's apparently what I need to.
So for me to say something pro, what I observe that, of course, we have to agree on what is screening MRI, because you do, for a patient with metastasis and metastatic setting, you do this PET or this CT, and you look at everything except the brain. So why? Because you know they are at risk of brain mets. So why don't you want to look at the brain? This is something that I don't understand. And also this argument with whole brain radiotherapy. Now we don't do it anymore. So because it's not efficient, but now we have other drugs. So for me, it's really, why not, when you have an indication for a follow-up with CT or PET, why don't you want to look at the brain? And maybe the solution will come from nuclear medicine, because at the end they will have markers that show everything. And Natalie wants to say something. I'm sure to support me.
A marker that shows everything. That's
full support, Emilie. If you're arguing for PET, of course, I'm supporting 100%. Now, I think it's a very very important aspect. So, in the past, we have done all the staging PET CTs with FDG as the PET tracer, but you have such a physiologically high uptake in the brain that you really cannot see any metastasis in the brain with FDG PET. But what is coming next? So to also give an outlook on future developments in nuclear medicine, so FAP-directed tracers, so fibroblast activation protein, this is something that is really en vogue and really coming. And so I can perfectly imagine, because you don't have any uptake of FAP tracers in the healthy brain, and also because it cannot cross the intact blood-brain barrier, you will see very nicely the brain metastasis in these scans. And therefore, I think for the future, probably staging PET CTs are going to be performed more and more also with FAP-directed tracers, where you can, in one scan, see the entire body tumor load.
So you keep the debate.
Yes. Maybe one more thing to add to that. So I think I totally agree. I think as we are moving towards in screening programs as well of healthy populations, is that we want to identify those risk groups, right? So that you're doing risk-based assessment and therefore adapt your screening programs to that as well. And you can imagine that certain tracers would indeed, or certain whatever, liquid biopsies, etc., could help you identify those groups and indeed relieve the burden on patients having to go through, routine follow-up.
Okay, so thank you very much. Maybe a very last question. It's a clinical question. So in the German, also other guidelines, the MRI scan is performed if the patient is becoming symptomatic, if there are symptoms. Who is deciding that a patient with breast cancer is asymptomatic? Are there neurologists involved, neuropsychologists? Who is diagnosing the slight decline, which shows there must be mets?
I was prepared for this question because you asked that during the guideline discussion. So I'm happy that I heard it before, because my answer would be everyone has to listen to the patients. And that is something that doesn't require necessary neurology training. So I think our nurses, if a patient complains about headache and she has HER2 positive breast cancer, even the nurses know, well, there's something might be going on. You should send her to an MRI. And of course, the threshold for doing an MRI needs to be very low. Otherwise, you catch up patients with symptoms. But if you have so many additional patients, if you're doing with low symptoms, and doing staging and do it now would require, might be in the future different, but now would require an additional MRI with all these difficulties. And that was described. You have unclear findings. I don't have to tell you. And then I just briefly mentioned, I mean, there's a gap between our centers and the reality. And even in Germany, there's, I think, a good infrastructure. If you ask colleagues how many have really a neuroradiologist on their board to discuss these findings, and I don't have to tell you, but I'm always impressed about the difference between the interpretation of general radiologists of brain MRIs and then the discussion on the tumor board. And there's so many things that really differ. I can't imagine how to integrate in every staging in brain MRI. So I think it's we have challenges in really assuring quality for those patients that get an examination. And I think that was really a brilliant commentary from the patient side.
So thanks for this answer. I think in interest, a very, very final, okay, ultra final.
In the discussion, I think it's important that you showed a paper from 2003 about no association between survival and screening. So I think we really have to wait for the trials that are being done. And I would like to stress that in the future, I think there will be specific primary tumor types, subtypes that have a high risk on brain metastasis. And maybe in these cases, we should do the screening. And I would also think that we need screening when there are really subtle neurologic symptoms. And then you need a neurologist.
We always have, of course.
So thank you very much. I did not expect that we could solve this problem within this discussion, but thank you. Yeah, we're coming step by step. We are coming forward. Yeah, but thank you for this very open, very interesting discussion. We're going for lunch now, and we'll be back at 1:40. Okay.
I'd like to ask my colleague, Dr. Voglis from the Neurosurgery Department of University Hospital, to present his data on perivascular niche under immune checkpoint inhibition.
So I will talk about the evolution of the brain metastasis perivascular niche under immune checkpoint blockade, our ongoing work. So the idea was to look a bit better at the vasculature and brain metastasis, since we know it's the major obstacle not only for systemic drugs, but also for the immune response. And obviously, we also know that the contrast enhancement by gadolinium in the MRI scans is just an insufficient proxy for the leakiness of the blood-brain barrier. So there have been two major studies by the Joyce group in last year and I think 2 years ago. They were, I think, the first who really looked into in depth at the vasculature, so at endothelial cells, but also mural cells, and not only saw a profound alteration in the transcriptomic states between brain metastasis and primary tumors, but especially in brain metastasis, that the vasculature was increasingly had an increasing permeability and also increasing immune cell interactions. So we had some own data on the spatial organization of the T-cell infiltrates in melanoma brain metastasis, and that those were prognostic for immune checkpoint inhibitor response and survival. And that basically started the idea to look a bit better how immune checkpoint inhibition itself alters the vasculature and how this might also contribute to the spatial organization. So therefore, we assembled a cohort comprised of 3 different technologies. One is single-cell RNA sequencing of publicly available data sets for brain mets. The other one is spatial transcriptomics, so spatial technology. And the last one is spatial proteomics, or highly multiplexed imaging, which was exclusively done on melanoma brain metastasis to tackle this question. So first, a few words about our exploratory RNA-seq dataset cohort, which was a bit in line with the previous studies, which I mentioned, that there was indeed a large heterogeneity between the transcriptomic profiles of vasculature compartments between brain mets and healthy controls, but also in between brain mets from different primary tumors
And the usual suspects like proteins which are involved in the integrity and also endothelial transport processes, but also drug efflux transporters or cell adhesion and chemotaxis molecules. Since we were interested in how immune checkpoint inhibition affects the vasculature, we first looked at samples which were pre-treated versus the ones who had never seen checkpoint inhibition before resection. And on one hand saw different abundances of endothelial and mural cell proportions with lower abundances in the pre-treated samples, but then going a bit deeper into the cell subtypes, for example, for the mural cells, dividing them by to cancer-associated fibroblasts, but also smooth muscle cells and pericytes, we saw no differences. But looking at the functional annotation again, we saw what one would have assumed, that in the pre-treated samples we saw an upregulation of genes linked to immune signaling, but also transport processes over endothelial cells. Looking at the cell-cell communication, here in the upper row, you see the ICI naive samples. In the lower row, the ICI pre-treated samples. We indeed saw an increased communication, so sender to receiver communication between endothelial cells and different T cell subsets, among those also CD8 positive effector T cells. And most of the pathways which popped up again were involved in chemotaxis and cell adhesion pathways. So now next, we were lucky enough to get our hands on the spatial transcriptomic genome data set from Christian Lucia and the Spatial Biology Lab from Frankfurt. Those were also divided or stratified by ICI pretreatment or ICI naive samples. And the main advantage of those samples were that they really span a huge area, which you see here, for example, a lung brain met, or the blue dots are thousands and thousands of tumor cells, and then you see in between all the different in red vascular compartments, and that gave us the idea to look at what we call the perivascular density-based niche calculation. So we wanted to know, I think it's quite nicely shown here, how are the densities of endothelial cells or vasculature in the different areas of the tumor. And then based on that, computationally, we contoured or assigned those areas to either being density low perivascular areas to density high perivascular areas. And here in an ICI naive sample, you nicely see that with increasing vasculature density, you also see an increase of immune infiltrates, among those also, again, lymphoid cells. Now looking at our two groups for the ICI pretreated and naive samples, in the naive ones, you nicely see this increasing density or proportions of immune cells, and in the pretreated ones, you even see in lower density perivascular niches quite similar high influx or proportions of different immune cells. So that prompted us to look a bit better into those low density perivascular niches. So in the areas where there are not so high densities of vasculature. And therefore, we first looked at different endothelial cell markers, since we had the opportunity for those 5,000 different markers. And indeed, some endothelial cell adhesion molecules popped up, which were more pronounced in the low density regions in the pretreated ones compared to the high density regions from the pretreated ones, and other, like VCAM1, were exclusively expressed in the high perivascular density regions. And that was somehow also confirmed by the functional annotation. So in the low density ICI pretreated samples, we saw most of the genes were associated with cell adhesion, again, transport, vascular development, and remodeling. Last but not least, we had an imaging mass cytometry data set of melanoma brain metastasis. Here you see again two exemplary pictures. Blue are the melanoma cells, and in green are the infiltrating lymphocytes. And based on our previous data, we were here interested in how the, especially the CD8 positive T cells infiltrate into the tumor. So are they either dispersed, so more infiltrative between the tumor cells, or compartmentalized, or we call it localized, sitting next to the tumor, here in blue, the tumor cells, and not so much infiltrating it. So that's what we computationally modeled. Here you see some exemplary regions for ICI pretreated tumors, where you appreciate, at least in some instances here more infiltrative, dispersed phenotype of the green lymphoid cells, and here in the naive ones, a more compartmentalized or localized infiltration pattern. That was also confirmed on the whole cohort, on the sample level. And since we saw these differences in the spatial arrangement in those infiltrating cells, we also wanted to know how is this in relation to the perivascular niche. So we did some measurements calculating the distances from the different tumor and immune cells from those perivascular niches. And to make it short, in the pre-treated samples, we saw in almost all immune cells, but especially also in the lymphoid compartment, increasing distances. So increasing distances from the perivascular niche, which was also confirmed when you statistically model this. And this brings me to the summary that we have at least hints that immune checkpoint inhibition induces a remodeling of the brain metastasis perivascular niche and also the spatial arrangement of the TME. And more precise, it seems to trigger a functional and spatial shift in those niches and allows also niches which were previously restricted to lymphoid infiltration to infiltrate those. And it also promotes a transition, or seems to promote a transition, from a more localized to a more dispersed lymphoid or in general immune architecture. This brings me to the end, and I would like to thank all the collaborators.
Thank you very much. Again, very interesting data that we will discuss in the panel discussion after the third talk about brain metastasis immunity given by Anna Salomero. Welcome.
I hope you can hear me well. Okay. So thank you very much for allowing me to present this data that I was doing during my PhD with Lizza Hendriks in Frankfurt and in Tübingen. So today I'm going to talk about adenosine pathway in the context of brain metastasis. So in this laboratory, we had a very strong interest in targeting the brain tumor microenvironment, which we know it's very diverse and consists of different immune cell types, both brain resident and also ones that are recruited upon the presence of a tumor. So what we wanted to do was to remodel this brain tumor microenvironment that is usually immunosuppressive in the context of brain metastasis into an environment that is more linked to anti-tumor response and also more to pro-inflammation, so that hopefully we will be able to see a reduction in tumor growth. So for that, we were checking the literature, and it comes to our attention the possibility to target the adenosine signaling pathway, which mainly consists of different ligands, mainly ATP, which is supposed to have a much pro-inflammatory function, and also adenosine. That adenosine, upon the binding to the adenosine receptors, is supposed to be more related to the immunosuppression. So in the cancer field, there has been a strong interest in targeting this pathway by reducing the amount of adenosine, so targeting the adenosine converting enzyme CA39 and CA73, but also by targeting adenosine receptors such as adenosine receptor 2A and 2B. So since we wanted to also study what were the effects of targeting this pathway in the context of preclinical tumor models, what we did was to analyze the presence of different components of this pathway in the preclinical setting we had of mouse with brain metastasis. And what we could see is that the enzymes that are responsible for the conversion to adenosine, such as CA39 and CA73, were upregulated in some immune cell types that are detected upon the tumor. And the same holds true for adenosine receptors. We also verified those findings based on the RNA sequencing at the protein levels, and there we could see that in this preclinical model of mouse brain metastasis, there was a higher expression of CA73 in the tumor compartment, while CA39 was highly expressed in myeloid cells such as microglia and macrophages, and the T-cell compartment expressed mainly CA73. So with that in mind, we wanted to evaluate the effects of targeting the single components, so CA39, CA73, and adenosine 2A in mouse that had brain metastasis. And what we observed was that the single targeting of this pathway at this level didn't improve mouse survival. Neither did that the combined treatment of like C39 with C73, C73 with A2A, or C39 with adenosine receptor 2A. However, it was a triple combination treatment of targeting C39, C73, and adenosine 2A inhibition in established mouse to brain metastasis that prolonged the survival of those mice. So since we knew that this condition was favorable, and we also are aware that most patients can or are eligible to receive radiation therapy, and the fact that radiation therapy also induces the death of cancer cells, and then it can release the amount of ATP. What we also wanted to do to combine this adenosine pathway inhibition also with combining it with radiation therapy. And what we saw is that when we do that in this preclinical mouse model of brain metastasis, we do see stronger mouse survival into those mice. And when we look at the tumor growth, we see that those mice with brain metastasis that are treated with the inhibition of this pathway at these 3 different levels and whole radiation therapy, they have a reduced tumor growth, suggesting that there might be some effects that are combined both by radiotherapy and the targeting of adenosine pathway that can account for these differences. So when we analyze the tumor microenvironment of those mice that are undergoing these 4 different treatments, we observe that there is an increase of the amounts of CD8 T cells into those mice. And also when we look at the bulk sequencing analysis, so transcriptomic analysis, we also see that there are more pathways related to adaptive immune response into those CD4 T cells, for example, of mice that are treated with adenosine pathway inhibition and whole radiation therapy, suggesting that some of the effects we see in tumor control have also to do with the remodelation of the tumor immune compartment in brain metastasis by targeting this pathway in combination of radiotherapy. So we also wanted to explore what were the effects of targeting this pathway at the immune compartment, like more broadly. And for that, what we did was to analyze CD45 immune cells. And it was striking to us to observe that also in relation with the literature is that upon whole radiation therapy, there was also a decrease of the presence of exhausted T cells. And this was also accompanied with an increase of effector T cells, such as natural killer T cells and some KT cells, tissue resident memory T cells, and gamma delta T cells. So cell types that have been linked to having an effector phenotype and also to be able to control tumor growth. And this was observed in the condition of whole radiation therapy with and without the inhibition of the adenosine pathway. When we look at the macrophage compartment, we were able to see that in the presence of whole radiation therapy, there might be a compensatory effect that is linked more to the presence of lipid-loaded monocyte macrophages, whereas in the combination of whole radiation therapy and adenosine pathway inhibition, there are also more macrophages that are supposed to have an antigen-presenting capability. And when we then interrogated the cell-to-cell communication, what we can see is that in those mice that have brain metastasis and are treated with the inhibition of this pathway with whole radiation therapy, it seems that there are more communication with antigen-presenting capacities with the grams and KT cells, suggesting that those cells are not just there, that they are also active. So in summary, what we could observe in this mouse model was that the inhibition of adenosine pathway inhibition with whole radiation therapy led to the remodelation of the tumor microenvironment by changing the presence of the different T cells and also different subsets of macrophages. And this was interesting to us, but we also wanted to assess how was this pathway in tumor microarrays of brain metastasis. And for that, what we did was to do the staining of these enzymes that convert adenosine, as well as to adenosine receptors, and also adenosine deaminase that drives adenosine into inosine and adenosine kinase. And then we evaluated the staining as intensity and also by the amounts of the different tumor cells that are stained with that. And what we could see is that the single expression of CD39 correlated with patient outcomes. So those patients who have higher levels of CD39 in the tumor cells tend to do worse compared to the other ones. And this was also correlating with the immune cell infiltration. So the higher CD39 in the T cells, the less immune infiltration into those patients. And the opposite holds true for adenosine deaminase, that both kind of have the opposing effects when it comes to adenosine as CD39. When we also look in a more unbiased way into this staining, we were able to identify the presence of 2 clusters, being cluster 1 majoritarily expressed by CD39 adenosine receptor 2B, and this also correlated with those patients that had worse probability of survival. As well, this also correlated with those patients that have less infiltration of T cells. So in summary, what I could show you here is that the pharmacological targeting of the adenosine pathway inhibition by triple treatment of CD39, CD73, and adenosine receptor 2A in combination with radiotherapy led to adaptive anti-tumor response in a mouse model of brain metastasis from breast cancer, and that also the analysis of tumor cells in human brain metastasis reveal a correlation of between adenosine pathway component survival and immune infiltration. So for that, I'm coming to the end, and I would like to thank my supervisor doing my doctorate, Professor Lizza Hendriks, as well as all the collaborators, so Patrick Harta, Cal Platter for providing us with the human sample, as well Franz Goder for the radiation scheme, and Katharina Keller especially also for the bioinformatics analysis. And with that, I'm happy to take any question. Thank you.
Good, thank you very much. I would like to ask actually the three presenters to join us, because we are happy to open the discussion. And I would like to ask everybody if somebody has a question on to our very interesting studies. Okay.
Yeah, thank you. Really interesting talks. One question to this perivascular profiling. So you might know that Thomas Welchley, a Swiss neurosurgical colleague of you, is now in London. He has published in Nature 2024 these oncofetal programs in endothelial cells, right, that apparently seem to hinder. So programs that are up in development and also upregulated again in the tumor, they seem to hinder, let's say, in development, but also then in cancer, that immune cells extravasate in meaningful numbers into the brain. The question is, have you checked endothelial cells after checkpoint inhibition? Is that changing? Have you checked these oncofetal programs and endothelial cells? You have done a lot of single cell analysis. Have you talked with Thomas?
So no, we have not checked. I mean, we know the study, we know the resources he published. I think the major difference is because we were mainly interested in the effects of immune checkpoint inhibition, and therefore we didn't have that much sample at hand. So we only took those samples where we had an annotation available, whether they were at the time point of resection under checkpoint inhibition or not. And additionally, I think one would have to prospectively collect samples and then enrich them for those cellular compartments to really check for those distinct programs he described in his data set. Because I think he also used like pre-enriched samples for vascular compartments.
Yes.
Hello. Thank you for the great talks. I have two questions for two of you, and very basic questions. So for the TLS, I know it's hard to get a sense of this with this kind of study, but do we have any idea of the kinetics of because you mentioned, like, for example, in some tumors, it's possible to see the presence of the FDCs and, have a mature TLS. But for brain metastasis, from the advent of a brain metastasis or, in the advanced stages, is there a sense for the kinetics of the formation and maybe resolution of TLS?
Yeah, thank you for that question. So in the, of course, in the patient cohorts, it's like a steady state we see. And we've noticed in the glioma that there's indeed different, the certain patients, they have a preference for less or more mature TLSs, so to say. And we cannot judge anything about the development. But there's animal studies that have been done by Anna Dimberg's lab in Uppsala. And there's also a study in work in progress where they looked into that, similar to what we have done, but also complementing with animal studies. And some of the early factors, these early recruiting chemokines, CCL19, 21, and so forth, they are important for the initial recruitment. And also the quality of the vasculature. I mean, of course, you have angiogenesis going on, and in the study by Joanna Joyce, it's clear the vasculature is different in glioma versus brain mets. And we have not seen any of these early HEV markers in glioma and in brain mets, or very, very rarely. So we don't know if there's compensation or how this occurs. So it's still open.
Thank you. Yeah. One more question about the perivascular niche. I think it's a very nice segue. Do we, because as in the Joanna Joyce's paper and other papers, do we have a sense for what phenotype of T cells are more likely to cross into the, because I think that the dogma I know is that activated T cells cross into the blood-brain barrier. Are there any, like, because you've been able to do the spatial analysis, are there any like pre-markers sort of to say, oh, this T cell with this makeup might make it into the, I know it's a very weird question, but thank you.
I think it's a very good question, but we couldn't look into that because most of the samples were not longitudinally matched before and after. So it's from different patients. And I think that's a major obstacle in brain mets because they so rarely get re-resected. So we have only very few ones who are matched in the same patient.
Okay, thank you very much. In terms of the time, I would like to thank everybody for your great efforts in basic science. Keep going like that. And I would like now to change from basic science a little bit more to clinical studies. And I'm very happy to ask my Dr. Rogers from Cantonal Hospital Aarau to present her study on the pre-op randomized trial.
Yes, thank you very much for the invitation to present an update on the data so far. This is very clinical, that's my disclosure. So I think for most of us, the current standard approach for brain metastases that require resection is still resection followed by stereotactic radiotherapy to the cavity. The problem is this has no impact on the future development of nodular leptomeningeal relapse, reported between 10 to 20% across various series, 13% in this meta-analysis. The bottom case is sort of an extreme example, but after resection of this brain metastasis from neuroendocrine carcinoma, the patient developed subsequent ipsilateral nodular metastases. We were able to salvage, but best avoided if possible. To this end, there's a compelling rationale for preoperative radiosurgery. It can sterilize the tumor cells that will be disseminated at neurosurgery, impairing their capability to develop in the leptomeningeal space. It's much more precise to contour an intact metastasis than an irregular cavity. This can contribute to improved local control rates. This increased accuracy enables us to reduce the clinical and planning target margin, so less irradiated tissue, which is subsequently resected. So both of these can contribute to reducing the incidence of radionecrosis. There's also less delay to systemic therapy postoperatively, and it's highly attractive for patients to have a single fraction in the few days it takes to organize neurosurgery, as opposed to coming back 10 days later for planning and treatment over 2.5 weeks. Many centers have explored or adopted preoperative radiosurgery without any prospective evidence to date. This means that there have been multiple retrospective series published, resulting in 2 meta-analyses last year and the year before. In 25, 4 comparative studies were published in a meta-analysis, 221 preoperatively treated patients, 400 postoperatively, and they did indeed confirm a significant reduction in nodular leptomeningeal relapse in preoperative radiosurgery. 1-year local control rates of the cavity were equivalent, and 6-month radionecrosis rates were also better. In the second meta-analysis, over 3,000 patients included, 6 preoperative series, 33 postoperative series, so there was much more heterogeneity in the studies, but they also confirmed a statistical decrease in leptomeningeal relapse. But in this case, local recurrence was better, but radionecrosis was equivalent. But their own conclusions were, while these results are intriguing, we need randomized trials for genuine validation. So I believe we still need prospective data. Preoperative radiosurgery is coming into algorithms such as these, and we saw a sneak preview today of the new S2 guidelines. But as here correctly stated, that the optimal combination, fractionation, sequencing, interval between radiosurgery and resection is still needed to be tested in clinical trials. So in an initiative to generate some prospective data, we first undertook the PREOP-1 single-arm, single-center feasibility trial. We wanted to optimize the workflow before launching a randomized trial. We included 13 patients, 12 received preoperative SRS and did not develop any nodular leptomeningeal relapses. The 13th patient unfortunately bypassed us and went straight to theatre and did develop a nodular leptomeningeal relapse at 6 months, so this turned out to be a mini proof of principle study. And we also combined these data with retrospective data from the Peter Mac in Australia, from Korea and Canada in the INTERNEO independent patient pooled analysis. And we were able to show at 12 months very high local control rates, very low nodular leptomeningeal relapse rates, and very low radionecrosis, which was also sustained at 24 months. So we're currently recruiting into the PREOP-2 randomized clinical trial. Eligible patients have a brain metastasis for resection up to 4 centimeters in diameter, a gross total resection predicted by the neurosurgeon, up to 3 other brain metastases for primary radiosurgery, and a known cancer diagnosis. And the primary endpoint is time to leptomeningeal disease. Arm 2 is the standard of care, postoperative fractionated stereotactic radiotherapy. Patients asked to complete validated ERTC quality of life questionnaires at baseline, proceed to surgery, a postoperative MRI to document the extent of resection. Either, as I do, 5 times 6 grade to the cavity, but as we couldn't achieve a consensus, the local standard of care is also acceptable. 6 times 5, 3 fractions, occasionally 1 fraction are also popular. And then patients have follow-up MRIs, 3 monthly as standard, with the quality of life questionnaires for 12 months. Arm 1 is the experimental arm, similar baseline quality of life questionnaires, then a single fraction before resection, and postoperative MRI and follow-up as before. About 18 months ago, we performed a planned interim analysis. We wanted to assess feasibility in a multi-center setting, regardless of prior experience with radiosurgery, center size, platform. And preoperative SRS was performed in all 100% of the 21 patients who at that time were in Arm 1, and postoperative fractionated stereotactic radiotherapy could also be delivered in Arm 2. We also looked at toxicity. At this time, we had 19 SAEs that were all grade 3. There have been no grade 4 or 5 toxicities, and 11 of these were neurological. 9 were possibly, and 2 were probably related to either the radiotherapy or the neurosurgery. But looking at these 11 in more detail, only 1 was in the preoperative arm and 10 were in the postoperative arm. And 12 months later, we did a follow-up analysis, had 14 neurological SAEs, all grade 3, but with the same pattern, 2 preoperative and 12 postoperative. So no red flags for preoperative radiosurgery in the prospective setting. So in conclusion, single fraction preoperative radiosurgery is safe and feasible in the trial. All the retrospective data consistently show lower leptomeningeal relapse rates and equivalent or better local control and radionecrosis rates, but that prospective data are needed to elucidate the optimal fractionation and timing. Just a word to the trial. We're continuing to accrue. It's a highly pragmatic trial. It's essentially comparing to standard of care. There's very minimal administration. There's a modest patient fee. We're at 73 of the 200 necessary patients. Every time a centre opens, there's an upkick in the recruitment curve. We were monocentric at the beginning. We were oligocentric a year later. I think we're now multi-centre. Q1, we're looking forward to opening in Bellinzona and Köln. We have additional interest from 1 Austrian, 3 German, and 3 Dutch centres. And I think if we can open all these centres in 2026, we should be finished next year. The only requirements are a quality-assured, established single-fraction radiosurgery program and close cooperation between radiosurgery and neurosurgery. So thank you for your attention. Thank you very much to the participating centres, predominantly in Switzerland, but also Austria and Germany, to the patients who've agreed to participate in the trials, and to our sponsors. Thank you.
Thank you very much. We'll address any questions in the panel discussion after the next presentation. I think Lizza Hendriks may not be here, so I'll ask the co-chair of the trial, Emilie, to present the IVO-BRAIN trial. We're quite proud of it in the UTC brain tumor group, as it's been quite a struggle to set up a brain metastasis trial, but in the end we achieved together with the colleagues from the lung cancer group, and Emilie will show us the design of this upcoming trial.
Thank you, Matthias. So yes, Lizza, it's a bit delayed, so that's why she cannot present. So we'll jump in for her. So yes, that's URTC trial that is planned, so it's not yet started. That's the IVO-BRAIN trial, and that's a collaboration between the lung cancer group and the brain tumor group. So the background, of course, we are interested in brain metastasis in a patient with non-small cell lung cancer. We have current standard of care for patients without actionable driver mutation in first-line treatment with more or less chemo and immunotherapy. And there is always this question when you have brain metastasis, of the question of the timing of the radiotherapy. So when should we do it? Up front, maybe the tumor metastasis will be better controlled, or after, to avoid toxicity. And you have a lot of arguments, always pro and contra. And here there is this new drug, Ivozimab. IVO, it's easier. That has showed some interesting results, and also it could be interesting because that's antibody against PD-1 and also VEGF-A. And so it has showed an improved PFS compared to the standard treatment, and with also interesting overall response rates in patients with brain metastasis without safety issue, and reduced number of grade 3 bleeding events. So this is a trial design. So that's for patients with non-small cell lung cancer without genomic alteration, for which one line of target treatment is available. So brain metastases have to be asymptomatic, they have to be active, meaning it's active. It's not really a terminology that I like too much, but it's so newly diagnosed brain metastasis or metastases that are clearly progressing after first treatments, and we can prove really that they were progressing, so it's not stable brain metastasis. They have to be measurable. Patients in a good general status, 1 to 10 brain metastases, and so no previous systemic therapy for the stage 4 non-small cell lung cancer, no previous brain radiotherapy. And then so the patient will be randomized versus local radiotherapy, SRS, to all brain metastases, followed by IVO plus chemotherapy or IVO plus chemotherapy. And that's a 1-to-1 randomization with stratification factors regarding number of brain metastases, PD-L1 status, and histology. Here, the primary endpoint is really to look at the intracranial progression-free survival because of radiotherapy. Radiotherapy is a local treatment, so it would be unfair to ask radiotherapy on the brain to treat liver metastasis. So that explains the primary endpoint. It will be assessed according to this RANO-BM criteria locally. And as a secondary endpoint, intracranial PFS, that will be also centrally reviewed post-hoc. Time to next CNS intervention, OS, response rate, toxicity, with focus on radionecrosis, because of course with anti-VEGF. expect to have less radionecrosis. So you have the endpoint. I can go quickly maybe on this one, and that's planned study activation timelines. So protocol version 1 has been released already, and the regulatory submission are planned in May, so soon. So we anticipate to have regulatory approvals in September 2026. And first site active in October. And so that's study sponsored by URTC. So total number of patients, 158, 30 sites across 9 countries. And that's funding, study is funded by Summit Therapeutics. And so we expect to enroll the patients in 34 months. And this is a list of selected countries. So normally people who are participating should have been informed already. So so I think that's the last slide. So thank you for your attention.
I ask both speakers to join us. Great to see more and more clinical trials emerging and being completed in brain metastasis. Any questions to our speakers?
I have a question for you, Susanne.
Yes.
Congratulations that you're still not giving up getting it done. Can you share with us who the CRO is and who the sponsor? How does that work over the borders for your trial?
Well, we're the PI sponsor in Switzerland, and then we have, so I thought there would be a lead center in Germany. So Kiel Oliver Blank is the most active center. He had the ethics, and then I thought it would just cascade down as it does in Switzerland. But I discovered that every Bundesland has its own regulations. So it's really taken a long time. So each center is just opening locally with their own ethics.
But who's monitoring? I mean, how do you do the monitoring of sites outside of Switzerland? I mean, you see that we have thought about similar constructs also, and we found it very difficult to do the monitoring across the countries.
Yeah, so we actually scaled down the monitoring plan. So that's why there's the patient fee. The monitoring is basically going to be done locally. It's really standard of care. So obviously we confirm the informed consent and all the basic regulatory aspects, but that will be done by someone not involved in the trial locally. And then we're doing a central plan review and a central radiology review for the quality assurance of the endpoints. But the monitoring is, schlank.
I also have a question concerning the pre-op2 trial. Would it be possible to pull up the slide with the complication rate? Was not the last one, but the one before?
Can I just go off? No. How's it linked?
It's just that it went a bit too fast for me. But it was a comparison on complications or serious adverse events, pre-op versus post-op. And it seems that most of these things like subdural hematoma, repeat craniotomy, hydrocephalus, these are mainly surgical complications. So I really wanted to check or know why a repeat craniotomy would be more frequent when you do post-operative radiotherapy.
I don't believe there's a connection. So I think, possibly we're just taking any neurological SAE because we want to be as transparent as possible. But one of them, the patient fell out of bed, banged his head and had to have a second surgery because of bleeding. Was that linked to anything? Probably not,
Right.
So
Okay.
I'm not saying they're causal at all. I'm just being very transparent with any neurological SAE.
Okay.
I think the important thing was to show, I think people are concerned about wound healing, maybe with preoperative radiosurgery. Wound healing.
Wound healing, yeah.
That was a concern with preoperative radiosurgery, for example, predominantly. And it was really just to compare pre and post-op. But as there was no excess in the pre-op arm, that for me was a green light. But I think these are just things that happen still in small sample sizes. I think with larger patient numbers, maybe it'll balance out. But I don't think it's related to the radiotherapy in any way. No.
Okay, and there's no indication that wound healing or would be an issue in preoperative. Okay.
We did have that. We tried preoperative radiosurgery for glioblastoma,
right,
and 5 fractions, obviously much larger volumes and up to the skull. You know, these tend to be more deep-seated, smaller. We haven't had any issues.
Pre-op for glioblastoma. Okay.
Lisa, you can join us on stage. Thanks for coming. So the difficult questions now. You want to say something about the trial?
I hope the contract will be signed. Sorry, I hope the contract will be signed soon and that we can submit to the ethics committee and start activating sites in the meantime. So sorry, I had some delay with the flight. So
there's a question there. Come with the microphone.
Hi. I was wondering how the rate of radiation necrosis will be measured in the study.
For the IVO-BRAIN, it's according to the local investigator, but we will collect all images and it will, in the end, retrospectively, centrally be revised. But I think to have it pragmatic, I think you cannot do this in real time. So it's really according to the local investigator. And they can choose whichever imaging method they want.
I also have a question for you. What is the experimental arm and what is the standard arm?
I think that's still a huge discussion. And in the end, we didn't label it. We just stated A versus B to also avoid discussions about refunding the stereotactic radiotherapy. So it's a good question, and we stayed away from it because of financial issues.
Okay. Any more questions?
Sorry, I have a question for the pre-op study. So I need to declare I'm a dermat-oncologist, so I'm not a radiation oncology. And I have a question. So your primary endpoint is the time to leptomeningeal manifestation. So in terms of systemic therapy options, do patients have also systemic therapy? And do you think that because, you know, your primary endpoint is time to leptomeningeal disease, so do you believe that the systemic therapy do not actually delay the presence of leptomeningeal disease?
It might be. I think that those are very active questions at the moment. It's certainly documented. It's one of the some of the data that we're being collected. I think the outcomes will be interpreted in those contexts. We won't be able to say absolutely, but these are, on the whole, are patients who've developed metastases on their systemic treatment.
Okay. If there are no more questions, there is a question. Oh, Thomas. I'll run.
Yeah, good.
Thank you. I have a question regarding pre-op radiotherapy. Do you have a possibility to biobank a tissue? I mean, like that, have a tissue shortly after SRS is quite valuable for subsequent radiobiology, radiosensitive analysis.
Yeah, currently we're only banking the immunohistochemistry, so the paraffin embedded blocks. We did think about what we could do. In the pre-op one, we did a little bit of analysis. We didn't find any change in expression of markers, but we were only operating on a mean of 1 day post-radiotherapy. The Americans did report some changes. They had a mean of 2 days. So this is what I mean also about the optimal timing in terms of activating immune response or other effects. I would love a biological sub-study. It's not too late. It's not something that I can offer, but if anyone wants to collaborate, I have had a few conversations, then there's a lot we could learn. But the problem is we don't have a comparison. We don't biopsy these brain metastases before irradiation. And I think to compare with the primary tumor is also irrelevant. We know that there are so many changes. I highly expect that the radiosurgery will change something at the genetic level. There weren't any changes at the protein level in such a short time frame. I was thinking this morning about the tubules, you know, and maybe it's having an effect on that and not just the mitotic inhibition, which is what we're assuming 1 × 10 gray in vitro is enough to cause mitotic damage such that the cells can't then establish themselves. But I wonder if it's a lot more about the cell signaling. I'd like to look at calcium levels after this morning, all sorts of stimulating ideas. if anyone has the capability to collaborate, I'd be very pleased.
Lisa, we spent a lot of time discussing the endpoints of the trial, and we ended up with intracranial PFS period. So can you discuss what the considerations that we went into that, OS, response rate, and so on?
Yeah, I think the intracranial PFS is one of the most objective endpoints. With the caveat, I think that radiation necrosis is still an issue to distinguish from true progression. We discussed maybe overall survival is in the end the most relevant endpoint, but it will not be feasible also for timing and duration of the trial. We discussed response rates, but I think then you're automatically biased for the radiotherapy arm because these patients will respond. We also discussed time to switch of systemic therapy because in the end, I think you want to avoid that these patients progress in the brain and need to switch to another therapy because of the progression. But that's also very subjective. So I think in the end, we discussed, I think, all possible endpoints, and with all the caveats of intracranial progression-free survival, we thought this was the less biased and still a meaningful endpoint for patients.
Is quality of life and neurocognitive function included?
It's in the secondary endpoints.
Okay. Are there any more questions? If not, we thank our speakers of the session very much. I thank my co-chair, and it's time for us to hand over to the next set of chairs.
So we can proceed with our next section. I am Sarah, I'm the neurosurgeon responsible for neuro-oncology and skull base in Zurich. And it's a pleasure to welcome you all also from side of the neurosurgical department of Zurich. So we are in time and we can start, and it's a pleasure for me to introduce Professor Goldbrunner from Cologne, who will present a talk about neurosurgical approach on CNS metastasis. Where are we going? So,
thank you very much. Yeah. So thank you for the opportunity to share my 5 pence of wisdom with you all. It's about a simple question: what's the role of neurosurgery? But the answer is a bit complex. So you're very familiar with all the epidemiology data about the incidence and so on. I don't want to repeat all that. Just maybe it's in our center, we see an increase of gastrointestinal primaries. So it's more than 10% in our series meanwhile. And out of all the therapeutic options we have heard about or will hear about, it's about the role of resection. So there is a general role of neurosurgery. What are the principles? What should we achieve? What's a must for us? So the first thing is we need a safe approach to the tumor. We need to dissect it out of the healthy, more or less healthy brain. We know about infiltration, and we need a resection control to be sure that nothing is left over. We have our toys, which we like to use, the neuronavigation, the microscopes, the ultrasound, proving the resection control. Some use intraoperative MRI, but this is very time consuming. We prefer the ultrasound. And there is also another very important issue. I will come to that even later, the preservation of function. So absolutely must is to bring the patient in a very good condition after surgery. And so we have to know about the individual function, the localization of functions, especially tumors of in eloquent areas. We can use the functional MRI and integrate the functional data into the anatomical 3-dimensional data set we use for navigation. And we also can do transcranial magnetic stimulation, which allows even a more precise calculation where the individual functions located around the tumor might be or are. So there are a variety of classical indications for surgery, which are more or less without discussion. It's the single space occupying mass lesion, of course. Even if there are 2 or 3 mass lesions which are immediate life-threatening, we can remove them. We have been discussing about pseudo progression or the radiation necrosis. You can deal with this problem with different medications, but if it's really a strong edema with a mass lesion, you just can remove the problem. Then there is no more discussion about it. These are very old data from the early 90s from Spatial, showing that the resection of single brain metastasis makes sense. It's much better than radiation only. But if we agree with all these indications, there are a lot of open questions nowadays. What about multiple mass lesions? Not immediately life-threatening, but symptomatic. But there are different radiation regimes as an alternative, or there are different regimes with medication as an option. So the research question is the role of surgery for brain mets in the molecular era where all these options are available. So we started to work on that with a B-centric retrospective register, Cologne, Regensburg. We included some more than 800 patients, and we extended it to an octocentric project. You see the centers, it's in the DACH area, so Germany, Austria, Switzerland. The primary inclusion criterion was at least 1 resected metastasis, but also some more mets could be resected. We started as a retrospective database, but we redesigned it to include also prospective data. And we gave this a name, the role of neurosurgery for treatment of brain mets in the molecular era, results of the multicenter SUBAROMA study. This is a cohort from the 8 centers. Altogether, we included 3,203 patients. And after the correction for in- and exclusion criteria, there was a cohort of 2,751. And after excluding some patients with missing data, we still have a very good cohort. And we have all data about radiation therapy, systemic therapy, and extracranial tumor status in the numbers shown below. And we have a post-operative MRI in at least in close to 1,800 patients. So it's the worldwide largest data set up to now, and we can do something about it. So that's the baseline characteristics. The mean, it's very typical. Mean age 63 plus minus. We did it politically completely correct. 50.2% female, 49.8% male. So I'm happy. And 71.4% had active extracranial tumor, active primary tumor, or extracranial metastasis. And what is very typical for a neurosurgical database, almost all patients had symptoms. And 62% had one brain met only, got one brain met only resected, and 38% more than one. And in almost 60% of patients, the brain met was the first thing that was diagnosed before we knew the primary cancer. In almost 20%, it was at the same time. And in 22% of patients, the primary was known and the brain met detected afterwards. And this is also quite a typical distribution of primary cancers. The non-small cell lung cancer, 42%, followed by breast, melanoma, and all the gastrointestinal and some others. So all patients in this series have been operated. This was the inclusion criterion. And this is the number of resected mets, 92% one only. The irradiation was in 79.1%. Of these, more than 3/4 got local radiotherapy, and still 23% whole brain radiation, and systemic therapy in 58.3%, which is a relatively high number. And 61% were treated with immune or targeted therapy, and 39% got non-immune, non-target, so meaning they're more or less classical chemotherapy. And no adjuvant therapy at all, only 8.5%. These were the very poor ones. So the extent of resection in these 1,800 patients, the gross total resection was achieved in 73%, which is it's a more or less okay number. It's not from a prospective study. It's a everyday situation. Non-total and subtotal was 16 and 11%. But in the further evaluation, we put these two groups together. The only thing that counts was gross total or not. And this is the morbidity rate. There were complications, all kind of complications in more than 20%. That's very, very high. It's higher than compared to similar series published in the literature. If you look to the series advance, these are very low, but I'm a bit critical about the 20.4%. So this is honest numbers. And I'm seeing some tendency of our neurosurgical colleagues to be not very critical with their own numbers, which might explain the lower morbidity numbers in other series. This is a question to you. How was the clinical condition after surgery? Simple question. Are they doing better? Are they the same? Are they doing worse? What do you think? Maybe the answer is not too difficult if a neurosurgeon asks this question. They are better. This is measured at day 2 or 3, so just before dismission from the hospital. The light blue is Karnofsky 70 or above, and the dark blue is below 70. So 1,274 patients were stable, the same KPS. Some deteriorated, we have to admit, but more than 1,000 improved. And if you look at all the scores we took, they all became better. If you see the Karnofsky, so the Karnofsky 100 improved, the 90 improved, the 8 is a bit less, and all the poorer Karnofsky rates were the curves were shrinking. If you look at the neurological performance score, so 1 is good, so you also see a strong tendency to the better ones. And if you look at the very classical RPA classes, it's also improving after surgery, just only because of the KPS. And this is the statistics about its very high p-values, all parameters highly significantly increased after surgery. What about the risk of intracranial recurrence or new metastasis intracranial? So the meaning progression-free survival, more or less, intracranial progression-free survival. The mean was 8.9, median 5.7. That's okay. There's a long range, 0.17 to 74 months. And maybe it's important to see the difference between a real local metastasis. It was 19% at the edge of the resection cavity, and distant new metastasis 695, leptomeningeal disease in 145, and the local recurrence number of 19 is quite low. It's lower than in literature, but I believe, I suspect that in many series, these are taken together. If you have new mets, we have 37.7, and that's the typical number which is published for recurrences after surgery. And if you calculate the risk for new mets in 1 year, it's 44.4%, and 4 years, it's about 2/3. So what's the role of surgery, of gross total resection to prevent local intracranial recurrence? I absolutely do not like this curve, but that's how it is. If you compare the patients which got gross total resections versus these who did not get complete removal, there was so the blue line is slightly better, but it's not significant. It's 0.068. So we are all quite disappointed. So we have to need why. Why is this? So it's quite a busy slide, but if you look on the left side, it's about the patients who did not get irradiation after surgery. It's point C, and E is the patients without any adjuvant therapy. So in this patient cohorts, surgery did achieve a significant improvement of progression-free survival. But if you add postoperative therapy or if you add systemic therapy, this difference shrinks more or less completely away. So if you do postoperative irradiation with or without pharmacotherapy, the fact that we tend for gross-total resection is not really meaningful for the prevention of local recurrence of progression-free survival. Are there any other factors? This is a typical statistical evaluation. So any radiation versus no radiation is a strong factor. Immunotherapy or target therapy is a very strong prognostic factor. By the way, if you do any systemic therapy, including classical chemotherapy, there was no statistical difference in the multivariate analysis. So be critical with chemotherapy. Let's look to overall survival, maybe even more interesting parameter. So it was median and mean were 7.5 and 15 months with a very, very long range. Median follow-up was 8.5 months, which is okay. Loss to follow-up after half a year was 15.7%. I think it's acceptable. This number of patients died. They died at the end of the follow-up. And this is the cause of death. The majority of patients are not dying from the brain mets. They are dying from the systemic cancer. And this is at least one achievement that we made all together. We are shifting the problem away from the brain to the rest of the body. Yeah. So again, the question, is total resection meaningful? Do we as neurosurgeons do something beneficial for the patients or not? And this is a curve that I like. If you look how long live patients, how long live patients who got cross-total resections, they live significantly longer than patients who did not. And why is this? So cross-total resection is meaningful for overall survival. This is a curve you just have seen. Oh, that's very busy, yeah. And the more important curve might be this. This is a Karnofsky score after resection. It's a highly significant difference, and the patients who are good after surgery, these are the ones who live really long. So this is our job as neurosurgeons, to provide a very good extent of resection to achieve cross-total resection. That's our responsibility. We have to bring the patients in a very, very good condition. And this is not our hands. This is the presence or not of active extracranial tumors. It's also a highly significant difference. And that's the reason why 2/3 of patients die. So there are, of course, non-surgical factors for overall survival, which will explain why the Karnofsky is so important. This is a classical curve for radiation therapy. So the role of radiation is not discussed. It's absolutely clear. So the patients who got the radiation lived much, much longer. And this curve is a bit more interesting. The red line is the patients without any pharmacotherapy. The blue line is of the patients with classical chemotherapy. There is no difference to nothing. And the line, the green line is the immuno-target therapy, and this is a game changer. This is an absolute game changer, but the classical chemotherapy is not. So we have a very strong prognostic effect of radiation and immune or targeted therapies, but the patients must be good enough to receive these therapies. So there are also a lot of factors which we cannot influence, like age and primary tumor and so on. But the postoperative Karnofsky has a very strong impact in the multivariate analysis. Radiation has, and systemic therapy has. And the consequence, which represents the center of neurosurgery in many patients, even with multiple brain metastases, we need to resect the mass lesion. We improve the KPS. We enable them to get adjuvant therapy, and that's the main reason for improved survival. So I can come to the conclusions. So the single space occupying lesion might be a metastasis, might be maybe a radiation necrosis, whatever, should be resected. In patients with planned immunotherapy to avoid corticosteroids is a quite young indication. And in patients with single or multiple metastases to improve the KPS and enable them to adjuvant therapy. So if you take all together, all surgical means which we can offer for everybody, so the indication for a stereotactic biopsy would be a Cobb syndrome. You find a lesion intracranially, you have no idea where it comes from, you resect it, or if it's small, you just take a biopsy. You have the single space occupying metastasis. You have 2 or more space occupying lesions. We can resect more than one. We can resect 2 or 3. Yeah. If intent target therapy, you all know the molecular genetics, the expression profile of receptors and driving mutations might be completely different in brain mets from the brain tumor. If you want to target them, better get tissue to plan your therapy. If intend immunotherapy, resect mass lesions to avoid steroids. And if you have several metastases causing symptoms or a poor condition, resect them to improve KPS. And that's it from my side. Thank you very much.
Thank you very much, Professor, for your presentation. Do we have any question from the audience? So we have one here.
So it has been described that brain metastases have different growth patterns. Some are invading, even like glioma-like, and others are well circumscribed. Do you think that has any influence on the data you have presented here or on neurosurgical strategies overall?
It has a clear influence in our daily work, but it did not influence the series. Yeah, this is a bit simpler. But if you have a brain met, especially I believe it's more in the gastrointestinal ones, they have a more diffuse, they have no clear edge, they're more diffuse. And in the example for cerebellum, frontal, temporopolar, I resect more than the solid tumor. I do a supramaximal. It's very modern currently, also in the gliomas. I always do supramaximal research and resection, and most neurosurgeons started to do that. Yeah. But it has not been evaluated, neither retrospective nor prospective. It's a thing that should be done. Yeah.
Any other question? If I may just make a question to you from my neurosurgical perspective. At the beginning, you showed data from this octocentric study. And we also recently published a study showing that in our center, so more than 600 metastases, we also had 2.5% of permanent neurological deficit, as you had. Mm-hmm. And we also had around 20% of morbidity, which is not low, but is minor morbidity. Yeah. Now, this is not only in line with what you showed.
Minor or temporary.
Exactly, exactly. My question is, so I don't know where we are going as neurosurgery, but can we say that we are improving in our road from the beginning of neurosurgery up to now? Because if we compare to previous data, results are getting better and better. So can we say a give and take home message that surgeries become more and more safe?
Also, we neurosurgeons have some ability to learn, yeah, to learn to become better, to also to use the techniques to be safer with our approaches, to be better with the resection controls. Of course, we are learning. Yeah? And what I didn't expect it. Yeah, we have always asking the question: are these patients really better? Sometimes we have the impression, coming with a hemiparesis, going home almost healthy, but you are also seeing complications. Yeah, and this is from my standpoint, this was the most important question of the whole series: can we really make them better? We knew that the KPS is extremely important to make them qualify for all the pharmacotherapy. If the patients come to the KPS 50, what do you want? Yeah, you let him go. Yeah, but if it's really good, he qualifies, or she. Yeah.
Thank you. Any other question? Okay, so if this is not the case, I think we can proceed, and I give the floor to Professor Waller.
Thank you. I'm looking for my speaker. Johnny? Yeah. It's a bit embarrassing if you announce somebody who's not there. No, I know you're right. So we now come to a difficult topic, trials in neurosurgery around brain metastasis. We have Johnny de Rink here from Belgium for a very good reason. At the OTC Brain Tumor Group, we have been criticized for, I think, 2 decades, ever since we have been there, that we don't have any surgical aspects in the group. And at some meeting, I think Emilie, Matthias, and I were sitting together, hearing again the criticism. And I think Matthias then said, okay, we're going to create a committee. And that's what we did. And then we needed to find a victim. And I think we found a good one. Johnny has actually done quite a lot of trials, very interesting trials. One of the most interesting trials that some of you may have seen is about checkpoint inhibition administered locally in the brain already a few years ago. Highly interesting work that he has done here with his colleagues. And since he has taken over this committee position, I think we have at least developed a lot of concepts how to move forward. No, we're very happy to have you on board. Thanks for making the trip.
Thanks for the invitation and for the kind introduction, Michael. I do agree that we mainly have a lot of concepts at this point, but we're trying to change that. We'll discuss this next week. I was asked to discuss neurosurgical trials in metastasis, and it was a really thankful subject to research into, I think, and it was also a great talk by Roland just before me, which because I feel that even though we didn't communicate, it flows very nicely from what he just showed. Very impressed always with large numbers of patients, of real-world data, but there are always certain things that just observational trials cannot capture. So when I thought about this, I was wondering what are actually the questions that we need to address as a surgeon to ask ourselves, are we doing the right thing? Are we actually helping patients, etc.? So first of all, what is the current evidence for surgery? What are the gaps? Are there any gaps? Do we know everything about surgical treatment for metastasis? Should we operate better? That's also a relevant question. Should we study surgery tumor type specific, which is the question that Matthias just asked? What should be the optimal timing of surgery? Should we incorporate any neoadjuvant treatment, radiation therapy, or immune checkpoint inhibition? And also, what is the role of tissue? Because not only surgery is a treatment, but we can also provide surgery as a service to deliver tissue, which could also be part of trials, of course. So, as a first question to ask ourselves, is surgery effective for brain metastasis? It's a very relevant question that has been addressed some time ago. Some references that you see here that mainly demonstrated that if you do the combination, surgery plus radiation therapy, that is better than surgery alone or better than radiation therapy alone. So in essence, demonstrating in the 2 Spatial trials that surgery is effective for local control and for overall survival. So I put up this slide, which summarizes one of the Spatial trials, which was surgery versus surgery plus whole brain radiotherapy, but mainly to show what the problem can be with all the data that we have. because sometimes so this is a prospective study showing that if you add radiation therapy to surgery, you improve results. And so this is something that we've commonly grown to accept, whether it be whole brain or radiosurgery. But then when you look at retrospective studies, in certain instances, you get the opposite point, which of course is indicative of the selection bias of patients that you give whole brain radiotherapy as an addition to surgery. So we should always be careful with retrospective cohort studies, especially if they have low number of patients like here. So for a single metastasis, the evidence is that if you perform surgery, then you get better survival and you get better local control. In terms of multiple brain metastasis, there's not really very hard evidence. There was this trial from '93 where they compared groups where they resected all lesions, where they didn't resect all lesions, and then they had a matched cohort of patients that had a resection of a single metastasis. And they found that when you do a resection of one or all the lesions, then you get the same survival. But when you do not resect all lesions, then you get worse survival. But this and all the others mentioned here are all retrospective studies, so very prone to selection bias, of course. So I don't think we can draw very hard conclusions from this. If you choose not to resect all patients, it must mean that these metastases are in a bad location. And so you cannot really compare this to patients where all lesions are resected. But anyway, this data, which is only retrospective, suggests possible benefit when you have limited brain mets, when you have multiple symptomatic brain mets, and when you have possible access to the same craniotomy, it's a bit more accepted that you take out more brain metastases. So this, in line with what Roland just demonstrated, remain the classical indications for surgical therapy. And many of these things, like if you have a really large size, and especially if you have a hydrocephalus, if it's in the cerebellum, like in the picture here, it makes sense. And I'm not sure whether it always requires a randomized controlled trial to demonstrate that these are actually effective. It's a bit like the remark that's always made that there's never been an RCT testing for parachutes, whether they actually save lives or not. In some of these cases, you just know that you're helping the patient by relieving the pressure, and that if you don't, they will die shortly after. So I think we can state that if we perform surgery for patients with brain mets, that we can improve local control and we can improve survival. But then another thing is, how can it be optimized? And interestingly, I found a publication coming from the Cologne group, which is probably part of the larger series that you presented now, because at that point, so as you showed local recurrence, the difference between subtotal and total resection, there is no difference in terms of local recurrence. That you also demonstrated. But overall survival, there was no difference in the series coming only from Cologne either, which changed when you added more patients. So this was interesting to learn. But I just put this in as also a warning for like retrospective analysis and that you sometimes can get mixed signals. But there are other series. There is the PROPS-BM. It's also a registry, observational registry on preoperative radiation therapy, where they also looked into the patients that had subtotal and gross total resection and compare them. And they did see that you have local recurrence hazard ratio of 9.1, so a lot higher recurrence rate if you do subtotal versus gross total. So, and this also aligns with the data that you now showed. So I think we can also state that if you do complete resection, then it's probably better than not doing a complete resection. Now, it's of course very important to note, many of you here are not neurosurgeons, but never at one point during a surgery has a surgeon ever asked himself, should I leave half of this brain met in? Perhaps it will be better. So whenever you have a residual, a partial resection, it's unintentional. Especially for brain mets, this is the case. So rarely you would leave something in for safety purposes, but a brain met is typically easier to resect than a glioma, for example. The borders are more easily recognizable. So the intent would always be to go for a complete resection. So if you have a partial resection, most of the time it's the fact that you have a brain shift, for example, that you don't recognize the tissue well anymore, and it's unintentional. So an important question would be, how can we improve this? How can we improve the quality of the surgery? And then another thing, there are some publications on supramarginal resection, some centers that perform this. It has been demonstrated already in 2006, apparently by Brigitte Baumert, that you, much like glioma, that you find tumor cells within 1 centimeter of the cavity wall in up to 60% of cases. So this could be an argument pro supramarginal resection. And there have been some only retrospective studies about that, with data that you probably cannot always fully trust on, like like the one from the Milan group. So they had 69 patients, and they state in their publication that they had 100% local control over one to 2 years, it said in the publication, which is a bit strange. But anyway, if it's safely feasible, then it might be something worthwhile considering. So some general considerations in terms of surgical technique, as I just said. Typically, you would have like a capsule. This is dependent on the type of tumor that you're operating on, the type of primary. But in general, you would recognize margins of a metastasis better than margins of a glioma. Maximizing extent of resection improves the local control, and en bloc resection is preferable in order to avoid leptomeningeal dissemination. This is a general principle, and most also retrospective studies demonstrate that you get lower numbers if you do an en bloc resection, which might also be dependent on the tumor type, which I will come back to in a later slide. So in terms of advancing the extent of resection, how to improve the quality of surgery, it's image guidance, it's fluorescence guidance, and it might also be brain mapping. I'm not always sure whether brain mapping is really indicated in brain mets, and there's not many centers doing it, I think, but it is definitely a possibility. This slide has shifted a bit, but anyway, so the first thing, so this was about the intraoperative ultrasound. So I think personally that for me it's a very, very important aid. And I use a navigated ultrasound, which means that it communicates with the navigation, and it shows you the ultrasounds. It shows you the sound. Okay. It shows you the ultrasound slides overlaid on your MR, so you're not confused by the orientation. So this is very convenient. It also allows you to update your navigation, so you don't get the brain shift. And you do, especially for brain mets, you do recognize the edges very well. So this has been very helpful, and there's also a publication which should be under here somewhere. I can't pull it up. That shows that you do get more complete resections, but this was not a randomized study. Same thing for the use of fluorescein. I personally do not do it. I use ultrasound alone. But this is also supposed to give you a better visualization of what the exact edges of the tumor are, and allow you to get more complete resections. And then awake surgery, there's the groups mainly focused on awake mapping and surgery, like from France. They see mentioned here that attempt to do awake surgeries in many patients, and they say that it's definitely possible. You get high resection rates even within eloquent brain areas while preserving overall neurological condition of the patients. So it could be considered. And this is true, but I would say that it's only a tool and nothing more than that. So I wouldn't necessarily use it as a focus to do awake surgery in brain mets patients, and I think you can do it as safely asleep and still get a good extent of resection. So anyway, if we wonder what matters in brain met surgery, it's the extent of resection. So we need to avoid accidental remnants, optimize the technique, en bloc versus piecemeal, which is not always easy to do an en bloc resection, by the way. But the true extent to be targeted, whether it be supramarginal or complete, is still to be defined. And although there's only retrospective data, I think it makes sense to consider that the local control counts per metastasis. So if you have multiple metastases and you are able to resect all of them and then irradiate all of them, then that counts for local control. So it's more about the number of lesions that can be surgically treated. So when we're thinking about studies evaluating the extent of resection in brain mets, these are valuable. I also believe that they would be impactful, and they are also very simple in essence. Much like the 5-ALA trial, for example, where the primary endpoint was the amount of complete resections that you obtain. This is something that you can determine on a postoperative MRI already. So, of course, you would include, if you would devise such a trial, you would include overall survival and local control, etc. But to me, these are more, let's say, secondary endpoints, because the goal of this type of surgical trial would be to determine, are we able to achieve a complete resection more using a specific tool? So the fact that it's so simple, I think, makes it perfectly cut out for neurosurgeons to do this sort of thing. So, for example, ultrasound versus no ultrasound in brain met resection, piecemeal versus en bloc. It can be more selective. I will come back to that later. Fluorescein versus no fluorescein in brain met resection, and supramarginal versus standard resection, which could also be tailored. So these are all quite simple trials that would have immediate outcomes, and or we could quickly get results as long as the patients are there, of course. And then in terms of combination strategy, so I'm not going into the preoperative radiotherapy trials. I didn't put on the pre-ops too. These are the American trials alone. So, but this is something that's very important to study, and that's also concerns the surgeons, because in essence, you also want to know, can we compensate with some other treatment for partial resection, for example? Does it matter more or less when you do preoperative radiotherapy? Does it matter more or less when you have systemic therapy? If you do immune checkpoint inhibition, for example, do you do it neoadjuvantly or adjuvantly? So these are all open questions. And I think at least they should be controlled for the quality of surgery if the surgery itself is not the primary thing to be studied. And then a important question, I think, and this was one of your questions: are all brain mets comparable? No, of course not. And so we know from all the Sperduto publications and the development first of the disease, the diagnosis-specific GPA, that there's a huge difference between molecular subtype. This is for breast and for all the brain met primary tumor types. But the same is also important for surgery, and this was a publication that I assume that Matthias was alluding to, that Anna published some time ago, that there's different growth patterns for different types of metastasis, that melanoma metastasis and small cell lung cancer typically have infiltrative growth patterns, while breast cancer and renal cell carcinoma, for example, have a more pushing growth pattern. So these have a more clearer capsule and a more well-defined border compared to an infiltrative growth pattern. So it would make sense in these patients to have a specific type of surgical intervention proposed to them. For example, in the infiltrative subtypes, like the melanoma and also the non-small cell lung carcinoma, supramarginal resection might be a better approach. So this is something that you would evaluate in these patients. You could evaluate it in all, but it is very likely that a complete resection in tumor types with a capsule is like oncologically equivalent to supramarginal resection in an infiltrative subtype. So this could also be a question that you ask yourself, and should we differentiate our surgery based on the type of primary cancer that you have? And I think this is the case, but it remains to be proven, of course. So, and then in terms of outcomes, what are the optimal outcomes for a clinical trial studying surgery? So to answer this, we need to ask ourselves the question, what is a bad outcome? If we do local brain met treatment, because to be clear, I don't think anybody doubts that, but we are not curing the disease, not the radiation, well, not the radiation of the brain at least, not the neurosurgeons, but we are performing a local treatment and hopefully improving survival. But so if we do a local treatment, whether it be radiotherapy or it be surgery, so these three are actually bad outcomes, and these are failures of the local treatment. So I think that an endpoint of a trial should probably be a composite of these three things, which is already the case in the preoperative radiation therapy studies. But then, depending on what we want to evaluate, and this is something that I said before, complete resection, for example, so the number of complete resections can be a perfectly adequate surrogate endpoint, because I don't think it's actually a surrogate. It is the main thing to be studied, because we're not curing the disease anyway, and we seem to have established that if you have complete resection, then your local control and survival is better anyway. So the endpoint for such a studying technique would rather be complete resection than anything else. And then, as I said, there is another role for surgery as a provider of tissue. And since we are a provider of tissue, this can have many, well, or some consequences at least. So there is the phenomenon of the subtype switching, mainly for breast cancer, that has been described, which could have impacts on the treatment of the patients afterwards. But how this would fit in a surgical trial is not fully clear to me. It has been established that this exists in about 20%. So this is something that in the clinic we would be able to use. The same thing for differentiation with radiation necrosis. If you can have it serve as a treatment anyway, and then you have the diagnosis also that it's a radiation necrosis or not, or a progressing tumor. That's also a role of the surgery. And then, of course, if you do surgery anyway, you could also do it when you do neoadjuvant treatment, for example, in the context of a window of opportunity trial to document how the drugs penetrate the tissue. So, in answer to the question that was on the first slide, of course, it's the way to go to do trials in metastasis. We need to demonstrate and provide evidence of that what we're doing is actually useful and effective, and that we are constantly improving. And I think the things to be focused on are combination with other modalities. We are not working alone, definitely not in this context. So it is important to look at the combination strategies, optimal timing with radiation therapy, with systemic therapy, etc. The extent of resection needs to be improved. How to do it, and well, whether it matters, that then as a secondary endpoint, because that is something that is more established. So I think it requires the fact that there's differential effects of surgery in the different subtypes requires at least stratification. If you do a catch-all trial, so meaning you include all brain metastasis patients, but in some instances might deserve focused attention, like for melanoma brain mets only, if you do surgery there, or for breast cancer patients, for example. And then, of course, there's an important role of tissue using surgery as a service versus demonstrating that knowledge of the tissue actually makes a difference in the survival and the treatment of the patients. Thank you.
Thank you very much, Johnny. Any questions?
Thank you, Johnny, for the nice presentation. I was wondering whether it's really needed to do a trial piecemeal versus en bloc. Is the data there not ready? Is it a sort of parachute trial? We already know that en bloc resection is the best, isn't it?
Well, I think there is what I can say is that there's definitely still evidence needed to convince neurosurgeons that this is the way to go.
Okay.
And there's also another thing. I think it deserves good documentation of what it actually means in terms of results, because it's not always easy or even possible. And if a surgeon is always going to feel bad about doing a debulking, because the standard in any surgery that we do is that you do a central debulking, because if you leave the mass effect in and you go for the edges, then you're destroying more normal tissue. So that's why piecemeal resection is something that we're more used to doing and is safer for other structures. So if you would do a trial, you have more leptomeningeal spreads. This is probably true, but perhaps if you do this, you have more neurological complications as a result of surgery. And I think it's finding the balance between those two before we can definitely establish that en bloc resection is the standard.
And do you want to do it only in melanoma then? Because I saw on your slide that you would like to do it in melanoma.
Well, because it has more chance of leptomeningeal spread in melanoma than in the other tumors, but no, not necessarily. I think it's important in any trial to stratify, at least stratify per tumor subtype.
Thank you. Surgeons talk to surgeons.
Be happy about that. I'm also a bit skeptical with a piecemeal prospective trial, because usually it's not a question that the neurosurgeon decides. So the tumor defines. Yeah? Exactly. The deep-seated big mass lesion defines. If you can take it out as a whole, a deep-seated big tumor, it's not possible, or almost not. And it's easy in a superficial small tumor, and then you cannot really randomize. Or you find a very, very, very super small subset which needs to be randomized. So I'm skeptical about that. I agree to all the others.
So if Michael were to donate you a couple of million euros to do a trial, which one would you do? Well, if it's a couple of million, then it can be a cheap one, like no ultrasound versus ultrasound. But I think that ultrasound is really an extremely useful tool, and I have the feeling that it's still underused in surgery. So that's something that I think deserves establishment in an RCT. For me, I don't know if you use it in standard fashion. Of course, it's good.
Johnny, I want to ask you about leptomeningeal disease again. So there is a very huge variation regarding the risk of leptomeningeal spread after surgery, even in a randomized trial. When you look at the MD Anderson trial, where all the surgery were en bloc resection, we still have a 17%, 17, which is a high number, of leptomeningeal disease. When we look at Paul Brown trial, where comparing radiosurgeries versus whole brain radiation therapy after surgery, then it's around 7%. There are two randomized trials. And also when you look at retrospective study, they go from 0% to 25-30%. Any explanation, for this, or in your activity, in your experience?
You mean why the reported numbers fluctuate so much? Well, why it fluctuates so much between studies, I really have no explanation for that. But I can tell you that how a surgery takes place is not is very, very different between patients. And in some patients, you get a really clean resection, you take out the brain met, and then in other patients, you know that you have to go through cell because it's deep and you're spilling more. So, and I think in many of those trials, perhaps all of them, there's not enough control for how the surgery actually went. And I think a clue will be in how the surgery was actually performed, whether it was, for example, en bloc or not. And this is rarely mentioned. So something that you could control for, but I don't know if that's the explanation.
I think also a difference in the diagnosis of the disease. can change.
Of course.
Because especially after surgery, we have some very localized, despite the spread, which for me are not exactly the meningeal diseases. So probably also the variation depends on how we diagnose it.
On the definition of okay. So the definition has been established.
I just wanted to agree. I think a lot of series don't distinguish.
Yeah, next one.
Well, just a comment to Dr. Minetti. Some series don't distinguish between nodular leptomeningeal, which is what we think are the post-operative complications, and then maybe the diffuse sugar coating, which we think are natural progression, hematological for breast cancer, for example, and requires good definitions, and you only find it if you look for it. Yeah.
Yeah, thank you. So I think from now having kind of also being involved in this guideline writing, etc., last 2 years, I mean, and with all my also personal experience with how brain met patients are treated in Germany, in large centers, all over Germany, etc., it seems that surgical interventions, resections are far too really rarely given to patients, so they should be much more. I think it's important to convey the message that it's not glioma surgery, right? So you often can resect it very well, and then the patient is in excellent condition. After that, you resect very little normal tissue, etc. Access is easier. So I think it's not clear to many that after resection, the patient can benefit very, very much in terms of clinical outcome and conditions and benefit, etc. So they are the key. And then, of course, in addition, all the molecular information that are relevant for therapy planning, etc. So we try to honor that in the guideline to kind of clarify that the existing data, which is pretty good, and of course we need more and better data. That's clear. What is your common joint surgical view on that? How can you implement resections and neurosurgery better into, let's say, broad everyday care for brain metastasis patients? I mean, clearly you're writing guidelines, etc. But what is your idea on that, or would you disagree in countries outside Germany?
Okay, so sorry. So your point is that we are not operating on enough brain mets?
Yes.
Is that yeah? Okay. Yes. So I fully agree. So how can we improve on that? I think it's actually performing trials and performing serious trials, and not only retrospective series like we all do. These have their value, especially if you get to really high numbers and from multiple centers. But I think it's providing actual evidence, real evidence that you're not making the patient worse, and that you're even making them better, and that you're so that you're doing it safely and improving their outcomes. I think that's just it. And I think also, because in response to your question, Giuseppe, perhaps, when I think of how it works and this is probably the case in most departments, because brain mets are a bit easier to do, more localized, easily recognizable. This is a bit more often delegated to residents. And so I would be quicker to delegate a brain met to a resident than a glioma, which is more difficult. So this in the real world data will probably also play a role, and it's actually looking into the literature and seeing how it evolves over time that made me take it more seriously as a not wasn't taking it seriously, but you were quick to delegate it, and then you learn about, okay, en bloc versus piecemeal, there is some finesse to it, and I think that culture needs to change. And with that change of culture in the neurosurgeons, I think and the fact that neurologists support our cause, I think that's already a good start.
Maybe a question for Joanna, because you're here. So can you comment on that? Because you're a melanoma girl, so...
Sorry. So I was wondering if I need to comment or not. So I think, speaking of melanoma, because actually I'm a dermato-oncologist, so we treat actually melanoma here. We do have actually very effective drugs the last years, and we have the combination immunotherapy with ipilimumab, nivolumab, which in patients with brain metastasis, small brain metastasis being asymptomatic, we have like 50% response rates. So I don't really agree that we need to perform more surgery to all patients. We need to find in which patients we need probably to perform surgery, because, and I think that we need like trials, which is the best sequence, for example, like local therapies at once, or during the immunotherapy. There are some trials ongoing. So for melanoma especially, we do have actually very effective drugs, and we have the combination immunotherapy, which is in 50% of the patients. We really have also 20% complete responders. It's not all patients, so I don't think that we need to really perform surgery to all, but I think we need to define, or we need to find which are the patients probably that they will need it, and what is the ideal time point.
Yeah, I agree with that, and I think my experience in brain mets is also a bit skewed towards melanoma brain mets, because we have a melanoma expertise center, and so I'm not always involved in the first decision on how to treat the patient with the brain mets, and they do get a lot of checkpoint inhibition. But I have been operating on more hemorrhages for patients already under ipilimumab, nivolumab. So there's this all of a sudden creates a whole problem. And I think this is just indicative of the fact that we need to differentiate between different tumor subtypes, because they're completely different diseases.
To come back also to the point of Frank, I think you know what you organized at your, we have this MBM at the tumor board.
We actually have a presentation.
Okay, so she will reveal some secret later, how to have a good collaboration. Maybe I think it's always when we discuss, I think if for a medical oncologist, if it's difficult to reach out to a neurosurgeon, of course you will have no surgery. If there is a good collaboration, good contact, they know they can call you. That's always easier to.
True.
And
true.
Maybe I can also. And what I find also very important is also, like in melanoma, we have a lot of neoadjuvant trials right now. We have actually in the early disease stage, but there are also some studies actually with metastatic disease, and probably there are also actually these neoadjuvant trials in other entities. And I think this neoadjuvant trials is also a very well panel for new drug development, because you treat and you resect, and then you have the tumor. And this is actually a very nice idea of performing actually analysis, and you have very quick results on new novel drugs, how actually it changed. You have the responses and how it changed actually the tumor microenvironment. So I think also this would be an ideal approach, like new adjuvant trials in patients with brain metastasis, in order probably to test new combinations or new drugs.
It doesn't matter. We're done anyway. Thank you, Johnny. So we continue with the controversy. Carl will take over again. The controversy I like because it makes radiation oncologists allergic, and sometimes it's good to wake them up.
And from time to time also surgeon, I must say. So let's introduce this new topic about new therapeutic option that is emerging. So it's you're not Richard Grossman again. Exactly. Sorry. It's a pleasure to introduce Professor Grossman from IFA, Rhun Institute, starting the debate about the use of laser interstitial thermal therapy for neurosurgical lesions. And we will have a format with 5 minutes discussing the advantages, the pro, and the 5 minutes of about disadvantages. Please, Professor.
Thank you very much for the kind invitation, and good afternoon. I hope I will make it on time. So I will talk in the next few minutes about laser thermal therapy in the context of brain metastases. Actually, I think about lead as a complementary tool and not instead of radiation therapy and not instead of surgery. So since there is since a patient with cancer live longer, we see more patient with recurrent intracranial disease. And a very common scenario is to see a patient with a post-SRS progression, means that the patient receive stereotactic radiosurgery and then they develop a new enhancement. And actually now we cannot distinguish between active recurrent tumor and radiation necrosis. So usually this lesion are located deeply, sometimes in a very functional region in the brain. And these patients are heavily been treated and they're not good surgical candidate. And the idea to give them another session of radiation is not ideal because the previous one was not so effective. And if you give them another radiation, you expose them to the toxicity of radiation. So it's not a good idea. And this is the point where the lead comes in. And actually, it fill an unmet need in the treatment paradigm of brain metastasis. And I think it's something that we should consider in our mind. So what is lead? It's a thermal ablation. We insert a laser probe in the OR, and we do it stereotactically through a very small, tiny hole in the skull, and then we take the patient to the MRI suite, and under a real-time MRI thermometry, we do the precise ablation of the lesion. It's important to know that in addition to the ablation, we can take a biopsy. So it's not just a therapeutic tool, it's also a diagnostic tool. I will not get into the mechanism of lead, but we need to remember that it's actually control and predictable damage to the tissue. And you need to be familiar with several zones of ablation. There is the inner zone with the higher temperature that immediately cause the coagulative necrosis. And then there is the peripheral zone, which lead to sublethal thermal time-dependent injury with a thrombosis of the small blood vessel, the aberrant blood vessel of the tumor. And also it leads to permeability or modulation of the blood-brain barrier. And then there is a very steep thermal fall-off that left the normal brain around the ablation area protected. So it's not just burning or a kind of a cyto reduction of the tissue. It do probably much more than this. It probably goes to modification of the tumor microenvironment, and from our point of view, it has a lot of potential. So the evidence of lead in the context of brain metastasis is usually talking about the post-SRS progression. And there is several of case series, multi-center. None of them is level 1 evidence, but all of them talking about the idea of steroid reduction, of improved symptoms and quality of life. And also there is a kind of comparison between the local control that we gain by craniotomy to this that we can achieve by lead in case of a very highly selected patient of small lesions that actually can be a good candidate for lead. Also, another issue that we need to be familiar with this is that there is a correlation between the extent of ablation and the outcome. We need to cover by the ablation the whole volume of the lesion, and this will correlate with the outcome of the patient. And of course, we need to be familiar with the limitation of this technique. If we have a huge mass effect, probably lead is not the right treatment. So lead actually fit into the real life workflow of oncological patient of nowadays. It's minimally invasive. Patient go home after 1 or 2 days. Steroid can rapidly tapering down, and the patient has only minimal interruption in the systemic therapy. And in case they were on immunotherapy, after they stop the steroid, they can return to the previous treatment. And because of the modulation in the blood-brain barrier, we can think about potential of a synergistic effect with systemic therapy. And also, we can repeat this procedure more than one time. So we need to know when LIT is not the right procedure. It's not ideal for large lesion. It's not for decompression, usually not for the first-line treatment, but it gives us a very good precision salvage option treatment. So it not a replacement of surgery, and not going to replace stereotactic radiosurgery, but it gives us another treatment option that actually cover a very critical gap in the treatment flow of the neuro-oncology patient. And it's especially important and have a lot of value in the case of a post-radiation progression in a deeply seated small lesion in a functional region, and in a patient who are not a great surgical candidate. So if we aim for a personalized neuro-oncology treatment paradigm, probably LIT deserve a clearly defined rule in this treatment plan. So I will stop here. Thank you.
Thank you very much. Perfectly timed. Thanks so much. And now I will go directly to the colleague, Dr. Kazda from Rhun, exposing us the contra of LIT. And then we can reserve discussion for after your presentation.
Good afternoon. My name is Tomasz Kazda. I'm working as a radiation oncologist in Brno, Czech Republic, and thank you for invitation. I have no conflict of interest, but I must declare that I had no patient with LIT ever. This is not available in my hospital, in my city, and in my country. So it was quite challenging to accept the invitation. But as always, I take this as an opportunity. American Nobelist Richard Feynman, he said that in the case you would learn something, you should have a talk about it. So thank you. Anyway, but what is your experience? So may you please raise your hands who has available LIT in your hospital? 1, 2, 3, 4, I guess like 15%. And how many of you has available intraoperative MRI in your hospital? Yeah, around 10%, maybe less. Good. We already heard lots of pro attitudes. And I must say that actually I'm personally on the same boat as radiation oncology. Of course, I'm afraid of radiation necrosis, and I would love to have the other options for our patients. So there's quite lots of people being in pro attitude, including me. So who is in the contra position? Hospital directorate, Ministry of Health, healthcare payers, insurance companies. So in this talk, I will speak like more from their perspective. At the beginning, with the negotiation with them, they will ask, what the hell is that? So LIT is like quite, you can see, simple procedure. It's a laser navigated by stereotactic placement to lesion for both primary metastatic lesions, most commonly for radiation necrosis or recurrent tumor. But they will, of course, notice that in the shadow, there is MRI. So you need intraoperative MRI. So it is not quite easy when they will see something like that. That at the beginning you must have co-registration, stereotactic placement of the catheter, some electrophysiology testing, laser is placed inside. You must go to the intraoperative MRI for thermometria, and then finally laser is on and treatment is done. So it's quite complicated. Now they understand what is it and are continue asking what is the evidence. We heard about the evidence, so I can only highlight the last one, which was the systematic review from 2025, showing a summary of 17 studies, many hundreds of patients, and quite nice overall survival of 14 months with reasonable local control. And these are patients who have a recurrence after stereotactic radiotherapy. So quite reasonable evidence. This is about overall survival, local control. How about patient-reported outcomes like quality of life? The other study, and now see, I'm in a pro attitude, right? This study was a multicentric prospective registry showing that among these 90 patients undergoing lid for radionecrosis, during less than 2 weeks, 13 days, patients were without steroids. Very good for subsequent immunotherapy. So now again, they understand what is it and what is the evidence. But what is their opinion? What they will say? A limited level of evidence. No large randomized phase 3 clinical trials, mostly retrospective, heterogeneous indications. So in the end, they will conclude high-cost technology without high-level evidence. Next. Unclear added clinical value. Well, yeah, for small lesion, SRS is standard of care. For radionecrosis, there are bevacizumab here in Switzerland. We have Boswellia serrata. And actually, there was one systematic review comparing bevacizumab with LIT, and bevacizumab was slightly better. So they will conclude narrow indication niche. What else? High capital investment, probably the most important opinion from them. You must have intraoperative MRI device, and it's not only about machine, but also about dedicated trained team. High upfront cost for limited patient volume. This low patient volume is especially relevant in small countries like my Czech Republic with only 10 million people. So probably only 1 to 2 hospitals will be reasonable for LIT treatment. In the end, this weakens cost-effectiveness argument in national level. Reimbursement complexity would be a last argument against from healthcare payers, because payers prefer some clear coding. In many countries, there is no official performance code to be able to be paid. They need controlled indication criteria. So they will argument, you even don't know whether it's radionecrosis or tumor progression. So it was quite a lot of their contra opinions, but it is not as bad. In this report from 2024, it is stated that currently over 40 neurosurgical centers across Europe are offering this treatment. So it is not so bad. It's from 2018 when the system received certification for Europe. As I said at the beginning, no treatment is in my country, so of course starting to think how to make it in Czechia as well. How let's initiate the transition. I need a strong neurosurgeon, a neuro-oncologist with passion and motivation. So these are believers, and then we can discuss the place where is intraoperative MRI. It is only in Prague. I'm going to the end just only to show you that the neurosurgery department in Czechia is quite rich, including the rest of Europe, as you can acknowledge in this map showing the neurosurgeons density. So I'm sure that also in my, as well as in your countries where it is not available, we will be able to find motivated neurosurgeon. And then paperwork will start, proposal submission to Ministry of Health. highlighting clinical and economic justification. It will be very difficult. Expert review from Ministry of Health will hopefully agree. We will need negotiation with payers, and it's more than 1 year work. And then finally, we believe that the official inclusion in a national list of medical procedures like this will be done. But it is not done. Then you need to find some million of euros to reconstruction of your neurosurgery departments to buy MRI, all equipment, MRI compatible, and you must have human resources. So their conclusion would be like, for brain metastasis lacks high level of evidence. The technology is costly, requires MRI compatible infrastructure. Its clinical added value appears limited to a narrow niche of selected cases. And with that, I thank for your attention.
So thank you very much for your presentation. Do we have any question from the audience about this hot topic? So if this is not the game, I start. We have Professor Volkmar, please.
I just would like to share a small comment. In former days, I had some personal experience with LIT. We had the system in Cologne from 2005 until 2010, about in these years. In these years, we did less than 10 cases. So in every time, it was like re-establishing a very new method. So it was a lot of effort to do that. And the thing why we left it was we have the alternatives in most cases. It was very hard to image the effect. Yeah? We did not know what we see in the MRI after the LIT, and we did not really see an effect for the patient. So maybe this was not evaluated in a scientific way, but we were just a bit disappointed in the end. Yeah, might be it was too less professional from our side. Maybe the indications were not the correct ones. It was all recurring glioblastoma, so it's not a very sexy disease. Yeah, but that's the reason why we were very enthusiastic in the beginning, but then we left. Yeah. Maybe we did something wrong. I just want to share this, yeah.
But maybe just to comment on the recurrent glioblastoma, I think you have a completely different disease there with lots of non-contrast enhancing tumor tissue that you probably did not treat. So my guess would be that it's completely different whether you're treating glioblastoma or brain metastasis, right?
Want to comment maybe on these points?
So first of all, I must agree with your opinion and with what you said. I think that everything is true. But I think that in selected cases, we need it. And for these cases, I don't think we need LIT in every hospital. But I think we need to have this center that we can send a certain patient that have this very deep radiation necrosis, so I don't know, maybe it's a combination of tumor and radiation necrosis, and we don't want to operate on this patient. And I think in these cases, we want to have the LIT in the toolbox. So I agree, it's a very long procedure, and if you can operate on this patient, probably it's much faster. And the recovery after surgery is good as well. But still, in selected cases, you could see example of a tumor located in the thalamus, because you can get there very easily and safely, and you can make a change, and the patient can stop taking steroids and continue with systemic treatment. So it really can make a change, I think, in a very highly selected patient.
Yeah, I can only agree that and add that there is also non-oncology patients. So all of these people who are pro are also those who are not working in oncology. There are also epilepsy patients or hamartomas and the others. So I can only agree, and it was also in my presentation, that probably one or two selected center offering LIT is much better than offering it in many hospitals.
Expanding indication would be a tool to convince the Contra people. And how big should be a country, your recruitment area, to make it palatable for, because also Israel is not a huge country. So what would you say to catch up to that point?
Yeah, it's difficult, I must say. I mean, like that, having 10 million of people, maybe there will be not enough indications to have at least one center. The cross-border collaboration is always difficult in terms of reimbursement. And again, I'm speaking like that, healthcare payer, etc. But I feel like that when we maybe also compare LIT with stereotactic radiosurgery, maybe the advance of the technology to daily routine simply takes more time, more evidence. We can say that Alexa Gamma Knife is here for almost 60 decades, maybe more, but only recently stereotactic radiotherapy is available and standard of care in, let's say, all hospitals. For LIT, maybe we are decades behind in terms of accumulating body of evidence and enlarging the network of centers.
Absolutely.
Yeah, we know that many recurrent gliostomas and recurrent brain metastases are in fact radiogenic changes that we treat, right? And so we are doing also LIT with glioblastoma quite a lot in my hospital, not we, but the neurosurgeons and colleagues. And I must say the experiences are very good. So I'm impressed, I must say. That different advanced technology, I don't know. Of course, the beauty is that you also get the tissue, right, doing LIT. And then you get solid tumor mass, or you get necrosis, right? And then you are kind of, and LIT is potentially good for both, right? So it's a kind of a nice dual activity. So you apply the technology, and even if the pathologist says a few days later, oh, this is all radionecrosis, you have not done something wrong. If you would have applied re-irradiation or radiosurgery, this you would probably have made things much worse, right? And have created a real problem for the patient. So I think this speaks very much for LIT. We have no experiences with brain metastasis. I think they're starting now. So I'm really looking forward to that. Because, I mean, in recurrent brain mets, we often also have this situation when they got radiosurgery before, right? Is it recurrence? Is it a mixture? Is it radionecrosis, right? And so a dual activity therapy, I think, makes sense.
Could I just ask you about the? Thank you. Because we've got some like 30 years experience of extracranial hypothermia in combination with radiotherapy and chemotherapy for recurrent tumours, but we're only just now developing planning systems analogous to radiotherapy treatment planning systems to really target the heat. How is the procedure with LIT? You insert the catheter for a certain amount of time till you reach your target temperature? Because I know you said there's a sharp dose fall off, but I wasn't sure about that in the brain. Certainly when you see RFA in the liver, it becomes very diffuse. But you just heat until your target temperature and then you stop? Also, yeah.
So you do it under the MRI thermometry, and you have this kind of nice colors. And you want to see a nice coverage, and you can sometimes, if you're near a very eloquent region, you can decrease the energy, but make it longer, for example. So it's a kind of fine-tuning between the amount of energy and the time you spend there. And sometimes you need to insert another catheter because there is a limitation. It can cover up to maximum 2 centimeters in the diameter. So if it's larger and it's not all the dimensions are equal, so you need to do kind of to build it, to build the shape. I think that another thing is the idea that to operate on radiation necrosis, it's not always clear what is the target. I mean, you see it very clearly on the MRI, but when you get in, you don't see a really clear tumor like you see in a primary brain met. You see like something which is not clear. Usually undergo fluorescence with the 5-ALA, but what is the target? What do you need to resect? I think that it gives it another maybe kind of advantage because I think it can it gives a good resolution of the mass effect and edema associated with radiation necrosis and all of these associated symptoms and steroid consumption. All of this.
Okay, thank you.
Maybe one last question.
Sorry. Well, that's also for you. So because it's super expensive, so there is money there. Why don't we have a randomized trial?
I would like to add that that was my exact question. How would funding a trial should be done before it becomes common practice, and how would funding for such an expensive device and machine, etc., take place? Because now there's some hints that it does work, no real evidence, and I know that also in Belgium centers have it and they are using it, and this is something that benefits the company, because imagine that you do a trial and it's negative and they lose. So how would the ideal funding scheme for a good trial proving that lit is indeed effective in whatever combination, what would that look like?
Yeah, definitely to have some strong prospective clinical trial would be very important for their perspective as well. One of the possible limitations may be in ensuring the homogeneous cohort of patients. Now I speak about brain metastasis, and when we imagine how range is now portfolio of systemic treatment for patients with brain mets, with melanoma, lung cancer, breast cancer, and how narrow is also the indications for like small lesions, maybe lesions after stereotactic radiotherapy will progress. So patients must be altogether in better general condition, like to survive long enough having brain metastasis. So first in my mind as a limitation and what would be needed to be addressed correctly in future clinical trial will be the patient selection. Maybe different story in primary brain tumors.
I'm not sure it's possible. Sorry, because I think it's a different population. I think those who are good candidates for LIT, you don't want to operate them. They are, deeply located. They are tiny.
These patients are treated with radiosurgery, for example. So I think radiosurgery versus radiosurgery, something.
Or maybe bevacizumab, but yeah, but it should be multi-center with many centers, of course, extremely expensive, and you will not get the clear homogeneous population that you probably have in your mind.
The alternative is continuing.
Yeah, to guess.
Perspective to sell an expensive machine.
Right. I agree. I mean, we all do the extrapolation of what we think. Yeah.
That was just also that my comparison with Lex gamma knife. Yeah.
I think the answer is very easy. The company has to pay it, right? That's what you ask from drug companies as well. I don't think there's any controversy. You should stop using it without a trial. Good. With this peaceful comment, we go to the break.
A panel discussion afterwards.
So many times to ask to see the studies for, so you can think about that. It's always the challenge we have in the clinics somehow is to treat the patients
So now it should be better. So the question patients ask, will this treatment work also in my brain if I have brain metastasis? And we have an increasing number of brain mets, of course, but we have some lack of knowledge, especially in the development of new drugs, and we have many new drugs for metastatic patients. So this is data from Germany because I just didn't want to point another country. It's for breast cancer, which is my focus, of course. And the prognosis is bad. That's one message. And the other is HER2-positive patients do better, but HER2-negative patients, hormone receptor positive and triple negative, do worse. So that's one of the points of this, especially of this drug, I think it seems to work also in HER2-negative patients and also in triple-negative patients. So that's a big improvement. I think one of the important points I took away when I saw the data. We had data from drugs for efficacy. I'm going to show you some of these also working, but these trials, small cohorts, were done after these drugs were approved. So it took quite a while from just having a new drug in breast cancer treatment, also in cancer treatment at all, to the availability of data for the efficacy of the brain. And that's a big lack of development. And having these many new drugs, it has become a more severe problem. And of course, patients with brain mets were excluded, with LMD anyway, they're excluded from most phase 2 or 3 trials. And that has to be changed. And there's some changing things, but I think tuxedo is an important point to improve further. So this is capecitabine, a well-known drug in breast cancer treatment, other cancers also. And these are small cohorts. These are all important trials for the efficacy of this drug in breast cancer patients. Still, again, after the drug was approved based on phase 3 trials where brain met patients or even LMD patients were excluded. So this is a big lag and time until patients were really able to be treated with these drugs. And then there was this trial, HER2-CLIMB. I think that was a pivotal trial because this was for HER2-positive pretreated metastatic breast cancer, and it allowed the inclusion of patients with brain mets and active brain mets. Not LMD, that was excluded. So this was really important because the drug was approved based on this, also having data for patients with brain mets. And another drug that was already mentioned is trastuzumab deruxtecan, also approved first for HER2-positive, now also for HER2-low expressing breast cancers. And it took quite a while because active brain mets were excluded, and this trial was necessary, it was not randomized, to really explore the efficacy of this drug in HER2-positive patients with brain mets. So this was not well done. I would say the development of this drug is effective. Now we have data, but it took a few years, and I think that was necessary. So what we needed is the efficacy of drugs that are newly in development early on for brain mets and also, of course, for LMD, which is an increasing problem. I don't have to tell you. So this is the trial design, and this is again, the 3 publications with 60 patients, also congratulations for that, which is well deserved, I think. It's for non-small cell lung cancer, it's for breast cancer, and most important, I would say, but that's my point of view, it's for LMD. And if I had to say something bad, but that's always the discussion, I would say I would not see, but it's not too many centers, not too many patients. And one thing I think we should all work on is that this drug is developed further. I'm not so sure in breast cancer what the strategy is, but maybe somebody of you knows a little bit more about that. I would say even for this data, I would deserve further development also in breast cancer. So there's more. You've just heard mentioning this. There's, of course, if it's tuxedo 3, it's also 4 and 1 and 2. And there's also another trial, I think, which is similar and which is also a very big achievement. That's DEBRA. And that's an approach using TDXD, trastuzumab deruxtecan, in HER2 overexpressing and HER2 low breast cancer, and also in patients with LMD. And I think this is also a very good example how we should develop new drugs. So my conclusion, of course, congratulations. That's really good platform. But I think we need more examples of that, even a platform maybe where the design is similar and then the drug is exchanged early on in the development of new drugs, not only for breast cancer, obviously. So, I think it's great, and it's a pleasure to discuss this. Not so many open questions from my side, except, of course, what is tuxedo? Thanks.
Thanks. So would now have first a discussion on this trial, and then we move on to the next one. So please stay here. Now discussion, one trial after the other. To focus.
Tuxedo stands for, the T is for trastuzumab, the X comes from deruxtecan, and the rest is because
that was the first trial. You just stayed with that.
Yes, then we stayed for it with that.
Because cool things have cool names.
Yeah.
So Frank, please.
So looking at the data, it seems that the control in terms of neurological sterilization improvement was really good during the time. So one question is this, let's say, an area where we should need to make any further progress? We know that intrathecal administration of trastuzumab, for example, this can be very, very effective in patients. Very long half-lives, very great CNS penetration, etc. Including breast cancer patients. So, is there any way or discussion or toxicity trial to intrathecally inject these antibody drug conjugates? And do you think this would be necessary because you're losing CNS at some point? Yes,
Well, first of all, I think ADCs work so well given intravenously that I don't see the need really, probably. I heard discussions about it. I think I personally wouldn't be brave enough to do it. So the risk-benefit rate is for me not convincing enough. But there have been discussions about that, right?
Yeah. And you're not at some point using this good CNS control in the patients so that you think about it, or to have an ongoing good CNS.
Patients progress at some point in time. So this is by no means a curative treatment. And
progression was mainly LMD, I assume.
In the LMD cohort? Yeah, LMD, progression of LMD, yeah.
We have a question up here.
Oh, hi there. Yeah, I have a question about the radiological assessments. So across all three cohorts, I think it was RANO-BM that was used. But I just wanted to find out more about cohort 3 in terms of the radiological assessment of LMD, because it's quite challenging, isn't it?
It is, and that's why we chose not the response rate as a primary endpoint, but the OS rate, overall survival rate, at 3 months, exactly for that reason.
Great, thanks. There's a question up there.
Hi. Was there a correlation between systemic response and CNS response?
The intracranial responses were a little bit higher, interestingly enough, than the extracranial responses. There were mixed responses where in both directions, where we saw intracranial response, but progression extracranially, or the other way around. So there was not a perfect fit, but there was a correlation. So most of the times patients responded in both compartments.
More questions? You keep me moving today. Thanks for that. That's the workout.
Thank you. I assume that the HER3 receptor was measured on a systemic place, on a metastatic, but we have, of course, the heterogeneity. So what does this mean in the future? Do we need more often a biopsy from the brain metastasis?
Yeah, that's a very important point. So we enrolled patients irrespective of the HER3 status. It was not an inclusion criterion. That was based on prior data from other trials where it was shown that HER3 expression showed a poor correlation with response to the treatment. What we did is we collected tissue samples, at least those that we could get our hands on, and retrospectively in a post-hoc analysis looked at HER3 expression. It's actually one slide in this extended data set. And in some cases, we had metastases, extracranial metastases, in other primary tumors, in a few patients, intracranial tumors. So it was a very heterogeneous cohort and a small cohort. And there we saw no statistically significant correlation between HER3 expression and response to treatment. We did see a trend towards higher HER3 expression among responders, but limited patient sample, retrospective, post-hoc, so shaky data.
But that's in line with other ADCs. We have so many new drugs, and that's the big question now. How to sequence them? We have no predictive factors. Many things don't work. We don't understand why.
More questions? Maybe allow me to jump in on that question. So there is no good correlation of the receptor and the response to the ADC. Why would you think is that? Or is there a minimum receptor expression that you would think is necessary? And how is that determined?
Yeah, I think that we can only speculate about this. I mean, and also the biopsy is one point in time. So who knows what happens across time? So either liquid biopsies could be informative. PET imaging for certain targets could be informative. I guess that needs to be explored in the future.
And I think we have something on that tomorrow. So it's a little teaser again. Are there more questions? Otherwise, since we're a little bit advanced in time, I thank you both very much. And I ask Emilie to come next and talk about the ITIO trial. ITIO. ITIO. Yeah. ITIO. Oh, no, not RT, IT. ITIO.
Yeah. Thank you. Thank you, Anna. So just to just to report on this trial, this is not yet published. The publication is in preparation. So that's just really preliminary results that I will show. That's intrathecal administration, in fact, of nivolumab and ipilimumab in combination with systemic nivolumab ipilimumab in patients with non-small cell lung cancer. I always forget melanoma, of course, and newly diagnosed leptomeningeal metastasis. And so that's a phase 1 that we did here. So why we did that? Because first, optimal treatment for patients with LMD is still controversial. So we try to have some consensus in the guidelines. And in the guidelines, in fact, we agree to recommend the use of intrathecal therapy in patients with circulating tumor cells in the CSF or in patients with linear leptomeningeal disease. And so, this is mainly based on randomized trials that we did in patients with breast cancer and newly diagnosed leptomeningeal metastases. In this trial, it was an easy design. We had randomization 1-to-1. Patients with breast cancer LMD, they were randomized between systemic treatment alone or systemic treatment plus intrathecal therapy. And here we could show an increased LMPFS when we use a combination of both systemic and intrathecal therapy. So this is a randomized trial, and the primary endpoint was met. But we still have no data for no randomized trial, notably for non-small cell lung cancer or melanoma. So that's also something that we would like to look at. So we know that we want to use the intrathecal route. It's mainly to increase the dose of the drugs that we want to administer to the patient close to the target, so the tumor cells in the CSF or in the leptomeninges. And so, because we are not really sure, we have only very limited data most of the time. When we administer a drug intravenously, what is the quantity that really goes to the CSF is a bit unknown. And so we had this work that was published also from the team of Dita and Prim, so Amsterdam, and they looked at the CSF serum ratio of nivolumab used. It's 5 patients only. That's still the data that we have, and it's almost nothing that goes to the CSF. So that's why it could explain why we have this leptomeningeal metastasis there. Of course, we are interested in nivo-EP because that's part, or at least when we started the trial, it was like standard of care, and it's interesting for both for patients with non-small cell lung cancer and melanoma, and that's why we simply use both tumor types in this phase 1, where we look mainly at the safety of administration. So there is this publication that you probably have seen from the team of MD Anderson, also on concurrent intrathecal and intravenous injection of nivolumab in leptomeningeal metastasis. And here they could define recommended phase 2 dose for intrathecal nivolumab, which is 50 milligram of nivolumab. So this is our design, and so I will try. So here we have the intrathecal therapy, and then we developed the trial, as you can see, in collaboration with the dermatology team. So because in fact we just put, in addition to the IV treatment, the intrathecal administration. So, and that's the standard regimen for melanoma patient. And that was also this standard regimen for non-small cell lung cancer at that time, at least. And here, so we had the first intrathecal injection alone, and then start also IV in addition to intrathecal to look at the safety of intrathecal therapy. This is what we wanted to look. When it's something new, you have to isolate this, meaning that if we are interested in the trial, if we get more data, probably for next patient, if it's safe, we can skip this isolated first dose and move directly to a combo. And here, so we took the dose of nivolumab that was shown by the team of MD Anderson, so 50 milligram of intrathecal nivolumab, and then we decided to have 3 dose level of intrathecal ipilimumab, 5, 10, and 20 milligram. So eligibility, that's patient with confirmed, of course, non-small cell lung cancer or melanoma. Indication for systemic immunotherapy with nivolumab and ipilimumab. Newly diagnosed, confirmed, or probable, meaning that all the patients had to have CSF analysis with cytology, craniospinal MRI, and of course clinical assessment. Karnofsky, we took for the LMD trials, often we allow a bit lower, so between 60 and 100. That's immunotherapy, so we didn't want to have too high level of steroids. No rapidly progressive systemic disease. We could have some asymptomatic brain metastasis if there were small or treated by SRS. We didn't want to have patients, in fact, that will receive a whole brain radiotherapy, and the prior craniospinal therapy was not allowed. So that's results. So as it has shown here, that's feasible to do trial in LMD patients. So in blue, this is what was planned. So approximately 1 patient per month. And in green, this is what how the enrollment went. So we had these 19 patients enrolled between 22 and 24, 6 females, mainly melanoma patients. Median KPS was 80 at the entry, and 12 patients had the positive CSF, so that is type 1, and the other CSF was atypical or negative. And so we could complete dose finding without dose-limiting toxicity, so which was defined during this 3 weeks period initially. So that's something that is, yeah, so we could go and define the recommended phase 2 dose at 20 milligram of intrathecal ipilimumab intrathecal in addition to 50 milligram of intrathecal nivolumab. So if you look at the median overall survival of the cohorts, that's 3 months. So melanoma is 3 months and a bit longer for non-small cell lung cancer, which is something that it's a range of what we know. So what I found is interesting in this trial, I can show you, it was not the case in the trial of Isabella Glitza. She had not observed any responses. She had long stabilization, but here in our trial, we could observe, like for this patient, you see, for example, clinical stabilization, even better improvement. CSF was positive at baseline, so that's really confirmed diagnosis. And you can see the cranial nerves initially, and we have here a prolonged MRI response. So that's something that we don't see every day for LMD patients, so we were quite happy. So yeah, so I think that's something, yeah, we need to consider. So now it's what is the best approach for intrathecal therapy, because we know that's something that we can do. We have shown several times that in patients with tumor cells in the CSF, the OS is better if you use intrathecal therapy. The question is, yeah, with all these new attractive drugs, what about intrathecal LAG-3? So there is this trial also conducted now at MD Anderson, also by Isabella Glitza. And so the question, once we would have determined the best regimen with this phase 1-2, I think, again, it's feasible. We should plan also a randomized trial for this patient because I think this is absolutely needed for the community to convince people to use it. So yeah, the enrollment is close. So now we will have data for 9 patients at the recommended phase 2 dose. So it's already interesting, and we had no safety issues, I must say. So, and of course, we will need more information and more to continue, in fact, the effort. And we are working on the publication, so hopefully soon. And also, we have translational programs that will be analyzed also soon. And that's it for me.
Thank you so much, Emilie. So we move on to the discussant, Johanna Magner, and you will discuss the trial. Thank you.
Good. Good afternoon, everybody, and thank you very much for staying so late. And it's really my great honor to discuss the ITIO clinical study. So brain involvement represents one of the most the most challenging complications in patients with advanced disease. And in terms of melanoma, 40 to 60% of patients with metastatic melanoma will be diagnosed with brain metastasis during their course of disease. And 80% of patients that die from melanoma will have brain metastasis. Leptomeningeal disease complicates a little bit more with significant shorter survival and poorer outcomes in patients compared to patients with parenchymal disease only. And historically, these patients have been excluded from clinical trials, also from trials from brain metastasis mostly, and representing a high medical need. One of the most important steps forward has been the investigation of intrathecal immune checkpoint inhibition. And this first in human study, phase 1b study from Isabella Klitza from MD Anderson, investigated the concomitant intrathecal and intravenous nivolumab in 25 patients with leptomeningeal disease from melanoma. Primary endpoint of the study was the safety. Recommended doses, as you heard, was intrathecal 50 milligram and IV 240 milligrams every 2 weeks. And the median OS in these patients was 4.9 months with 26% of OS rates at 1 year. So building onto this concept, the ITIO study investigated the combination concurrent intrathecal and intravenous immunotherapy with nivolumab and ipilimumab in patients with non-small cell lung cancer or melanoma that had newly diagnosed, confirmed or probable leptomeningeal disease. As discussed before, there was no whole-brain radiation allowed, and also no planned or prior craniospinal radiation was allowed. So this was a very well-designed multi-center study across Switzerland. We had only 12 patients with melanoma and 7 patients with non-small cell lung cancer, but in total 19 patients, quite substantial number to draw some results. The recommended dose was 20 milligram ipilimumab intrathecal and 50 milligram nivolumab. Of course, it was only phase 1b study, so the primary endpoint is always safety in phase 1b studies. And the most awaited translational analysis is still ongoing. So here I have summarized the results of the study. 19 patients in total, 12 patients with melanoma. We have no information so far on the median duration of treatment or prior treatments. We have no dose-limiting toxicity. We had some serious adverse events, but they were not correlated to the drug, with the median OS in total of 3 months, and for melanoma, 3 months, and the 6-month OS rate of 20%. I need to say that cross-trial comparisons need to be made with caution, but however, I think it's important to have a look a little bit at the results from Klitza and Toll from MD Anderson. So in total, we have 25 patients. The median duration of treatment has not been reported also in this study. And the median prior treatments were 2. Again, there were no dose-limiting toxicity. And in terms of efficacy, again, it was only a secondary efficacy point, secondary endpoint. It was 4.9 months with a 6-month OS rate of 44%. So the question, of course, that raises is why we do have the difference. It's only actually study design, it's only patient inclusion criteria, or is it because ipilimumab probably does not add to intrathecal nivolumab? So to conclude, I think we can say that intrathecal application of immune checkpoint inhibitors is safe and an established therapy in patients with leptomeningeal disease. However, the best intrathecal approach, where is anti-PD-1 monotherapy, combination anti-PD-1 CTLA-4, or the new combination anti-PD-1 LAG-3, I think remains undefined. And as Emilie said before, we really need randomized trials in patients with leptomeningeal disease. And what is also clear is that patients with brain metastasis or patients with leptomeningeal disease, they really need a multidisciplinary management. And this is how we do it in Zurich. So we have developed or we have established a weekly melanoma brain metastasis board where we join every week and discuss our patients together with radiation oncologists, neuro-oncologists, and neurologists and neurosurgeons in order to define the best local therapy, the best sequence between local therapy and systemic therapy, and of course the best systemic treatment. And at the end of the day, I think what is really fundamental in terms of moving actually the field forward is to build a research network. And we're very happy that we have the brain evidence trial open. So this is the best one of the first personalized approaches in patients, pharmacoscopy approaches in patients with brain metastases. And with that, I would like to thank the entire dermato-oncology team, Cancer Center Zurich, and of course the collaboration with our neurologists. Thank you very much.
Thanks a lot. So Emilie, please come forward and we have some time to discuss. Thanks.
Could I just ask about the whole brain radiotherapy? Because verbally you said it wasn't allowed, but in the inclusion it said prior was allowed as long as it was completed a month before. Would the leptomeningeal disease have been diagnosed subsequently? I wasn't present at the time of whole brain radiotherapy. Maybe that was done for parenchymal metastases, because I think it could still continue to work, obviously. So it could be a confounder.
So initially, in fact, we had windows of 3 months, and then in the next amendment, we went to a windows of 1 month prior to inclusion in the protocol, but it had to be a newly diagnosed, meaning that at the time of previous whole brain radiotherapy, the diagnosis was not made. Yeah.
Just to comment a little bit on the whole brain radiation, maybe we have some radio-oncologists here, they might not agree with me, but I think also in the new ESMO guidelines, the whole brain radiation, I think for melanoma is really dead. So I don't think that we really need to discuss the effect of whole brain radiation. As I said, we have radio-oncologists here, and they might really disagree with my opinion.
I agree with you. Yeah. No, I think we can do better than whole brain now. Yeah.
So, not particularly in the study, but in your board, do you take in consideration, is there a cutoff for example, the opening pressure where you decide this patient can do intrathecal or not? Like, is there does that help in guiding you who can get this type of treatment or who cannot?
So we are sitting together, in fact, at the melanoma tumor board for the part dedicated to CNS metastases. And so we look at all the items together, so the clinical assessment, MRI, so cerebral and spinal, and we do lumbar puncture. So we plan when we have a doubt on a brain MRI, we say, okay, we should complete the LMD workup, and this is done in collaboration between the dermatology team and neurology team. And once the workup has been done, then we decide if there is an indication for intrathecal therapy or not, and this is done in collaboration together.
She wants to know about the opening pressure. We don't care.
No, the opening pressure, no.
You don't care.
No.
Train CSF, the pressure is down.
Yeah. No, sorry, I've not understood your question.
Yeah, Emilie, very interesting trial. And as you already stated, nivolumab ipilimumab is not standard of care anymore for non-small cell because the EMA withdraw the approval. But we have now nivo-ipi short dose of chemo, or short course of chemo, or even prolonged chemotherapy. Do you think there is a role to also evaluate the addition of intrathecal chemotherapy, and especially pemetrexed in this patient population, or not?
I think we could discuss, and I think that we need to have more data, and at the end we need to define which is the best intrathecal agent that we want, because we saw that in breast cancer with this old agent that was liposomal cytarabine, so it's very old time, and we had positive trial with prolonged LMD PFS. So I think there is really a role for intrathecal therapy, but I think, of course, here that's phase 1, so we took everything, but once we have a dose, I think we should go per organ and try to do
and interesting, I think in the trial, more patients, so the non-small cell lung cancer population, they had actually higher OS. They had actually 4.5 months, despite actually withdrawing combination immunotherapy. So, but there were very few patients, right? We had only 7 patients.
Yeah.
So,
just to also comment, because you show the comparison with Isabella Glitza trial. So also we had more patients with positive CSF at baseline, and if you look also at the graph of Isabella in the publication, you see the long survivors. There are also patients without tumor cells at the beginning. So I think, yeah, we have to, yeah, but it's nice that you show like that. We can discuss and, yeah.
Thanks. More questions? Allow me one question. I was a little bit puzzled by the distribution of melanoma and non-small cell lung cancer, because I was thinking that you would have more non-small cell lung cancer patients included in the trial. So what kept the lung cancer patients from enrolling in the trial?
Can we skip the question?
Is that a question that is allowed, or?
It's a political question.
Even data is understood. That's politics, and yeah.
No, let's put it positively. We have a very famous melanoma center.
Yeah.
And that makes everyone.
Yeah. Thanks. Just from Tuxedo 3, because they're lung cancer patients enrolled, I was wondering whether there was anything in the protocol that you would deemed problematic. But politics is something that we should keep silent about. So, having said that, any more questions? Otherwise, we're a little bit advanced in time, so I give over to Matthias and Emilie for the concluding remarks for today.
Thank you. Yeah, not much to say. I hope you enjoyed the meeting today as much as I did. I thought it was an extremely interesting day. We covered many topics in basic translational and clinical research, and we'll keep on doing so tomorrow. We will reconvene tomorrow at 8:30, if I'm incorrect. And maybe you can give us some more housekeeping remarks. Thank you.
Yeah, thank you for attending. Yeah, we can close. I was just asked for the speakers' chairs if they can come here for a photo from today. And yeah, and otherwise have a nice evening and see you tomorrow at 8:30.